of participants throughout the day. So we're really excited and grateful to have you all here. We look forward to the shared learning experience that we'll get throughout the day. We're going to kick things off with an announcement of a visionary new partnership between Pitt Health Sciences and Vizy Incorporated, whose aim is to catalyze a shift in how we think about and how we deliver healthcare. So we'll start things off with a brief welcome from Dr. Anantha Shgar, the University of Pittsburgh Senior Vice Chancellor for Health Sciences, and the John and Gertrude Peterson Dean of the School of Medicine. He'll be followed by Joan Gable, our Chancellor of the University of Pittsburgh, who also will be with us this morning to welcome all of you and to celebrate the partnership. So, Anantha?
Thank you, Mark, and thank you, everyone, for gathering here this morning. It's wonderful to see all of you to announce this exciting new partnership and several launches from Pitt. So first of all, I want to thank all of our leaders, including our Chancellor and the Chair of our Board of Trustees, John Verbanek, and many wonderful supporters and foundations and city. Saw Sam Ryman somewhere. There he is. From R.K. Mellon Foundation, and I know many other foundations are really behind this great initiative. What we're going to announce today are, first of all, an amazing partnership, and I will talk a little bit more later about some of the things we're going to be doing. But we're also announcing two major commercial initiatives that are joint between University of Pittsburgh and Vizy Incorporated. Vizy and many of the other guests here, I will introduce in a second, are here to partner with us in creating the next generation AI-driven transformation of healthcare, the next generation molecular medicine-driven transformation of healthcare. So this is all about not only great research, great understanding of human biology and human diseases, but also using cutting-edge technology to create not only better understanding of our diseases and health, but also provide better care to maintain health. So it's about not only transforming healthcare, but it's actually disrupting our currently siloed, unaffordable, and poor quality healthcare. So that's really what we are here to do. And that is the mission of Pitt. And we want to do this at global scale. And that's what we want to announce today. There will be two platforms I'm going to talk about in a minute, but then also some of the amazing things that Pitt brings to both our community, but also to the world. I want to particularly recognize Vishnu Vardan and his sister Harsha, who are the co-founders of Vizy. And Vizy is a company that is not only an AI company, but it also has a proprietary large language modeling, which means that Pitt will have a proprietary AI capability, that we don't have to depend on the tech giants. We don't have to depend on Google. We don't have to depend on ChatGPT. We don't have to depend on DeepSeek. Pitt will have its own AI, and it'll be dedicated to healthcare. And that's the first thing I want to emphasize. And thank you, Vishnu and Harsha, for bringing that to Pittsburgh and bringing that to healthcare broadly. I also want to recognize a couple other guests here, Dr. Mani and Seema, if you want to stand up. These are two primary stakeholders in L&T, which is an Indian conglomerate that has multiple business units, but they are particularly in charge of their technology and AI and building infrastructure like data centers, et cetera. So they will also be anchoring L&T AI programs here in Pittsburgh, and we have many different things that we want to talk to them about. And I think there will be other stakeholders and speakers that you will see during the symposium today, including distinguished scientists like Leroy Hood and Adil from UK, and many others that you will meet today. But the idea is to bring greatness and build great products that will serve not only our folks, but also globally to improve healthcare. So I'll talk a little bit about the two platforms we're going to announce later, but first I want to invite our Chancellor, Joan Gable, to make a few remarks and then formally sign our agreements. Thank you.
Thank you, Anantha, and good morning, everybody. It's really lovely to see you all here. We're very grateful for the opportunity to host you today because it is a very important and meaningful day. It's an important day for the university, for our partners. Really, it's an important day for Pittsburgh. It's also an important day for every patient who has the opportunity to benefit from the outcomes of the platforms that Anantha briefly described and that all of our experts will go into more detail on through the course of the day. We have a unique capacity in this community of scholars and supporters and friends and partners to lead the life sciences century at the intersection of health, AI, and innovation. And that's a wonderful thing to say behind a microphone. But what it really means is that our patients have a better chance. And that's really what all of us are about who do this work, either directly as clinicians and health scientists or indirectly as those who are very happy to see this take place and to facilitate the possibilities of what might come from the other side of this. We're playing to our strengths here as a community. We've been rated one of the top 20 most innovative universities, and there's a reason why that means that there are things we can do here that can only happen here. But at the same time, without the right people coming to the table and giving us this opportunity to work together, then these things become more aspirations than realities, and today is a reality, which is really incredible. There are challenges that we are all facing, but no one more so than the patients who are seeking access to the most innovative ways to receive treatment and care. And this is a problem here, it's a problem across the world, and so we can center the problem here and then solve the problem across the world in ways that have real meaning for individual people, and then also in the aggregate for society as a whole. And what could be more tied to our mission as a research university at the epicenter of health sciences? So we're especially proud to be the first participants and beneficiaries and stewards of this global initiative. And so I'll say on behalf of the University of Pittsburgh, I want to extend my sincere appreciation to all of you, but especially to Vishnu Vardan and Harsha Vardane for their leadership and partnership and what this can mean for all of us. So thank you, everybody. Thanks for being a part of this. And of course, hail to Pitt.
Thank you, Chancellor. I'd now like to invite Drs. Shgar and Dr. Vishnu Vardan and Harsha Vardani to come up for the signing ceremonies. At this stage, they'll be signing two agreements, one launching GainMed, and you can see behind me the description of that, and the second launching the Pitt Vizy Longevity Labs. After the signing, Dr. Shgar and Dr. Vishnu will give you brief descriptions.
Why don't I describe what we're signing so that by the time he gets the documents from maybe they were in the health sciences office, we're not sure, but we will get this done. So let me talk a little bit. Do you want to sit down? You want to sit here? Yeah, that would be great. Yeah, that would be great. You can sit there. So the two platforms that we are launching today, and these are both commercially facing but academically run entities. So they're true public-private partnerships. One of them is the GainMed platform. GainMed platform, it does stand for an acronym, but I think we'll stick to the GainMed. But for those of you who are curious, GainMed is actually an acronym for Generative and Agentic AI-Navigated Multiomics Medicine. So that's really the concept here. And the whole concept is, how do we characterize humans, and particularly patients, in the deepest biological way through multiple molecular omics? And these include not just genomics, but genomics, transcriptomics, exome, proteome, lipidome, as well as microbiome. So essentially, this means that we understand the human biology at the deepest level, and then we overlay on that a person's medical history, their illnesses, their treatments, etc. So that's the classic medical records and medical history. But then on top of that, we also have field-generated mobile app data. These could be things like continuous monitoring of heart rate, blood pressure, and various other biological parameters. So this is a combination of all of that for deep, I would say, characterization of the molecular as well as physiological and physical aspects of human person. And then using that to create not only decision support for physicians, but really decision and behavioral support directly to patients and their caregivers, and create value-based outcomes for the payer. So it really connects all three key components of healthcare and uses agentic AI and big data analytics at every one of those stages. So a patient would be told, based on their genetic makeup and their history, whether it's good for them to have certain types of diet, or whether it's good for them to have certain types of medications, or is it good for them to have various types of activities. All of those can be agentic at their side, so they will have an ongoing nurse practitioner on their phone, if you will. Same way, doctors will have in their offices a continuously learning system that will individualize patients' information, individualize patients' problems and care pathways, at the same time digesting the massive amount of medical literature, massive amount of molecular information about that person, and providing real-time cues to physicians and providers, whether it's a nurse practitioner or a pharmacist or a person who's doing their physical therapy. The third component is population health. Then the aggregation of these disease cohorts and aggregation of their practices would provide payers incredible value to save both costs but improve care. Today, for example, 100 people may get GLP-1 inhibitor, so Ozempic, at thousands of dollars a person. 40% of them will not benefit from it. And we will only learn that after 3 to 6 months. So what we have done is 40% of the time we have added extraordinary costs to society with no benefits to the patient just because we have to do this trial and error method, as opposed to precision-focused methods. And that's the kind of stuff that is going to change how payers will behave in this new environment. But what is extraordinary about this is because of the technology, this is easily scalable to not just 100 people, not just 2,000 people, but 20 million people. And imagine the cost reduction at that scale that we can do. And that is the real concept of PittMed. And this is going to be a joint product of Pitt and Vizy, and we will initially start with 20,000 subjects with healthcare partners in multiple segments of society, including the United States, UK, Sweden, Turkey, South Korea, and India. So that's where we're going to get the initial cohort of 20,000 people to build all of these models, refine physician behavior, refine payer behavior, and all different payer types, U.S. payers versus national health systems versus completely fee-for-service as in India. So that's the whole concept of PittMed. And we think this will be a globally transforming platform. And at some point, we will make this available to every patient, hopefully, in every community. So that's the first product we are launching. There are wonderful partners, Vizy, of course, L&T, and many others that will be behind this. And this will be entirely focused on healthcare. And not only is it entirely focused on healthcare, it is completely free of any tech giants' patent-busting activities, because we're going to have our own LLM. We will have our own cloud servers with friends with L&T. We'll have our own data centers that would be able to provide the GPUs, and hopefully Sam and others will help us with the power supply for that. But everything will be in Pittsburgh, and that's the concept. We want it to be soups to nuts in Pittsburgh and launch a global initiative that's going to transform the way we do healthcare. So that's GainMed as the first product. So thank you. And thank you, Chancellor, for making all of this as part of our mission. So, the second concept is, of course, in order to do all of this, it's not just about technology. We also need deep biology. And how are we going to do the deep biology? Part of that is building a laboratory that can do this at scale. And that's really the Pitt-Vizy joint molecular multiomics laboratory. And again, this would be housed in Pittsburgh. It will be able to do, as I said, 9 different types of omics. We have partnership with Illumina. I don't know if she is here. Oh, there she is. Yeah. She's the vice president of their multi-omics platform. They are going to be partners with us, as well as for other areas of lipidomics and other types of molecular omics. We have Thermo Fisher as our partner. I'm not sure if there is anyone from Thermo here. But the idea is that we will create this lab based on, of course, the great bioinformatics as well that we have here. This laboratory will be an initial investment of $50 million, both from Vizy and, of course, in-kind and other types of talent that we bring from Pitt. And we'll be preparing to do 1 million people sequencing over the next 3 to 5 years. That's a massive sequencing effort, and it will involve additional labs. So this Pitt-Vizy lab will have 3 additional laboratory sites. One is going to be in Turkey for the EU region. The other one's going to be in South Korea for the Pacific Rim region. And the third one is going to be in Bangalore for Asia and India. And that would all be part of the hub and spoke from Pittsburgh to global laboratories. This is also because many of these samples, it's very hard to get across national lines and across continents. So we need to also develop these labs in individual continents. So that's really the concept. And that would be the database from which all of this learning and all of this knowledge and all of this caregiving will happen. So again, these are the two big initiatives we're announcing today. And this would make Pitt not only a global leader in transforming healthcare, but it will also someday say, people will say, hopefully, that the transformation of modern healthcare began in Pittsburgh. And that's really the proud point for all of us. So thank you, and we are very much looking forward to the next 5 years. And all of the details we'll hear more in this symposium. So thank you very much, and maybe... Perhaps we have something now? Yes. Thank you.
It's a docu-sign, so this is
Yes.
ceremonial.
That's fine. So I'm just going to ask Vishnu to make a couple of comments, and then we'll break up for the symposium.
Yeah. Before I start anything, I think I feel that when you are trying to do something big than what a single person can even dream of, things have to happen as per the destiny. So it was a destiny that I met Professor Vanithy sometime back in India. She had come for a talk, and that's where I understood that Pitt has amazing bioinformatics department, which can do a lot of work on this, and that's how I came to Pittsburgh 2 years back. And I really liked what they were doing, and we started a center here for women's health on my mother's name. And again, she has come back, my mom, to launch this Vizy lab. So Vizy is an Americanized version of my mom's name. Her name is Vijay Lakshmi, and I started this company to just cure her diabetes. That's the purpose of that. I didn't have any other purpose. I just wanted to see that my mom was diabetic, and I said, like, as a doctor, what can the best thing I can do? Is it like just keep on giving her medicines for next rest of her life and fighting one complication or another, or attempt something, which I can change it and then prevent her diabetes or stop her diabetes? I didn't have even a word for that. So now it's been 5 years. She's no more a diabetic. She's not even insulin resistant. So her HbA1c is like 5.1. Since last 5 years, it's continuing to that. And then my sister said, why did you only do for one mother? Why can't we do for... There are 1 billion diabetics in the world, and India has perhaps highest in the world. So can we do something for this? And that's how Vizy started. But once we went deep, we realized the limitation of doing it one person at a time. Without technology, without scaling, global scaling, this is not going to happen. I could have treated maybe perhaps 1,000 people in my whole life if I had done one by one. That's the reason we went behind technology. So we didn't say that, okay, we both are from MIT and let us invent a technology and go and build a business. We said, what technology can we bring to solve this problem? We didn't take a technology and then go and try to build a business. And that's how we started building the whole AI platform, generative AI. We are the first company in India who have built our own ChatGPT kind of model. But I didn't want to just make it another portion of local ChatGPT. I said, can we go deeper into healthcare and then solve this problem? Because healthcare is one domain, if you look at the business, but healthcare is all humanity. Everyone needs healthcare. There are only two things which everyone needs. One is healthcare, one is education. And we said, like, why can't we just build generative AI, and whatever AI you call it. We didn't even call generative AI at that time when we started. We said, let us build for all the people on earth. And this was technology part, but I have to tell another amazing story. So I've been a big, you can call me a fanboy of Professor Leroy Hood. He doesn't know me till this year, but I knew him since a long time. What he was trying to do is P4 medicine, is data cloud, a lot of work I used to read, and I used to tell my sister that we should really do something like this, where we can get all the possible data of humans and then create a perfect model for a human being to be treated rather than a population medicine, right? If you're a diabetic, you have only 5-6 classes of drugs, the new one is Ozempic. Everyone gets that. That's population medicine. We said, why can't we create a model which can just deliver what you need based on many things, your biology and also what you are exposed to, where you live, what is your lifestyle, what is your understanding also. Based on all this, can we do something? He has done an amazing job on that, but I used to say, can we scale this, can we build this? And that's how we started thinking that we should build a platform for multi-omics. And that took me in two different directions, which made us a company which is uninvestable in traditional sense. I'm chasing two things which are like totally different things, and then trying to build a company, which is very complex problem from anyone. If someone looks at my stacks, like when I proposed this in MIT 5 years back, they said, this is impossible. As a business, it will never come to any stage. You'll be just, dreaming. Don't do this. That was one of the top professors who has built so many unicorns told me that. So my sister was in tears. We had one MIT 100K prize, which is one of the best prizes for innovation. But this is the feedback we got, that you were chasing a chimera, which is not even possible. But we didn't stop there. We went on doing whatever we want to do. And that's when I met Dr. Adil. And we just hit in one Zoom call. I was amazed that what I was trying to build in multi-omics, he has done amazing work on that already. Built a lab in Turkey, worked on a huge population. Virtually, he's the guy who has done this in real. Then we said, why don't we collaborate on this? So we said, let's build these labs across the world, do 1 million people's data, and then bring this biology model. Because I have AI, you have multi-omics capabilities. Let us build together. And then we have been become partners after that. And all this was happening on one side, but there was one person who was had a vision that we should transform healthcare. I never see that from a place which is built such beautiful healthcare institutions, has its own insurance company. There is nothing which needs to be transformed here, because this is the place which is in the top of the world. Why does anyone want to even transform themselves when they are doing so well? That's what Pitt is. But when I met Dr. Shekhar, I really was surprised that this is a person who has everything going so well for him, but still he is not satisfied with that. He has a vision that, I call it as Framingham 2.0. Framingham was only about cardiovascular health. This is about the whole human biology. We were all there working on the background, but this was the vision where I needed an anchor. because US is world's #1 healthcare business, market, everything comes from US. Though we all say that US healthcare is very expensive, I have another take on this. I have written a paper in MIT on that. US healthcare is very expensive because all the US taxpayers are funding the research for healthcare in the world. And India doesn't do much research. I don't think we've spent anything on research in healthcare. But Ozempic also comes to India. This GainMed platform will go to India. So virtually US taxpayers are really doing a lot of research, paying that, and that is used by the globe. So that's the greatest. That's why US healthcare is also expensive, because all that goes into your bills, your premiums. And we said that, okay, let's transform US healthcare, then we can transform the globe, because ultimately US healthcare is what teaches everyone in the world like what to do. So, and we needed an anchor place, and we got Pittsburgh, and this has happened. And I'm very excited today that all this is coming together. This is something like a dream come true for me. Stage 1.0, because this is just the beginning. It's not like we have done everything. We are just starting to do it. But I'm sure that with such a great place and all the great people getting involved, we will surely achieve something which we have been dreaming of, transforming healthcare, because it affects every person on earth. It's not like a business which will be like only helping few people, because healthcare is everyone's right, and everyone needs healthcare. And thank you, Dr. Shekhar, and thank you, Chancellor, for giving us this huge platform to start what we had dreamed. And again, want to thank Dr. Leroy Hood. He has come here. He's going to deliver the keynote. Yesterday, he was speaking about like half an hour in our room, and I didn't even speak, because when he speaks, I just listen, because whatever he speaks is like something which I will be noting in my brain, that this is exactly how you see the future, how you plan for it, how you do it. And again, Dr. Adil, we are like going to be like, we both are the super excited guys in this whole room, because we are going to build a biological model for 1 million people across the world. This is something amazing. And we have so many other colleagues. There is one colleague who is sitting on the back, on the stairs. He has come from India. So we have L&T, who is like been great support for me since last few months. Mr. Sunil, he was the first person who gave us GPUs in India when we were trying to build our models, because it has been a contribution of a lot of people, and most importantly, my family. My sister is my co-founder. I have another co-founder, my brother. So virtually he was in some other business, but he saw what I'm doing, and then he has also joined us. And my mom, who has been the inspiration behind this whole company, and also me becoming a doctor. And my wife, who bought me this suit, because I had forgotten to buy a suit for this great occasion. She went yesterday evening in the night and then got me this. I asked her, how did you buy a perfect suit? Because I didn't even try this. She said, like, I know what size you have. So I got it, even the shoes. So thank you, Swati. And my daughter, on whose name my company is, but she has some problem. Whenever she sees a crowd, she doesn't enter that room. So she always misses all the programs which we do, because she will not be there. She'll be outside somewhere. And thank you, everyone. It's been amazing in Pittsburgh since the time we came here for establishing Vihar. And we hope that it will be only going bigger and bigger. And great things are going to happen in the world healthcare through Pittsburgh. So that's, everyone asks me, like, what does Pittsburgh gets from this? I say that Pittsburgh, again, will make history through healthcare. Thank you very much.
Thank you, Vishnu. I'm going to have 1 minute for Adil to come and say something about the lab. So you want to come on up?
First of all, thank you so much for organizing such an event and preparing this entire day for this very exciting symposium. In a couple of minutes, we will actually have we will start for Symposium, and I will be telling you a lot about what we've been doing. But for this moment, I would like to take the opportunity for thanking the Chancellor and the Dean of the Pitt and also all the Vizy team. To be honest, we've been working for this more than 3 years, 2.5 and 3 years, and we are very excited for this start. So we look forward to completion of the project. Hopefully, we will be meeting for the next event for where we will be actually celebrating the first bunch of results that we generate here at Pittsburgh. Again, thank you so much for the time and for organization.
Thanks, Adil. So something this big and this complex doesn't happen without a big team. And I want to thank several people here for making this happen and coordinating, organizing, bringing together lots of itineraries, lots of agreements, and number of collaborative opportunities. So I want to start, first of all, by thanking my great team, Jeff Bees, Leanna McKibben, and Udua Kendo. These are the IT lead, the finance lead, and the operations lead. They have helped us bring this to this point. So thank you very much. It couldn't happen without all of your hard work. I also want to thank several of the Pitt officials, particularly the tech transfer, research operations, data management, various types of extraordinary talent we have. So I want to thank Evan Thatcher and Human Rashidi, as well as Wanati and Mike Vizy is here somewhere. There he is. So all the bioinformatics infrastructure we have here at University of Pittsburgh, which will be the core competency of a lot of the work that's going to happen. And then finally, as Vishnu said, every person, every half-functional man needs a very strong woman who can actually find the right suit for him, right? And so I want to thank Gina, my wife, for finding the right suit for me, as well as supporting me in everything that we're doing. So thank you all very much. It's a wonderful occasion, and I'm very happy to launch this great effort and hopefully will make Pitt a household name globally. Thank you.
Yeah, we'll be going up
at 10.
Yeah, we'll be going downstairs to Ballroom A. We'll start the symposium at 10 o'clock. You have a little bit of a break, and we'll get upstairs. You said downstairs. I listened to him. You said downstairs. Let's go upstairs. We'll see you at 10.
Nothing like a vice dean who doesn't take responsibility.
Ahead and get started. Please take your seats. All right. Okay, I'd like to welcome everyone back for the presentations. I have the enviable position of not only emceeing this event, but trying to keep it on time. And so we'll be introducing each of the speakers. We will leave a little bit of time for questions. There's a microphone at the front, so if you anticipate a question, you may want to line up. And we'll try to keep everybody not only participatory, but on time. So it's my distinct pleasure to introduce to you Dr. Leroy Hood as our keynote speaker. Dr. Hood is the Chief Executive Officer for Phenome Health. He's also the co-founder and professor of the Institute of Systems Biology and the Chief Innovative Officer and professor at the Buck Institute for Aging. So without further ado, we'll bring Lee up.
Well, it's a pleasure to be here. Data-driven health, I think, as you'll see today, has really reached an enormous tipping point, and the opportunities are really going to be magnitude. And what is striking is it's the integration and fusion of big data, of systems biology, of AI, and something I'll talk about at the very end, of informationally based drugs that can be done at scale. I'll talk first about a little history in this field. When I went to Caltech in 1970 as a young assistant professor, I kind of was interested in human complexity. And the question is, how did you deal with it at that time? And I think it was clear we needed to do big data. There was not the technology to do it. It was clear we needed some way to take all of this data and make sense of it. That later became systems biology. And my career then passed through what I'll call 8 paradigm changes, each of which changed biology, but at the same time it led to the next direction that one had to take. So initially, we developed a series of 6 instruments for reading and writing DNA, and one of them was the automated DNA sequencer, which got me invited to the Human Genome Project meeting, the first in 1985. 12 of us passed judgment on it, and it was A, agreed it was feasible, and B, it was split 6-6 on whether it was a good idea. And those opposed were terrified that big science would take over and destroy small science. And of course, that didn't happen. The genome then gave us the ability to look at an individual and say what is the potential of their health trajectory in the future. What soon became obvious is my lab got very large because of all across disciplinarity, and that was an anathema to the Division of Biology at Caltech. So I proposed that we create a cross-disciplinary department where all the expertise was embedded in faculty members. And biology at Caltech categorically refused that, and Bill Gates made it possible to go to the University of Washington and start the first such department. And it was spectacularly successful in the 8 years it existed. For example, 2 of its faculty members, Rudy Abersol and John Yates, pioneered the first 2 major techniques in proteomics and then went on to be leaders in this field. And we developed the inkjet technology for very rapid DNA synthesis, which we'll talk about with the drug company when I come to later on. But it was clear in a big institution, something as dramatically new as systems biology, which I was then pushing, was absolutely paralyzed by bureaucracy. So I quit. and went to start the Institute for Systems Biology. And I had a wonderful meeting with Bill Gates, who said, "Why are you leaving?" And I said, "Because I need to start something new if you're to create something new." We'll come back to that later. And he said, "Well, how are you going to fund this?" And I said, "That's why I'm here." And he said, "You know, Lee," I knew him pretty well then. He said, "You know, Lee, I never fund anything I think is going to fail." And that's how we started the Institute for Systems Biology. And of course, what it did was take this global holistic approach, and one with Trey Eidegger, we applied it to the yeast galactose system, and showed dynamically you could explain with biological networks the biology of that. And we did actually the very same a little later with prion disease in mice, where you could initiate it and then follow everything that happened. And what was marvelous about systems biology in that case is it beautifully recapitulated the 4 major stages of the disease in mice that occurred over a couple of month period. From there, I proposed this idea of P4 medicine: predictive, personalized, and participatory. Those 3 are science. The 4th P, the participatory, is how do you persuade the world that this new type of medicine is going to be possible? And that's an educational problem, a sociologic problem, a psychology problem, and mostly, in many ways, it's an economic problem. And we think we can solve most of those things, although it is the most challenging of what we have to do. From that point, I proposed P4, and I found a whole series of the things we wanted to do in technology got turned down at NIH because they were unrealistic and too far in the future, and I hadn't proven that they worked. So I was able to persuade Luxembourg to give ISB $100 million over a 5-year period. We developed all the technologies, and they framed the possibility of then creating a data-driven healthcare. And of course, the Luxembourg project made it possible to bring my colleague Nathan Price to ISB, and he pioneered together the data-driven science that we'll be talking about here. So the idea was you could actually take data, convert it into actionable possibilities, and improve health. And that led to a company of 5,000 where we, over 4 years, really reinforced that possibility. In 2016, I took on Chief Science Officer of Providence with a mission to bring genomics and scientific wellness, as we called it, to a major healthcare system. And I realized, again, bureaucracies are utterly intractable for really new ideas. So I started in 21 Phenome Health, and that's been the driver for doing this thing. The final paradigm change we'll talk about very briefly at the end is to bring in information-based drugs, because this approach to health and prevention opens up hundreds and thousands of new possible drug targets. And the question is, how can you take advantage of those opportunities effectively? The major conclusion I drew from all of this is 7 out of 8 of these paradigm changes were really catalyzed by small, nimble organizations. And you can do that in the context of nonprofits, or you can do it in the context of for-profits, and they do very different things. Nonprofits can do conceptual new things. For-profits focus on opportunities and commercialize them, and so forth. And I think a really relevant question is to ask why health is so fundamental. And it really is fundamental because it allows you to attain your own potential with development, with proper health for education, jobs, community, work, creativity, productivity, and all of these different kinds of things. This data-driven health leads to these actionable possibilities that converting data into opportunities that make it possible to get started on this trajectory, and you can start at any time you wish. The ultimate objective is really to extend our health span out into the 90s or 100s, and that is a remarkable extra 20 or 30 years of being creative if you have purpose in your life, and that, I think, is one of the really interesting challenges. And of course, this enables then the optimization of the individual, and it gives society 30 years extra for each individual in a society where we don't replace population in any developed country these days. So the genome, as I said, sets the framework for health. And of course, the whole important question is, if you set the framework for what you can do, what is the actual phenotype from birth to death that you choose? And that's really determined by two things, your behavior and your environment. And phenomics is the ability to measure the response of those two activities in the context of your life phenotype. So blood analytes, gut microbiome, digital health, et cetera, are all possibilities. Now, what is really key if we want to see this move into society rapidly is what we'll discuss today, a global 1 million person phenome initiative where we have the data over a period of 5 years to show improved health and decreased costs are the hallmark of this kind of opportunity. How do we handle this whole big data? Well, it's the idea we can take the informational molecules and place them in the context of a human being. And one of the first ideas that emerged from the Luxembourg project was the idea of blood as a window into health and disease, because it gives you the ability to look at the information secreted from each of your internal organs in the blood, and its quantification gives you an idea of state changes in all of these organs. So it gives you a global assessment of how you respond to disease, not just in a molecule, not just in a cell, but in a larger level system and the integration of larger level systems. The digital data, for example, that we took from the 5,000 Airvale individuals over 4 years then could be handled in two ways. One, you could map it into the networks and communities that came from systems biology that dynamically reflected normal physiology and disease physiology. And number two, you could use AI with all of the different tools, the knowledge graphs and the digital twins and large language models. And we'll talk very briefly about that. I think Nathan Price and Craig Mundy will discuss this at the very end of the symposia in considerable detail. So in Airvale, where we had 4 years, we did it twice a year, what kind of data did we generate? There were 6 different data types, from genome to blood analytes of 3 different types in the gut microbiome and a digital health and forms in the form of Fitbit. And these were the types of data that we had to be able to analyze individually and integrate together to give the various kinds of actionable possibilities. And from this, we've had more than 25 different papers. So once you get a large data set like this, or the 1 million people we have, it's enough for the entire world to operate on and each come up with original points of view in these large data sets and everything. So it turns out that there were 5 major insights from the Airvale Coalition cohort. One, there are 8 categories of genome variants, and each of them lead to enormous numbers of actionable possibility, thousands in some cases. Number 2, we could take the data of the 6 different types and make statistical correlations and map them into communities and networks, and I'll describe those in just a moment. And then finally, we could use these data to begin creating metrics that assessed different aspects of physiology, biological age, metabolism, frailty, all of those kind of things. And we could look at transition states, and I'll show you 2 examples of how we do that in just a moment. And then it made it very clear that the scientific wellness that we have is just the other side of the coin for healthy aging. They are, in many ways, when integrated together, one and the same kind of thing. And of course, what we have to be able to do is take the 3 higher level systems, brain health, body health, and your gut microbiome health, and integrate them together. And those are the kind of things that you'll hear about here. Here are the 8 classes of genome possibilities, and I'm not going to go through them. I'd be glad to talk about anybody who wants them. I've indicated the fact that there are tens of thousands of actionable possibilities. An incredibly small fraction have ever been used clinically in this regard. And the actionable possibilities really do 3 things. 1, they allow you to optimize wellness or healthy aging. 2, they let you repair the deficiencies that exist in your genome or your phenome, your behavior. And 3, they give you the ability actually to detect disease very early at a point when we can think about preventive measures for avoiding ever a clinical manifestation and so forth. On the statistical end of things, with the first 108 people we looked at, we got 35,000 correlations, data bits, and 2 of the 6 different data types that we had. And of course, the first thing that could do is it let you take these statistical associations, go to the literature and understand them, and then map them into the various kinds of biological networks that looked at. And of course, you could look at the statistics at different times during the development of wellness and see how things changed and all. And I think in many ways, equally important was the idea you could take this hairball of associations and you could use an algorithm called community analysis, which fundamentally asked, in this whole set of analytes, what are the most closely related and, in a sense, the highest statistical probabilities of association? And what we were able to see in the 108 people were 70 communities. And what was striking is that communities could be mapped into normal physiology and or normal pathology and so forth. And of course, what was really exciting about that then was the idea we could look at one community, the cholesterol community you see exploding out here, and it showed 12 associations with LDL cholesterol, a proxy for heart disease. And what we saw strikingly was thyroxine was really negatively associated. What was interesting is Lilly was just in the 3rd phase of developing that as a drug, which they went on to do. So this opens up an enormous number of drug possibilities. In the first analysis of the 108 people, we used 500 proteins. In the second analysis, we used 5,000 proteins. We got 10 times as many statistical correlations and enormous numbers of new communities, and the communities fused together in really interesting ways. And what's important about communities is they give different hierarchical levels of information association. The biological networks are one-dimensional in that particular context. We were able, for example, from the 5,000 to determine an algorithm for biological age, the age your body says you are. The lower it is, the better off you're aging and so forth. And the biological age demonstrated for women in Arafale, for each year they stayed, they lost 1.5 years of biological age, and for men it was 0.8 years. We were able to use this metric to assess biological age in major organs, and we were able to actually, from the metrics involved in creating this biological age, began to provide advice for how to optimize a particular individual's biological age. If you're sick, in every case we looked at, your biological age goes up, presumably returning when you get to be healthy and so forth. And what we can do with the three metrics I talked about, age and metabolism and frailty, is map these networks, the features of the networks, into knowledge graphs and connect them in an extended way with the known knowledge for each of those features, thus expanding enormously the horizon of how you can think about each of these metrics. And finally, I think what's really incredible about Arafale is for a 4-year period where we had people from 21 to 90, we saw the entire spectrum of human development. So 4-year analyses in chronic diseases, if you spread out the state of the individual when they go into the disease, can give you enormous new information, and we'll talk about that. Classic medicine, what happens is when you don't see early diagnosis of disease, and it progresses up to a point of where you can make a clinical diagnosis and so forth. In Arafale, we had 167 transitions from wellness to disease, and we looked at 10 that transitioned to different cancers and at the bloods prior to diagnosis. In all 10 cases, we saw elevated proteins that often beautifully mapped into the disease-perturbed networks relevant to the particular cancer. So this gives you a really good place to start thinking about preventive therapies in the future, quite obviously. And so with the phenomic medicine then, it'll be early detection, which we can do now, as I just described. And then what we can do is go to the networks and see which drug targets or behavioral targets we could attack then to reverse the disease before it manifests itself. And the hope here, of course, is in a 10-year period, we can make a dramatic reduction in the fraction of chronic disease, which in the US are 86% of $4.5 trillion. So you're talking about trillions of dollars in savings, and we'll come back to that. Health really has then 3 stages: health, optimizing wellness, disease and its progression and how you deal with it in precision medicine, and the transitions. They are important to detect and to be able to manipulate appropriately. And a second approach we can take that's really interesting is to recognize that your internal organs all have proteins that they make, 90% of which is in that organ, and in some cases they're secreted into the blood, so they're organ-specific proteins. And we were able to demonstrate in looking at enormously detailed transcriptional profiles for the organs that there were potentially thousands of organ-specific blood proteins. And they had really 2 interesting features. One, they mapped beautifully into networks. It gave you an idea of, if perturbed, what the system was. And 2, they often had architecturally specific sites and things like the brain, so you could localize where it is. So the really important point is the brain, and the placenta, for example, has 31 organ-specific proteins. And again, they appear to have locale-related specificities in interesting ways. And with a project we're doing here with Pittsburgh with Yoel Sadowski, we'll be able to examine the transition all the way across pregnancy in very early ways. As I said, systems biology and big data and scientific wellness provide us a window into health and disease, and there are all these different metrics we take here, but the approach is discovery-driven. Get a lot of data and try and figure out what it's saying. And that's absolutely in contrast to the geroscience approach of the Buck Institute, which focuses on the hallmarks of aging and specifies the molecular nature of those. And my argument is if we integrate together those things using AI, you'll get an enormous increase in the understanding of wellness and aging. And really, they're one and the same thing. They aren't actually different. And we can do that integration beautifully with knowledge graphs and ultimately the large language models. So let me just summarize this by saying what data-driven health does is allow you to extend your health way beyond what the conventional health companies do with clinical data and anthropometrics and electronic health records and so forth. And each of the different omics give you an enormous extension of actionable possibilities that bring in totally new dimensions for scientific wellness: the microbiome, the metabolome, the proteome, and now the wearable data and so forth. And of course, what these data allow us to do is to select from the tens of thousands of actionable possibilities, those that are relevant to you and your genome and your phenome. And in doing that selection, we've reduced enormously the dimensionality of the opportunities so that they are manageable for each individual. And that at the same time gives us then the possibility of responding in various ways to those actionable possibility to provide tailored N-of-1 optimization of health, a very powerful forward movement. And that's what it's, the N-of-1 healthcare is going to be really important in the future. And here are 25 of the publications that come out of that system. Where do we go next? Well, one thing we're really excited about is new areas of omic and other data kinds of analyses. So one will have new analyses that are 10 to 100 times more data rich than the airveil population is. And then we've added a whole series of specific things. Polygenic scores allow us to calculate the genetic risk for each individual. And we've shown that genetic risk can be identified as to when it begins to manifest the potential by looking at blood analytes. So if you're at high risk for X disease, your doctor just has to wait until those analytes come up. Then you know you're near a transition, you can do something preventive in the future. We're doing single-cell analysis at each blood draw to look at innate and adaptive immunity. We did this on a COVID-19 test of 300 people, and it was very powerful in what we could do there. And we're looking at a new multispectral analysis that our partner has shown beautifully can take saliva and in 3 seconds distinguish a normal from a COVID individual. It has 1,500 spectral lines. What they do, what they correlate with, we don't know, but we're going to try and make saliva a second window into health disease, looking at entirely different data collects and so forth. And there is a structure now called alpha sheets that give us the ability to do diagnostics on virtually all neurodegenerative diseases that have amyloid-like behaviors with different gene products. And I can talk about that in the future. And we'll talk about brain health next. And what we're doing is using studies that are pioneered by Michael Merzenich and a company called Posit that have the ability to assess 25 different cognitive features. And what he showed is the normal person has cognitive maximi in their late 20s, and then you decline the rest of your life unless you do special things. He showed the brain was flexible in that at 80 years, he could convert many people back to near their original potential. So it's hopeful if you do the exercises. And the Brain HQ exercises are easy and fun to do, and they can be used to train and repair deficiency. So it's a very powerful approach to brain health in the future, and we'll be doing that kind of thing. And two metrics that are coming out of Luxembourg now are voice analysis, where they've actually can take saliva, can take a cell phone, analyze your voice. They've done it for 40,000 individuals in various stages of diabetes, and they can use the voice patterns to classify individuals beautifully. And they've just started to explore that new dimensionality. And a really intriguing possibility that's come out of them is hair analysis. What they can do with a bit of hair is show whether or not you have any one of 150 environmental contaminants. And I think what's really cool is for each year, each inch of hair you have, it measures another year of contaminants. So with long hairs, you can go back and look at the history of your exposures. And what they've done in 14 different clinical centers in Luxembourg and surrounding countries is they can identify the geographical location of the individual from the contaminants. So it's a very powerful kind of assay. We need to have a computational platform that can ingest genome and phenome data and do the analysis that's compatible with the 3 different clouds that builds in the pipes to be able to take in a knowledge graph and digital twin data, and that is modular in a way that can readily be used by partners. And we have this, and I think we'll have to universalize this for the project you're going to be hearing about in a few moments. Our approach to using AI is to take the data we're interested in, genome and phenome, and map it into knowledge graphs. And what we map in is the unique feature, relatively unique features of any individual, and connect that to the basic knowledge of these areas. And with large language models, you can instruct them then and do ask them to classify and further extend the actionable possibilities so that we, in the future, will deliver actionable possibilities neatly categorized with regard to their functional classification for each individual and deliver them to the physician, making the physician in this partnership a domain expert for many different areas of medicine and enormously increasing the healthcare and so forth. We're doing some of the same things with digital twins. We're looking now 2 classes of genome-phenome opportunities. One is the first class, where we look at individuals and analyze their genome and phenomes, and this international million-person phenome project is one possibility. And then the second is to take the big data approach toward the 4 major chronic diseases, plus things, normal physiology like pregnancy. And we've started such a project on diabetes, and we're beginning to start another one on Alzheimer's at this stage. We recently got a ARPA-H grant with the Buck Institute for $52 million to essentially analyze fundamental features in phenomics, the rhythms of analytes expressed in the blood, the need to be able to blend together different large phenomic data sets, the ability to look, in our case, we chose diabetes, at a 15-year study where they had enormous clinical data every 3 years. They showed 70 transitions that we could follow, and we're going to do the omic analysis on that and get the entire life history. We're doing an impedance match between omics and wearables, so in the end, we can democratize healthcare by making wearables at home equivalent to what we have to do blood draws and omic analysis on in the future. And Nathan is leading the N-of-1 analyzer, where we can begin to do the kind of genome-phenome analysis we've talked about. We've raised the money and started the type 2 diabetes project and so forth. With UL, we have, over the last 4 or 5 years, consented 420 possibility subjects, and we've looked at 150 with regard to measurements at 5 times across pregnancy with the multi-omics, and this will be a gold mine as we start to analyze it of what's going to come in the future. And then the global 1 billion person initiative. So, there are 5 major problems in healthcare for society: quality, aging, chronic disease, equity and diversity, not a popular topic, and increasing costs. And we actually think we can meet all of those with data-driven science. And the savings comes from keeping people wealthy and avoiding disease. There are some diseases that cost $200,000 a year or more to maintain people. The possibility of these early transitions and reversal will have enormous savings. The ability to optimize drug use, where a study in 95 in Nature showed only 10% of the patients actually use drugs effectively, and suppose you make that 100%, you've saved a lot of $600 billion a year then. And then the insights that come from these large data sets are obvious. So the global 1 million phenome initiative will, 1, prove health, 2, it will reduce healthcare costs, and it's going to catalyze innovative new technologies, and we've talked about some of the things we hope to do, and generate thousands and thousands of new actionable possibilities and drug targets and the whole business. So what are the paradigm changes that'll come from this? It'll push a wellness and prevention. It will democratize data-driven healthcare, so we can really think about bringing it to all socioeconomic groups and to even underdeveloped nations, as long as they have cell phones. And then finally, we'll be able to match a partnership between the physician and the individual on this whole thing. And we have a terrific team in Phenome Health that is putting this all together. And we have a wonderful group of advisors here. Our board is absolutely world-class, and George Church has been a marvelous. George Church and Ralph Snyderman both have been enormously effective at giving us lots and lots of advice. And I think I'm running out of time, so unfortunately, you won't get to hear about the drug kinds of things. But the idea then is we've taken the genome and the phenome, and from it we've extracted the opportunity to optimize health using AI. And what I would have told you about in the drug company is we have the chance to make billions of drugs and select them very rapidly, either phenotypic selection or targeted selection. Because these are 30 amino acid residues, they're unstructured, they have totally different binding capacities than any kind of drugs that have been used before. 60% of the genome is unstructured, can't be folded, but can be interacted with by these peptides. And what we're actually taking is these peptides, and for each individual peptide in a library of 10 to the 8th, we're going to look at 100 human cell lines at their transcriptional patterns, map that through knowledge graphs into training for AI, and we'll have a powerful, uniquely different ability to direct drug targeting based on integrating with Phenome Health and their AI ability to analyze the individual. So the grand integration. Thank you very much.
Did you have time for questions, Lee? Are there questions for Dr. Hood? Again, we have a microphone up here.
It was an amazing presentation, the canvas of all the discipline merging together. The question I have is that how do you use studies in aging? What does it tell about the process of Alzheimer? So how it deviates, when it deviates, can you detect it? Do you have a technology to capture the cognitive decline?
So the answer is, I think, absolutely yes. So let me talk about Alzheimer's, because that's something everyone is interested in. What is really interesting about Alzheimer's is Mike Phelps at UCLA demonstrated 15 years ago that if you use metabolic PET scanning on individuals, you can detect Alzheimer's 15 years before the clinical diagnosis. So the question is, what kind of biomarkers can we use to be able to detect that? And this new class of alpha peptides really looks like a very exciting possibility. And I'm not talking about a year or two before you transition. We're really talking about 10 to 15 years before you transition. So I think with the possibility of using unstructured peptides to interrogate things in ways we haven't been able to interrogate them before, and what these peptides will be able to do is not just Alzheimer's, but all the major chronic diseases have amyloid-like displays for different proteins. The alpha-like sheets can detect any of those beautifully. So I think there's a very exciting possibility, and that's coupled with the fact we did a very small clinical trial at Providence with about 40 patients and controls, where we used state-of-the-art against data-driven wellness plus the Brain HQ to improve cognition. And the difference between those over 3 years was unbelievable striking. So I think if you get Alzheimer's or neurodegeneration early, there are a lot of behavioral things that you can enact to change that. Okay? Mm-hmm.
Wonderful talk. I had a question about what have you found about regarding the thoughts that we think influence gene expression and therefore also influence
What influence it?
thoughts we think. So it's a very interesting thing. We have a very materialistic view of everything from a biological perspective, but often it is really your mental state. To a large extent, something that you can't easily characterize. It could be influenced by culture and driven, and it can be changing the what you call epigenetics or any of the other things like gene expression.
Right.
What are we doing in terms of research to be able to associate those changes so that we can learn about how we can age better and age well?
That's a great question. So there are really a couple of things. One is there are some of the wearables that give you a lot of information about sleep and the quality of sleep and about emotional stress. And and have, classic suggestions for how to change them. Another thing is Michael Merzenich has done about 10,000 clinical trials with his BrainHQ on all sorts of things. And he's shown beautifully that you can detect before the events ever happen the likelihood of a susceptible person switching to depression, switching to drug behaviors, intractable drug behaviors. And so he can map with this all sorts of these psychological-like behaviors. So I think we're just beginning to explore that dimensionality, but as the wearables increase the dimensionality of the measurements they make in real time and constant, I think we're going to have enormously powerful tools for doing just what you said. Yeah.
Much.
Okay. Thank you, Lee.
Yep.
All right. I'd next like to introduce Dr. Adil Mardinoglu, Professor of Systems Biology at King's College in London and Group Leader in the Science for Life Laboratory, the Royal Institute of Technology. Adil, thank you.
Thank you so much, again, for organizing such an event and inviting us, and actually for the opportunity for being part of it. I actually want to take the opportunity to tell you a little bit about the vision of our Vizy longevity and also the work that we have performed before actually came to this stage. So the thing is, at Vizy longevity, we are very much interested in actually integrating all this multi-omics data and even big biological data, which I will briefly mention about that. And we want to lead the AI in healthcare for actually preventive and personalized medicine applications. In this context, we are very much interested in creating AI-based platforms for the analysis, integration, and interpretation of such biological data. And of course, in addition to the applications in precision medicine and also in the longevity applications, we are also very much interested in using such information for drug development purposes. To be very honest, we all talk about AI in drug development, in addition to AI in healthcare, AI in precision medicine. But we definitely think that there is actually a link between the functional and the molecular information is actually missing. And in this context, we also think that big biological data will be extremely useful to fulfill that gap and actually create the required information which is needed for large language models or any other systems biology-based analysis. In this context, our methodology is the integration of all the multi-omics data and actually use this information for the development of the diagnostic tools and therapeutic strategies. Actually, I had the privilege to take this stage after Dr. Lee Hood, so he has actually mentioned here about the multi-omics technology and how it has actually been extensively used for characterizing health and different type of disease applications. The main thing is, at Vizy longevity, in addition to the multi-omics technologies, we are very much interested in actually gathering the continuous information from the individual, from the variables, and also we are basically incorporating all the data from the electronic health records, from the diet, from the exposome, which is actually all about the drug consumptions, and also any other environmental stimuli. And of course, we are also very much interested in integrating the imaging data as well. And the moment that you have such information, it's around actually billions of data points per specific individual, you can actually integrate all this information and characterize the state that we are very much interested. We can also develop specific language model, like a generative AI tool for a specific individual based on his or her own biological data. We think this will actually allow us to provide better recommendation to the people and actually follow the health trajectory of the individual. The moment that you have such platforms, you can actually apply it into so many different areas, starting from the characterizing the health status of individual. If we are dealing with a patient, we can actually use it for subtyping the disease. And also we can predict the risk factors for specific diseases and then prepare personalized treatment plans and also treatment response based on all this information. It basically includes all the details that we need for medical applications. And the moment that we gather all this information, of course, we can also perform some additional analysis to reveal the mechanistic changes in response to health status between health and disease state. And eventually, we can look for new markers and new drug targets, which can be efficiently used for development of novel strategies. The main thing is, as I listed here a number of applications, and I actually, during my presentation, I will show you 2 applications regarding related to metabolism-related fatty liver disease and also Alzheimer's disease, where we have actually performed some multi-omics level studies and presented the results. I will be very happy to share with you those applications. The main thing is, in theory, to be very honest, Dr. Lee and many others in the field, like the leading individuals in the field, has been talking about around all these multi-omics technologies and actually about the precision medicine more than 20 years. To be honest, I don't think that I need to convince you about the need for such applications. Today, we are here actually about the scaling and application of this technology and actually scaling of this technology together with the Pittsburgh Medical School. And again, I would like to take the opportunity to thank the visionary actually outlined by Dr. Shgar and also the Chancellor of the Pittsburgh University. So in this context, we are very much interested in actually working closely with the clinicians, with the hospitals, and also we have established our own multi-omics data generation platform where we can actually overcome such barriers. Because by today, we have the technology that is needed for characterizing based on multi-omics technologies. It's already there, and we are very proud that we are working with Illumina and also with other leading companies in this field. And also, we have the platforms for integrating and interpreting such information. And we also have platforms where we can actually analyze this data and provide the actionable insights back to individuals using the platforms that we develop. So basically, we have already prepared all the setup that is needed for scaling up such technology. And in this context, the beauty of our methodology is actually we apply a holistic approach, and we basically, with our very established networks, we recruit the patients based in the individuals based on the plans provided in the study. And we have our own facilities where we generate this multi-omics data in a very standardized way. Because the main thing is we already have a lab in Istanbul in Turkey, which is providing extensive amount of service to Europe. Now, we are very proud that we will have the Vizy Pitt lab here in Pittsburgh. And we will also have very soon a lab in South Korea. By mid-May, we will be opening our lab in South Korea. And hopefully by September and October, we will open our facility in India as well. So basically, it is extremely important to standardize the collection of the samples, the handling of the samples, the generation of the data, the analysis of the data, and eventually providing the actionable insight using this very standardized pipeline. And in that sense, we are also looking for the opportunities to expand to other regions in Europe. And soon, hopefully, we will be announcing the inclusion of a number of other countries to our effort. And in this context, I can actually tell you that we have already started to collect samples in UK and also in Sweden for our studies as well. The main thing is, I want to also tell you how we perform such studies. The main thing is, with the advancements in the high throughput technologies, we actually don't need much of samples for all this multi-omics analysis. In this context, one tube of blood is more than enough for running this whole genome sequencing, whole blood transcriptome, plasma metabolome, plasma lipidome, and also plasma proteome analysis. And in this context, we perform 2 different types of proteomics analysis using homologic Olink proteomics. Or we also generate data using targeted proteomics that we have established in our own infrastructures. And in this context, we will be performing analysis for more than 10,000 protein using SomaLogic platform, or in addition to that, we will be generating data for more than 600 proteins using the targeted assays that we develop. We are also very much interested in collecting saliva and fecal samples, and in some cases, vaginal swabs and also skin swab as well. We are very much interested in characterizing the microbiome so we can actually study the host and microbiome interactions. And as I briefly mentioned, we are integrating all this information with the electronic health records, any kind of imaging data that is available, and also with the wearable information. And we have also created the platforms where we actually employ the systems biology-based and the AI-based approaches to analyze, integrate, and interpret such information. And we all finally developed the mobile platforms where we can actually deliver all this information, all these actions back to individuals who is participating in our studies. And we also have platforms for actually handling all the data for the clinicians so it can be monitored. The entire process is monitored by the clinicians. In our methodology, having the clinicians in the center of the entire platform is the key thing, since we think they will be the one who will actually manage the patients and also deliver all the information with the additional insights that we gain from the information. I actually went through this part, but it is great technology. The moment that developed, you can actually apply into so many different areas. So by today, we actually created platforms and provided services for the candidate moms and dads. And it can be actually applied during the pregnancy, and then once the baby is born, for newborn, for actually screening analysis and for characterizing any kind of health conditions. And also for following the development of the child, adulthood, and also for later stage of life for longevity applications. The main thing is, Dr. Lee has actually touched to the point, but I want to briefly mention what to do with this information, because we are dealing around 1 billion data points per specific individual. And of course, all this information is extremely useful for creating the digital twins of the individual by simply modeling the whole body biology, whole body human biology. And in this context here, you see that all the components of the human body should be characterized with the data that is generated, accounting the effect of the microbiome and also the diet. So the main methodology is we do generate biological networks and mostly genome scale metabolic models, and then transcription regulatory networks, protein-protein interaction networks, and also signaling networks. And the moment that we actually integrate all this type of biological networks, we can actually study the different part of the different type of the biological functions, and we can eventually have the global understanding of the entire human biology at health and disease. And that's exactly how we apply our methodology. And in this context, we basically created AI-based platforms for handling the big biological data. Because the main thing is, by today, we generated data for more than 10,000 people. And soon we will be expanding to 1 million individuals. And I will tell you briefly about that, as Dr. Lee has mentioned it. So basically, we think we should have solid platforms where we can actually store this data safely and also analyze and integrate all this information in a platform. And by today, we have actually created such platform where we can actually follow the entire sample, starting from the collection until the data is generated, and then analyze and integrate all this information and create these specific reports for specific omics data and deliver back to individuals together with all other informations on their mobile app. So, of course, it's also very critical to handle this data safely and securely. The main thing is, as briefly mentioned, we will be working in different countries, and in order to actually be careful for the data privacy and the security, we basically develop local solutions in addition to the cloud systems. The main thing is local solutions are actually quite important, since actually there are some sensitivity about the movement of the data from one location to another location. That's the reason that we have solutions for storing and keeping the data in the centers where we generate the data and where we collect the samples, to avoid any kind of security issues, and also to avoid the conflicts between the movement of the data as well, since we put a lot of attention on that. And of course, based on the GDPR rules, also there are some regulations that we strictly follow based on EU rules, basically. And in that sense, we also created an AI agent systems, and here you see the overview of it. And Dr. Vishnu will also give you a demonstration during his presentation, and he will actually tell you about the capabilities of our system. But by today, once we generate these billions of data points per individuals, we can actually create the specific language models for a specific individual, and the individual actually can discuss his or her own data using the AI agents that we already developed. So simply a specific individual can ask questions about the platform, about the data, and then how actually they should behave further, what type of diet they can have, what type of actions they should take to avoid it. And with the platforms that is now will be expanded by the Vizy team, we will also integrate it with the live biological information and the medical information, so it will be a living system which will actually provide additional information to the clinicians and to the patients in the clinic. So the main thing is I want to briefly tell you about our mobile application before I get into examples. So in the mobile application, we simply have the platform where we can generate this, where we can actually present this billion data point per individual. There are 8 different subsections where we have specific sub-reports for individual omics. And also, as I briefly mentioned, we have an overall large language model as well, so we can actually have the integrated report as well. In addition to that, we have also a number of games where we can actually follow the cognitive function, since it's actually an aging problem about the cognitive declines, and that's the reason that we like to gather information and then follow the process of the cognitive decline for our users and for our participants. In addition to that, we have the diet where we can actually take the photos and also record it with the voice. And also we have the wearables with rings and also with the smart watches where we can actually follow the movement of the individuals. And these are all fully integrated in our system by today. And of course, the reporting is quite important, like how to create a report for this individual. So our methodology was, in order to start with, we gathered all the literature information, and based on that, we created sub-reports for specific omics technologies, including whole genome sequencing, proteome, plasma proteome, transcriptome, metabolome, and lipidome, and also metagenomics. But by today, we generated the data for 10,000 individuals, so we continuously improve the reporting system. And that is the reporting system that we will start using at Pittsburgh, at Vizy applications. But in order to further improve it, the moment that we generate data for specific to population, we will actually improve the reporting system. So eventually, we will have a unique reporting system based on our own data, and that information will be used and improved continuously. So I want to tell you about our achievements by today. What have we done until today at Vizy team? So basically, in our lab in Istanbul, in Turkey, we basically analyze more than hundreds of thousands of biological samples. We already have extensive experience of managing this large amount of samples, starting from the collection until the data generation and providing the reports back to individuals together with the coaching. And then we already developed the platforms and now further improve that even with the generative AI tools. And we have the mobile application that is up and ready for providing all the services back to individuals participating in our study. And by today, we created almost the data for 10,000 individuals in Turkey, and there will be another separate presentation about the details of such study. And now the goal is actually to recruit 125,000 people in 2 years' time. It may be a challenging task, but we think that we can actually complete it, and we can actually recruit this much individual within 2 years, and hopefully even faster than that. So the idea was we basically characterized more than 150 different diseases with our 10,000 cohort. So by today, we generated the multi-omics data for characterization of 500 healthy people and 9,500 patients with different diseases. This includes a number of complex diseases, this includes cancer, this includes rare diseases. So now we are, of course, very excited to expand this even to larger numbers together with University of Pittsburgh, here in Pittsburgh. So the main thing is, I can also tell you another application that we are very excited about. The main thing is this multiomics technology can be applied to many different areas, but one of the interesting area for us is the newborn screening and carrier screening. And in this context, we have actually generated data for more than 1,500 newborns, and we basically look into actionable diseases using the genomics and other multiomics parameters. And we are very excited that actually this is providing extensive amount of information about the actionable parameters for the for the actionable diseases that we can actually treat for the newborns. And we think that itself is actually a great benefit for individuals starting from the birth of an individual. And of course, there is also another project moving on in UK with the 100,000 newborn projects. We are also actively involved in that and sharing our experiences and then learning from the experiences in UK Genome Project as well for the newborns. So I want to give you two specific examples how we actually generate such information for characterizing the fatty liver diseases. And in this context, we basically started by recruiting 60 individuals. This was the earlier studies that we performed. So I want to show you actually the power of the technology that we are using for characterizing the different stage of fatty liver disease. And in this context, we generated multiomics data for these individuals with varying degree of liver fat. We generated the genomics data and then plasma metabolome, proteome, lipidome, and also oral and gut metagenomics data. And here you see the differences in the clinical parameters on the ALT, AST level, and then uric acid, creatine, and all these clinical parameters. And here you see that the liver fat is significantly different in these individuals, which has been measured based on using magnetic resonance spectroscopy. So what we did is we basically look at the different stages of the liver fat, and here you see that there are significant differences in the clinical variables. We found significant differences in the oral microbiome. We have found differences in the gut microbiome, and we have also found significant differences in the protein markers and also plasma metabolome and lipidome markers. So the point is how to use this information for characterizing the health status of the individual. And in this context, we developed number of machine learning algorithms, and then we basically used all this information for stratifying the patients. And in this context, it is very interesting to see that even if you use all the clinical markers from specific omics, like it could be the clinical markers, it could be the whole plasma metabolome or proteome or lipidome or oral metagenome or gut metagenomics data. Here you see that the accuracy or basically the predictive score is almost around 0.8 or 0.7. Like we could never get something more than 0.8, which is quite interesting, even though you use thousands of data points for specific individual. But the moment that we generated data at different molecular level, we actually find out that if you select the 5 parameters from each omics, you can have an absolute predictive score of 1. So if you can actually push it, like you can even decrease the 3 to 4 different parameters from specific omics technology. So that is the reason that we've been encouraged to test and to apply this methodology and this technology into different disease areas. And here you also see the validation in an independent data set, which is almost 0.9, which is actually a very good score considering the heterogeneity of the disease and also of the differences between the patients. And here you see the important parameters which we can actually follow up for the characterization of the disease and for also development of the disease at later stage. So the main thing is I want to briefly give you another examples about the Alzheimer's disease patients. So in this case, again, we had around 70 or 80 patients. And in this case, we measured the cognitive function performing ADAS-Cog analysis. And we simply had 3 different groups as the mild, moderate, and the severe cognitive functions. And in this case, again, we basically studied the differences on the different omics parameter, and the technology and the methodology is exactly the same. We started by clinical variables. We also measured the plasma proteome, lipidome, metabolome, and also oral and gut metagenomics data, as in this specific case. And here you see the differences that there are significant changes between different stages of cognitive function. And the main thing is we performed the same methodology. We really wanted to understand if these omics parameters are actually holding same as in the Alzheimer's disease case. And it was very interesting to see here again that if you select the top 5 parameters from every omics technology, we actually had again an absolute score of 1, which is again too good to be true, but that's the fact. And here we also perform another independent validation, and again we had almost 0.9 in an independent cohort where we generate the exact same set of the data using our standard methodology. And here you see the important, basically the predictions. Except one patient, we had actually a very good predictive score for cognitive functions based on multiomics data. So we had an excellent question in the earlier presentation regarding the following up the patients and following up these parameters. To be honest, I can actually tell you that 2 of the protein markers that we identified here has been recently reported as the predictive marker of Alzheimer's disease 10 years ahead of the Alzheimer's disease development. So that's actually providing a great evidence that the technologies that we are applying or the outcome of our studies is actually can be used for following up the development of the diseases, which is quite exciting for us. In this context, we also develop platforms where we actually gather all our information and also other publicly available information or the data that is generated by others into one platform. And in this context, we have actually developed a platform called iNet Models, where we integrated all the multiomics technologies from Avial study, where we had the technologies from, these multiomics technologies from Stanford studies and also other precision medicine projects, so we can actually harmonize all this information and learn as much as possible from that. And in this context, we have actually recently published a review paper for Nature Review Genetics, where we presented and introduced the entire concept and shared our results. Actually, we are here, today to also introduce our global 1 million phenome initiative as well. So we are very proud with that, since we are hoping that this will be actually transforming the healthcare industry and the drug development. And the goal in our Global 1 Million Phenome Initiative, which has also briefly mentioned by Dr. Lee and Dr. Shgar during the ceremony in the morning, we basically want to recruit around 1 million individuals where we characterize the health and, hundreds of different diseases using multiomics technologies. So the question, the point is, the moment that we actually characterize the health, the end and the end spectrum, which is the disease, we will actually have opportunities to study the transition as well. But without characterizing the endpoint, we think there will be a big gap for studying the transition and actually using all this information for the benefit of the patients right away. And in this context, we will be recruiting, by today, we recruited more than 14,000 individuals together with the other efforts in Turkey, Sweden, and also with the Avial project in US. So our goal is to recruit 100,000 individuals in US, in Europe, and also Asia within 1 year. And and recruit another 300K within the next 2 years, and 600K subjects within the following 2 years. So within 5 years, we want to recruit 1 million individuals. We want to create these data sets, and we are confident that this data set will be actually the basis for creation of these very large AI-based platforms. And we are very proud that we work with in this project with Illumina, and also NVIDIA is actually supporting us significantly in terms of GPU and also in terms of the technological advancements. And of course, we will also have a great support from our colleagues from LNT, and that will also be a great addition to our project. So the main thing is how we will gather all the samples, and in this context we have actually a very large network, and we are very proud that University of Pittsburgh Medical Center will be supporting our projects and will be leading these projects in this space. And we also have a number of hospitals, large hospitals networks in EU and in Turkey. And then we also have very solid networks in India, where we will be gathering a lot of individuals together with Manipal hospitals. And you will also hear during the day from Professor Minnesong about our established network in South Korea, where we will be recruiting tens of thousands of individuals within the following years. With that, I would like to thank our collaborators at KTH, King's, and also Atatürk University, Stanford, Koch, NIH, and Helsinki, University of Helsinki, and the University of Leipzig. And of course, during the establishment of such projects, we got extensive amount of support from Newton Alice Wallenberg Foundation. By today, they supported us more than $250 million, so we are very proud with their very kind contribution. And then we also get support from Bill and Melinda Gates Foundation during the establishment of such bases, and also a number of EU projects that has kindly supported us. With that, I would like to thank you again for your time, and looking forward to seeing the exciting event.
Comprehensive presentation. In terms of technology, AI technology is now 20 years old. In terms of next-gen sequencing, it has been there for 20 years. But the reason why we have not made a breakthrough is the number of samples. What TCGA collection is, the maximum is a breast cancer 1,000. So the initially are launching 1 million people, different.
Hi, Kevin. Can you hear me all right?
That is a phenomenal. I think that will help us to actually optimize the model and the diversity of humanity, covering multiple countries. That would be most important. So in terms of creating insights from the large study, what are your plan to do that? Because more modes is also more expensive, yeah? How do we optimize the cost and generate new insights so that you can really take action quickly and most cost effectively?
Excellent question, and excellent point, actually. So I can, our view on that is, at the moment, we have been talking about this multiomics technologies last 20 years, 25 years. The technology was there. We had really great visionary projects. But the point is, if you actually look at the current ecosystem, there is an appetite of individuals on longevity applications. At the moment, the scientific wellness, scientific longevity is a trend, which is a good sign in our opinion. In addition to that, we had these advancements on the multiomics technologies. By today, we can measure 11,000 protein in the human plasma. We already have the shotgun metagenomics technology. We had the whole genome sequencing, new equipments. We are now considering, you will be hearing the recent developments from Illumina. So the technology is there. We are confident with that. And the other very important point is the handling of this information and providing all this information back to individuals. And we see AI era is a great opportunity for this point as well. So basically, individuals are ready for it, and then the technology is ready for it, and AI is there as an advantage. And of course, now we have the support of University of Pittsburgh Medical School, to start with. And this will also bring us a lot of opportunity. So to recruitment of the patient. So in that sense, we are ready for this. And about the cost. So the cost is actually, by simply establishing our own labs, we can simply minimize the cost at least 1/4 of the service provider's cost. And this is also helping us a lot. That's the reason that we started this journey by simply creating our own facilities. The cost is important, but for us, the most critical point was the standardization of the data generation. Because when we have a patient in US, when we have a patient in India, Turkey, UAE, wherever we are, we want to generate the data in a very standardized pipeline so we can actually use it and integrate it. That's another point. And to be honest, with the cost part, what we envision is, for the first 100,000, we will fund this project by simply raising the fund, and we work very hard on it. And I told you, I presented the case for 10,000, and we already generated data. It means we've organized the cost. But our vision is that for the second 900,000, we are hoping that people will even pay for it with the technology that we develop. And that will be actually even a benefit to the system, to the health system. So we don't want this 1 million project to be fully funded by the external fund. And we want patients to actually see the value and pay for it. And of course, there is also another good news that by today, there are a number of companies that are actually claiming that the genome will be $100. And then we actually also know that there are some other companies will be, providing these services. So simply, we think that if we can actually provide these successful cases for the first 100,000, we can, during the scale, it will actually scale significantly, so the cost will actually even come down further. So that's how we model it. And as you briefly mentioned, we need such information, such database for further development in AI in healthcare, not only in precision medicine, also in drug development. And we see that this project is a great opportunity since all the stakeholders are here supporting such project, all the ecosystem is ready supporting this project, and we hope we will deliver it as we promised during the presentation. Thank you.
Okay, so our next presenter through the wonders of air travel is stuck in an airport, but he is going to be able to present to us remotely. So this is Dr. Chandan Sen. He's the Associate Vice Chancellor for Life Sciences Innovation at the Health Sciences, and he's the Director of the McGowan Institute for Regenerative Medicine here at Pitt. So Chandan, unfortunately, I don't think can hear us very well, so he will present when we give him the sign, but we won't be able to do Q&A. So Chandan, take it away. We definitely heard him once before. What is, we see a muted button on the screen. Is that part of the problem? Chandan needs to unmute. So can we get a note to him? What's that?
Yes, hello.
We hear you.
May I proceed? Can I get a signal?
Yes.
Can you hear me all right? Excellent. Thank you. And first and foremost, this is a wonderful opportunity in Pittsburgh. And my apologies for not being there in my own hometown. We have our primary national meeting in progress in Dallas. And this year I had to come because I was elected sort of the future president. I was supposed to go back yesterday and the flights got canceled. So sorry about that. Unfortunate for me, but I've been listening to the talks, very inspiring. So I'm from the McGowan Institute for Regenerative Medicine, and this is a part of the University of Pittsburgh School of Medicine. And I'm the Pennsylvania PI for the NIDDK of the NIH Diabetic Food Consortium and the chair-elect of the National Food Consortium. I'm going to share with you some of the current proceedings that we have on the front of diabetic foot ulcers and wound care. So diabetic foot ulcers, the NIDDK Diabetic Foot Consortium was launched exactly with these numbers in mind, which is that this is a massive growing problem and a large number of these patients that have diabetic foot ulcers undergo amputation, and once you undergo amputation, your 5-year mortality is almost 50%, and there's a greater than 25% chance of dying within 26 weeks for people with diabetic foot ulcers. Now, obviously, as we can tell, the problem is not just the ulcer itself, the problem is the underlying health conditions and the ulcer taken together, so there has to be a comprehensive understanding of the systems biology of these patients if we are to make a significant change in the status quo. Now, if one takes sort of a hypothetical case, you have a patient, a 58-year-old male, type 2 diabetes, HbA1c 9.2, we have a moderate-sized wound, and the standard of care is failing. So what is the current standard of care, sort of? In offloading, collagen dressings, maybe negative pressure therapy, oral antibiotics, and it's failing. So one line of effort that is in progress currently with us is spatial multi-omics mapping of the wound. So in this case, wound zones are identified. So you have edges, the center, and deep. So molecular finding at the edge, high TNF-alpha, IL-6, clinical interpretation stuck in the inflammatory phase, the center, high MMP9, low collagen, meaning you have excessive tissue breakdown, primarily happens because of certain bacterial proteases that are very abundant in these wounds, and also therefore the scaffolding is sort of gone because these cells to grow back need scaffold, and that scaffold system is compromised. And in the deep tissue, you are detecting, say, Pseudomonas biofilm and hypoxia markers. So when you have biofilm, around those biofilm, you have respiratory burst by neutrophils, and this respiratory burst, as we all know, is a major oxygen sink, uses of oxygen to make reactive oxygen species. So after the respiratory burst is done, you have local hypoxia, which then feeds necrosis. So a precision intervention, as we could apply today, in the edges, you apply anti-inflammatory hydrogels. In the center, you go with MMP inhibiting collagen dressing, and in some cases, where appropriate, topical timolol to boost fibroblast activity. And for the deep tissue infection, targeted phage gels, and to correct the oxygen potentially where needed, hyperbaric oxygen. So these are, from what is in practice today or being developed in practice today. But beyond this realm of today is also a future that has more advanced therapies that could be in play. And one such would be cell-specific, gene-specific editing. So in a study of a large number of patients, we found one of the key barriers to the healing of these ulcers are this remarkable level of methylation of genes, in particular certain key genes in key cell types. So that led to our effort towards cell-specific, gene-specific editing, which we were able to do and rescue certain wounds. And depending on the type of the wound, the gene candidate and the cell candidate could be different. Later on in the slides, when I talk about single-cell sequencing, you will see how cell niche matters. So in another setting, in one setting it was P53, in another setting it could be PCL gamma 2. So depending on the setting, the target and the cell types could be different. Now, when we were doing to look at all these markers, that predispose these wounds to non-healing, we were looking at SNPs in a number of patients. So we had a training study and a validation study, and in this phase of the study, we had 84 patients in which we were able to find a number of SNPs that very strongly associate with non-healing. When we then took some of these SNPs and tried to then do a pathway analysis, what we saw was, these pathways that you see in the circles are the major hubs of these SNPs, and the ones that have a second brown line around it sort of overlap with the SNP story that you see in obesity, unrelated to ulcers. So there seems to be a significant overlap. between obesity SNPs and these non-healing ulcer SNPs. To your right is one example of a genotype of the RB4 gene, also known to be very well studied and extensively studied in the context of obesity. Now, about 60% or 61% of these genes that we see, they belong to the missense category and could lead to faulty protein production. So you see about 64% of the SNPs that we see have to do with protein coding genes, and about 30% of them are with link RNA. These are the two dominant sort of segments where SNPs seem to play a role in these non-healing ulcers. And if one then takes and tries to find out how the SNPs really relate to the known GWAS data in different other disease paradigm, the top candidate that comes up is obesity, and the top gene that gets affected by this is FTO. And as we all know, FTO is central to epitranscriptomics. So that has now led to an effort on our part to be now using CRISPR-Cas9 to be doing epitranscriptomic editing, as well as fixing of point mutations. So both lines of progress are currently in progress at the University of Pittsburgh School of Medicine. Now I'm going to a little bit switch gears and move on to a little bit of a description of the spatial proteomic work and the spatial transcriptomic work that we are currently doing. So this is the diabetic foot ulcer that you all more or less are familiar with. You go on microscope, and in a simple H&E stain section, you see something like that. You just go a bit deeper into the magnification, you see a classic H&E story like this. And now, when we use a PhenoCycler 2, so this is currently what we are using, you can now start seeing about 50 antibodies at the same time. We have now labeled about 140 human antibodies. So in different serial sections, we can trace all these 140 antibodies based on certain pathways we have suspected, based on other non-spatial type of interrogations. We have been led to certain hypotheses, which now we are gaining more granularity by understanding spatial proteomics and spatial transcriptomics. So these are the 140 plus antibodies that we are now labeled and more in progress. So, and these are all barcoded. So this is obviously not standard immunohistochemistry. Here the antibodies need to be BSA and azide free, and they had to be barcoded. So all these are now barcoded and more in progress. So folks would like to have many of these antibodies obviously are not very specific to the ulcer story. All of this is currently in progress at Pittsburgh. Now, if you turn on all these colors that you see to the right in the right panel are the antibodies, and you turn all of them together, you can hardly understand much, but once you start turning off colors and only turn on what you would like to see, you can now start spatially characterizing. So here you see, some of the vascular endothelial cells, with the fibroblasts and smooth muscle cells, and you're now seeing the, vascular structures, and then around the vascular structures, if you want to look at leakiness, you turn on some other antibodies, so on and so forth. I'll not get into the entire scientific storyline, but a little bit giving you an idea of the directions we are pursuing. Now, once you are looking at these proteins, we then, at the same time, so the protein data set, the workflow is that it flows through QPath, and then this Xenium, which is the transcriptomic workflow, passes through SEWRAT, and then we have a QPath SEWRAT data transfer, so we can actually superimpose the proteins with the transcripts, and that helps us build the right type of hypotheses. So if you now see the same sort of section with the image only turned on, the transcript and the cell types are turned off, as you see to your top right. So now we are segmenting the cell, and following the segmentation, you turn on, say in this case in Xenium, you have 5,000 genes. So you turn on everything, you pretty much see nothing except that there's a whole bunch of transcripts. But based on this now, you start studying the cell niche and the cell diversity and distribution. And based on this, here you're seeing some keratinocytes. In the bottom left side of the panel, you will see KERA1, KERA2, KERA3. These are the subsets of the keratinocytes that we had identified through single-cell sequencing, which I'm going to be coming to shortly. And then here we have turned on the endothelial cells, the fibroblasts, and the smooth muscle cells. And based on that, you're seeing the structures here. Now, if the interest here is to only look at keratinocytes and the subsets of keratinocytes and why are they not migrating, why are they senescing around the edges of the wound, we can now start turning on the transcripts and exactly know what pathway is being blocked, and you can then selectively go and unblock or rescue that pathway, and you can resume the healing process. If the interest is in vascular pathways, then you start turning on, the vascular genes, and depending on the genes of interest, you turn them on, and if you want to see it for leakiness, so in this case, you're looking at smooth muscle cells, and smooth muscle cell-related genes, and I'm just showing you a few examples. You get the message of exactly what we are doing, and should there be interest in collaborations, do let us know. Now here, we are looking at fibroblasts, and then followed by fibroblast subsets. We see 11 different subsets of fibroblasts, and some of them have regenerative characteristics, yet others are structural and housekeeping, so to speak. And using this approach, we have been able to identify a special type of fibroblast, which can actually replace the function of endothelial cells in making functional blood vessels at the wound site. So when endothelial cells and the conditions of diabetes or other conditions, they're sick and they cannot participate, there are fibroblasts that rise to the challenge, form functional vessels, and over time, these fibroblasts disappear and these cells then get replaced by endothelial cells. That's a storyline that we have been able to show and publish, and under conditions where you have endothelial cells that are not productive and functional, you then turn on the vasculogenic fibroblasts, which normally would turn on under healthy conditions, but under diabetic conditions, they are blocked by some epigenetic barriers. And more recently, subsequent to this paper, a few months ago, we published that if you undo those epigenetic barriers, you can now populate the diabetic wound with a bunch of vascular fibroblasts, vasculogenic fibroblasts, which can then rescue the vascularization process. Quick change of gear. Here now we are interested in also looking at the bacteria at the same time when you see everything else that I was so far speaking about. So now we developed the first panel with 10x that shows the bacteria and the human genes at the same time. So here you see, by way of example, a particular S. aureus, and as you know, bacteria could exist in planktonic form or biofilm form. So the problem is, if the bacteria exist in biofilm form, and the only test we have in the hospital of detecting this infection is to send these samples to the, for testing, all they do is CFU. And biofilm bacteria can be CFU silent. So we have had cases where it came back, no infection, and the patient died of sepsis, and the wound, when you go look for biofilm, it's all over the place. The bad news is there is no clinical test for biofilm infection. So therefore, we are working on this, and in this line of pursuit, we see, of course, around the biofilm, a whole bunch of macrophages, and among these macrophages, we are now finding some biofilm-inducible genes that segregate the macrophages. So when you now go back to the wound fluid, and you pull the fluid, run it through flow cytometry. So in this case, we have not named the gene. We have just dubbed it as biofilm inducible gene, BIG, positive. So indeed, you can now see that in the large spectrum of patients that we have, you are now seeing big positive CD68 macrophages. You now go back and do a biopsy, and you detect the biofilm using what we call PNA-FISH analysis, as well as scanning electron microscopy analysis. So that was a second line. Now moving on to the last segment of the talk, we are not talking about single-cell sequencing. Here we have completed, and we have data from 70 single-cell sequencing where we have the skin and the ulcer data. So in this case, you see to the top left, you have intact skin from that many, 39 subjects, and the foot ulcer from 31 subjects, and these are pair-matched. And so you see to the top right, you see the cell populations that we can see. There's a whole bunch of pericytes, vascular endothelial cells, keratinocytes. The reason we encircle the fibroblasts with a red line is they seem to be the biggest culprit on where the limitations are. It seems like if they do not lay out the right type of ECM matrix, these other cell types do not get the right type of cue to do their physiological functioning with respect to the repair process. So as we look at the different transcripts within the fibroblast, there are some pan-fibroblast markers, as you see to the bottom right. And then there are some specific transcripts that mark the dermal papilla fibroblast versus the dermal sheath fibroblast. Both of these are very regenerative versus the papillary fibroblast and the pre-adipocyte fibroblast or the reticular fibroblast. The reticular fibroblast mostly participating in scar formation, so on and so forth. So it's not all the same fibroblast, and it's very important to understand the relative composition of this fibroblast in the overall tissue. So going forward, if you now look at the intact skin, and if you just focus on the bottom right side of the equation of the panel, you'll see that from those patients that had such a bad wound that had to undergo amputation, just the cell diversity, the fibroblast diversity. So to your left, you see there are 12 different or 11 different subsets of fibroblasts. The diversity is dramatically different between those wounds that have gone to amputation versus that did not, the ones that recovered. If you now look at the same storyline with sort of a histogram representation, you will see an overt difference in the composition of cells among in these healing versus non-healing or diabetic versus non-diabetic skin, more so with patients that have severe wounds that then are followed up with amputation versus the one that are also diabetic but go on to heal. If one now looks as not the skin, but the tissue in the foot ulcer, so now you see the difference, the striking difference between healing and non-healing. So the fibroblast diversity in our hands are definitely revealing a lot of the secrets that then, when actually looked into the context of the other cells, gives us the cue of how to make the other cells more functional, but by having the right type of fibroblast and having the right type of ECM matrix. Again, the same diversity plotted in the histogram gives you an idea of how this shift from a more healing pattern to a non-healing pattern happens and what type of rescue could potentially be productive along these lines. With that, I'd like to end this talk and hopefully take a few questions. There are many people involved in this, as listed here. The major sponsors of this are NIDTK from the NIH. We have also substantial funding from the Department of Defense, including the Technology Development Award. We also were the only group in the country now that got funded to go to Krakow to manage some of the wounds that are happening in Ukraine, the military wounds, and that is through the DoD's MTech program. We have some funding from the NASA, and thanks to the UPMC Enterprises for being by our side, and to the Pittsburgh University of Pittsburgh School of Medicine. Thank you for your attention.
Chandan, can you hear us? He cannot. Yeah, I guess we could message him on his phone if there are questions. We do have a little bit of time. So is there a way to send him a note that we'll, does anybody have questions? Anybody have questions for Dr. Sen? If I see none, I think what we'll do is let Chandan get back to his airlines. Okay. Is Mani ready? We're a little early. There he is. Okay, so I'd like next to introduce Mani Venkatachalam. He's the Chief Business Officer at LTI Mindtree. And without further ado, Mani. So how do I get my presentation? I think they'll pull it up.
Okay. Well, he's pulling it up. There you go. All right. Good morning, everyone. Seems like I'm one of the privileged ones to come from Dallas well in time. I flew from Dallas. Now I know the reason why Harsha and Dr. Vishnu were behind me to come a day in advance. So it all worked out. So first of all, I want to thank University of Pittsburgh, the Dean, and the Chancellor for providing us this opportunity. And not a doctor by any means. So listening to all the distinguished speakers, my presentation is going to be a little bit different. It's definitely not going to be on any research data or any analysis that was done. So I'm going to actually be talking about three things. What is L&T Larsen & Toubro as a company is about? And the second thing is, what is some of the work that we've been doing related to healthcare and life sciences that is relevant for today's conversation? And the third one is, what is this partnership all about? What is in it for us? So during the course of my day today, I came across several things that kind of intrigued me. During the dean's speech, I heard him talk about transforming healthcare. In fact, he distinctively called about 3 or 4 words: siloed, disruptive, accessible, at scale. I felt those were all relevant for this partnership and also with what we have in our hands. I actually, when I grew up, I wanted to be a doctor. I come from India, but we had only 2 options at that time. Either you can become a doctor or you can become an engineer. Right? That is still an unfulfilled dream for me, that I couldn't become a doctor. I became an engineer. But being here today, being connected with this ecosystem, and this, by the way, this is the first time I'm even talking to so many doctors at once. So that provides me an opportunity to kind of take care of that unfulfilled dream, to be part of this ecosystem, to be part of this journey, that what we have in our hands to be able to transform healthcare. So it's really fulfilling from that perspective. Larsen & Toubro. So this is a company that's, for people who come from India, they would be able to relate it to. It's a household name in India. It's considered to be a nation builder. It is also considered to be a company that has delivered several projects at scale. It's a multinational conglomerate. And some of the examples that I would like to call out, and it's done at a global scale, right? Many of the airports and metros in India are constructed by L&T. The Doha Metro at Qatar, if you've ever been to Qatar, is actually constructed by L&T. The 2022 World Cup, Soccer World Cup, one of the stadiums which actually hosted the semifinals game was constructed by L&T. Approximately 7,000 miles of Indian road is put forward by L&T. So we have delivered several projects at scale. But again, then comes the question of, what does an engineering company have to do with technology here? So that's where LTI Mindtree comes into play. So LTI Mindtree, which is a group I am part of, Seema is part of, it's part of the technology services company, part of the broader L&T umbrella. Our vision is to use technology and to enable our customers and the communities to be able to flourish in this hyper-connected world. That's our vision. And our ability to do that, it's even more possible now, especially with the possibility of AI being in the forefront. I think I clicked the wrong button. Yep, here we go. Now, this is an industry, tech services is an industry that probably has been doing things a certain way for several years now. Now, we are at the cusp of doing something different. If we don't do something radically different, we will become irrelevant. And even that happens to our industry because of AI. So what did we do as a company? We have also kind of re-strategized ourselves because of the AI into 3 broad buckets. One is AI in everything. And these are the 3 broad pillars that we believe is going to help our company propel and then also help our clients. So the first pillar is AI in everything. What does this mean? Today, LTI Mindtree, we touch about 1 billion lives through the work that we do for 700, 800 of our clients. 1 billion lives. Now, we wanted to infuse AI into each and every bit of work that we do for these clients so that we could influence the 1 billion lives through AI. So that is the first pillar, which talks about AI in everything that we do. Now, the second pillar is everything for AI. What does this mean? I told you about building solutions in that first pillar, but if I'm not able to provide my clients with the underlying infrastructure, there were several things talked about today from an AI standpoint. We spoke about infrastructure, the compute power that is needed. We spoke about governance. We spoke about security, right? We spoke about data. So all of these is required to ensure that those AI solutions that are built safely lands in the hands of the physicians, the patients, and the consumers. So that is the second pillar all about. The third one, again, just with these 2 pillars alone, we will not be able to meet our vision. So that's why the third pillar is also important, which is AI for everyone. So what does this mean? No matter what happens, no matter how much the technology advances, there is going to be a human loop involved. So how do you make sure that this human loop is trained, is able to work with the technology, and then be able to help the consumers? So that's the third pillar. So now our strategy, at least for the next foreseeable future, for the next 5 years, is going to be this. Predominantly on these 3 principles. Okay. Now, I heard about the vision of University of Pittsburgh from Dr. Shaker today, this morning. And then I also heard the purpose from Dr. Vishnu. I think it was pretty fulfilling for us, at least for me personally, because our vision is also similar. Our vision, especially from a healthcare and life sciences perspective, the business that I represent, it is very simple. How can we make healthcare affordable and accessible to people? It pretty much aligns with what University of Pittsburgh is trying to do. But how do we do that? Our belief is it could be done in bringing 4 things together. One is collaboration. I mean, all the presentations that we heard this morning, it was all about not one person, not one group trying to do something just by themselves. It was by different groups, different partners, different players in the ecosystem coming together. So collaboration is very, very important. We believe that. Second thing is partnerships. And that's primary reason why we are here today. There are going to be disruptive partnerships in the ecosystem that will become even more prevalent going forward. And we believe that that is the second key for this vision to be fulfilled. The third one is building capabilities across the healthcare value chain. I mean, the whole work that Dr. Lee presented this morning, Dr. Adil presented this morning, it's all about putting these building blocks one by one together and then being able to build these capabilities over time. So we believe that is very important. And of course, the last one, but not the least one, is how do you execute this at scale? See, we are starting about 20,000 sample set of data to begin with. But if we have to scale that to 1 million, to multi-million, you need the underlying infrastructure, you need systems in place for managing that scale. So that's why all 4 of this is very, very important. Collaboration, partnerships, capabilities from a domain standpoint, and then last one is to be able to execute this at scale. Now, we have actually done a bit of work in this space. So today, between the pharma and medical device companies, it's about $1.6 trillion that is being spent. And our association in this space, we are able to influence about $250 billion of that $1.6 trillion through various AI solutions and work we have been doing. And this covers the entire value chain of the spectrum, by the way, right from research to be able to design some of these medical devices, to be able to do clinical trials, and help them with supply chain, manufacturing, and digital treatment for the patients. And towards the end, also making sure that insurance in the end gets innovated. So we cover the complete gamut of things. And what you see there are some of the representative examples of what work we have done here in this space by helping patients, either directly or indirectly, all with the use of AI. And this is just a very small sample, but now with our partnership, we believe we would be able to build even more meaningful solutions at scale that can transform healthcare. So that is the fundamental belief we have. Now I want to end my presentation with this one. Anytime I come across a partnership, I get reminded of this. I don't know how many of you can relate to what this picture means. Anyone in the room? You can? So this is from San Diego Zoo. If any of you have visited San Diego Zoo, they have something called as a show called as Cheetah Run. So we visited that place about 6-7 years ago along with my wife and daughter. And ever since, it has created a major mark in my life when I think about partnerships or when I come across partnerships. So what is this all about? So what you see there, a cheetah. Cheetah's name is Ruksa. The Rhodesian Ridgeback dog's name is Reina. They're very good friends. And how did they become friends? The cheetah was born with a deformity. Doctors said that, the cheetah was never going to be able to walk, let alone run. So they performed multiple surgeries on cheetah to see, if cheetah could walk. But the cheetah was very lonely, not able to cope up with the recovery process. So what did the zoo officials decide to do? They said, okay, we have to get something as an emotional support for cheetah. So they got the dog together, bond with them. Both of them were similar age. So ever since they started growing together, the only thing that they don't do is they don't eat because their habits are different. So what did this do to both of their demeanors? Cheetah by nature, all of us know, is very wary of the surroundings, doesn't trust the surroundings very well. Dog, on the other side, it's very loyal, very calm. So how does this cheetah run happen is, so they have like a stretch of 100 meters, lane that they let the cheetah run by itself. But before they do that, they bring the cheetah in the cage and let it look at the 100 meters stretch, sitting inside the cage. As it is sitting inside the cage and watching it, they let the dog run. So now the cheetah knows that I don't have to worry about the surroundings because my friend has already run that path. So when they open the cage, then the cheetah would run, knowing that I don't have to be worried or concerned about the surroundings. So what does this tell you? between what cheetah is trying to do and what the dog was trying to do. It is all about trust. It is all about trust. When you know that cheetah and dog coming from two different ways of life, if they can trust each other, I think the partnership that what we are trying to form here can be based on trust. So that is why this was too personal to me. And I thought, it will be worth sharing it here. That's all I had. Thank you, everyone.
We have time for questions for Mani. Adil.
Excellent presentation. Very nice information. I'm just wondering, how will you help us and how will you actually contribute to this project in terms of the engineering experience that you have? Because you have already developed very large platforms and you have a great experience in scaling the projects. Can you a little bit tell us about the scaling of our project to millions of individuals? So looking forward to hearing that perspective.
Sure. Thanks, Adil. So I see the partnership taking shape in stages. So the first stage is getting the infrastructure in place to be able to test your sample set that you have already identified. So that's where me and Seema are going to be working to make sure the infrastructure is available across all these regions. So that's number one. Number two is, for scaling purposes and also for creating some localized customizations that you were talking about, there are going to be engineering nuances that are involved. Again, from a localization standpoint. So we can help you with those engineering aspects of things, whether it is based in South Korea or in Turkey or in UK or in US, because we are a global company. So that is the second one I see as a possibility. The third one I see as a possibility is there are going to be a lot of data analysis to be done, because the model is going to output a lot of data, and you need data scientists to be able to look at that data, decipher, and then make meaningful sense out of it. So that's the third one I see as a possibility. And of course, the fourth one is, as you ingest lots and lots of data, the model is going to need tweaking and training to be done at scale. And that's where we think we'll be able to help. So those are the four things I see, Adil, by which we'll be able to partner. Thank you.
Thank you for the great talk, and love the cheetah story. But the question that I have is, as we form this partnership, given that you're familiar with the space outside of US, what are your advice in terms of how do we basically get our regulatory arms on board so that basically there is a smooth transition as we embed a non-US setups within US? And what is your advice to us in terms of next steps?
So thanks for the question, by the way. I think this is again based off of my limited understanding of the space, because we do work with several banks, by the way. And the two highly regulated industries, banking is one of them, and then, which is financial services, I encompass banking into that. And then the other one is healthcare and life sciences. So today, if you look at banks, even though they are global banks, the way that they tackle this problem is they've kind of segmented it. Same to, Adil, your point of localization. So they still would not have a comprehensive view of managing this centrally. They would break this down. Each region, like Europe, for example, would do it separately. US would do it separately. However, the governance model that they put in place, the standard operating procedure for guardrails that they have to work with would be still centralized. And I still see a similar framework being applicable here as well. All right, thank you.
Okay, our next speaker and the last speaker before lunch is Sunil Gupta. He's co-founder of and managing director and CEO of Yota.
Good afternoon. I'm the one who is standing between you and your lunch, so I'll try to be as precise as possible. My name is Sunil Gupta. I run a company called Yota. I'm from India. And I just traveled from India to Pittsburgh last night. So I'm just waiting for that. The jet lag is not catching up with me. I met Dr. Vishnu and Harsha about 2 years back in India. And before I come to what we do, so we operate actually large data center campuses in India. And in these data centers, not only we host people like Googles of the world and lots of banks, we host a lot of healthcare companies. We decided to build our own cloud, just like you see a cloud operator. We thought that as an Indian operator, why should I not create my own cloud? And 2 years back, when ChatGPT came into consumer life, everybody started using ChatGPT. India also caught onto the bug that India is so much ahead in terms of technology, but India has lagged in AI. So why don't India create its own AI? And we again jumped onto the bandwagon, and we partnered with NVIDIA and built in thousands of GPUs in India, built a GPU cloud in India. And the idea was to again create in India. There was no AI ecosystem at all at that time. And the idea was, can we create a huge AI ecosystem? Can we be a party to create that foundation in India so that the AI ecosystem in India grows? And whether it is a healthcare industry or government trying to build AI system for agriculture or education or for farmers, anything, we can become a part of that foundation making. So I was obviously looking out for people that while I'm creating the GPU infrastructure in India on my data centers, but where are the people who will create this industry-specific large models, which ultimately will be benefiting the end customer? And that is where I met Dr. Vishnu and Harsha. I think, there is a story which I think we have heard it today morning, how they were sacrificing practically their comfortable US life. They were doing great in Vizy, but they were trying to do something for India. They created a platform for Hanuman. And I literally got, too much attracted towards, what he was trying to do in India. And we started working together. His initial AI platform, which he built for India, were running on my GPU cloud platforms. That's how we connected. And when he kept on talking about what he's trying to do in Vizy for the last 4 or 5 years, what we all heard today. And a few days back, we started talking seriously about this symposium, that why don't you come, why don't you explore what we are trying to do in US? And this will be something on a much larger scale while we are creating the AI platform for healthcare. But ultimately, when it scales from 10,000 PMI to maybe 1 million PMI, what type of compute infrastructure it will be required in US? And instead of trying to depend on, let's say, third parties, right, paying millions and dollars of compute, right, to third parties, can the University of Pittsburgh create its own compute infrastructure right here in Pittsburgh? It takes care of its own compute needs. It brings in other healthcare providers as well. And obviously, gradually, we start serving the whole of US and possibly the rest of the world. So that is why I'm here in front of you. So to start my presentation, just to give you an introduction again about what my company is, we are a part of a group called Hirandani Group in India. This is a large conglomerate who is more into infrastructure, real estate, hospitals, schools, roads and townships and things like that. 7 years back, I had met them. I come from data center and cloud industry for 25, 30 years. And we met together and said, okay, if we are to run a huge infrastructure of AI in India, you need to have a marriage between the IT people and the real estate people, because in data centers we require both. That's how we came together and created Yota. What we do, you can see in the next line, is we actually are a concrete to cloud company. And you can relate it to if I'm talking about creating a data center in University of Pittsburgh. I will have to start from land. You possibly will give me the land, you will give me power connections, you'll give me water connections. But from that stage onwards, I can create a data center right here in University of Pittsburgh. And once the data center is created with lots of cooling and power, I can bring in NVIDIA GPUs and create a complete GPU compute infrastructure, which again the university can use and Vizy can use for running your AI platform. So that is what is the capability we do as Yota as a single company. There are two parts. One is from scratch, we design, build, and operate large Tier 4, it's the highest quality data centers campuses. And on the top of these campuses, not only we host other customers like banks and other healthcare companies, but we also create our own cloud services, GPU services to deliver those services to our customers. We are operating these campuses in Mumbai and Delhi. That is where Vizy was running their India operations with us. And as I said, we have created our own homegrown cloud services and GPU cloud services. one thing which will be required, and I think that is something which got referred to in some of the earlier presentations, that this entire compute platform which you are trying to build is running on NVIDIA's technology, because NVIDIA practically has become the de facto technology, you can say, provider for high-end computing, high-performance computing. We have been one of the only 5, what they call as reference platform NVIDIA partner in the whole world. This is only second in Asia and the only one in the 5 in the world, who are following NVIDIA's technologies completely end-to-end. So right from their latest high-end GPUs to there's something called InfiniBand, there's something called high-speed storage. They have got a huge software stack for individual vertical industries, including healthcare. All of that hardware and software stack is something we have implemented into our data centers. So that stack represents exactly what NVIDIA offers in the US. The same thing is available, let's say, in India. So if I create a platform here in the University of Pittsburgh for you, it exactly will represent what NVIDIA has been doing anywhere else in the world. And that is the reason, when Jensen Huang, who is NVIDIA's co-founder and CEO, he's too much interested in that Yota become successful, because we are just replicating to what his vision is for the whole of the world. So this is what is the GPU cloud which we have created in India, right? This is like we invested into this about 18 months back, and it's live for last 9-10 months. And this is like, while on US scale, it looks still small. US and China are the 2 countries which in the whole world are too much ahead in terms of AI. But the moment you go away from these 2 countries, from at any scale, the 16,800 GPUs, and then our plan to bring in the latest generation Blackwell GPUs. This is a very, very large scale deployment anywhere outside of US and China. Yes, as I talked about, the whole of NVIDIA's technology architecture is something which we are following. And one of the things which, the moment we talk about AI platform GPUs, and you run these huge computations for your AI platforms, these GPUs demand huge amount of power, right? They are not like our regular servers, regular laptops and devices. They require huge amount of power. And so besides, and these are the GPUs which are required in thousands, not just 1 or 2. So just imagine each GPU requiring too much of power, and you have to put thousands of them together. The overall data center building which you create, right, it is a very, very specialized data center building, lots of power and lot of specialized cooling. You cannot have a normal air conditioning the way we are sitting in this room. You require something called direct-to-chip liquid cooling. Those are the technologies which have emerged in the world stage in recent times, thanks to NVIDIA GPUs. We have implemented that in our data centers. So again, if these plans, let's say, materialize of me building data center here in University of Pittsburgh, we create this very specialized data center building here with all these technologies of lots of power, dual power connections, and getting direct-to-chip liquid cooling, putting thousands of GPUs here. This is just an image of the data center, one of the data centers I'm running in Mumbai. This is we call it NM1 data center. This is quite a high-density data center there. It represents everything that you can find in the best world-class data center. There's something called Tier 4. Tier 4 essentially means whatever you do to this data center, even if somebody manually do something, they break something, the data center will keep operating as if nothing has happened. It's a fault-tolerant data center. It operates practically 100% of the time, even if there's no person in the building. This is the data center where I'm running my current set of GPUs. So this is because of a high scale of this data center. I can run almost 24,000 GPUs in this single building, and I can keep on expanding that. And the same thing, I'm just trying to create a replica of here in this university. So if I build a data center here, I go for a larger piece of land, create a design of multiple data center buildings, bring lots of power, bring lots of fiber, bring lots of water. And the idea, you start with one particular scale, depending on how many GPUs you require for your current project scale. And as the project keep on increasing, you get more and more customers coming in. You need more and more compute. You keep on scaling with more and more buildings and more power and more cooling. So we design accordingly. So this is something, and I think I don't need to re-emphasize this. This is something which is coming out in every single presentation. Why I'm here, why I'm talking about creating this large data center here in Pittsburgh, because every single thing which is being talked about of doing precision medicine, handling multiomics, you will be processing tons and tons of data. Right? For a single human being, the amount of data which we generated on a regular basis will be huge. And if you are trying to do it for million human beings, and if you try to go to a scale like India's scale, you will be talking about multi-million number of human beings, not only to train your model, and after that you will be using those models to do inferencing, to give results to the people. The amount of compute you'll be requiring will only increase, right? So it is very, very important that if you have to handle that type of compute, the facility you are running in, number one, you need to have compute into thousands. You need to have the latest GPUs. Second is this compute facility should keep on increasing. God knows what will be the size of this, but you have to just make sure that this is very, very, very scalable, because you will be requiring huge amount of compute to build up your healthcare models. So this is the sort of a partnership we have. This is, as of now, under our imagination, which we are trying to put into reality. This is a work in progress. We are discussing closely with Vizy. We are discussing closely with universities. But this is what we are trying to, execute. So we design, build, and operate an AI compute data center at Pittsburgh, which houses, to start with, let's say 10,000 of NVIDIA's latest chips, which are like B200 and B300 GPUs. And this is something which can scale. Tomorrow, if the project scales, which it will, right, you've got more and more customers and more users using it. It should be scalable to more than 50,000 GPUs with terabytes of storage, so that it practically becomes, Pittsburgh is able to position itself as the largest center for AI platform for healthcare. It will require, obviously, a certain detailing, which we are working on with the university, but it requires some 50 acres of land with lots of power from dual connections. I'll be speaking to the officials about it. We create the best of the data centers here. We put in NVIDIA GPUs here. We put the NVIDIA's technology, they call it NVIDIA reference architecture, the whole software stack there. Idea is, if like today, our company also is getting listed on NASDAQ very shortly, and we have plans to expand globally beyond India. So when we are exploring US market, there's one state which is coming very forward in attracting outside investment, which is Texas, right? And they are rolling out offers and offers to DC operators in terms of subsidized land, subsidized power, and things like this. Why can't we have that in Pittsburgh? In Pittsburgh, if we are able to have the right benefits, and we talk to municipalities and university about it, that you have the land, you have the power connections, you have very subsidized power, and we create this huge center here. It not only serves the need of the whole of Pittsburgh, it serves the needs of whole of the US and the whole of the global community as well. So it's very, very important that not only you execute it very, very well, it's also important that you execute it very, very cost effective. And obviously these input elements like power, how we make it cost effective is something where we'll be working with the municipality for that. Now, given that this is something which will be a replica of what I have been doing in my own country, and we have with time sort of learned from our own mistakes. We know what goes right, what goes wrong. I feel given that certain minimum inputs are given to me here, this first phase of this large project can go live as early as in 9 to 12 months. This is a humongous project, right, for any AI project, while we talk about the software and the platform layer, but the real money is made by the underlying compute providers. So this project, over 5 years, I'm estimating, even if we go by 10,000 GPUs, will require almost like $1 billion of investment. And majority of that, it's because the infrastructure you have to build right up front. Majority of that has to be put right in the first year itself. And of course, we are in discussions with Vizy as well as the university for giving a tangible shape to this whole idea, and of course put it into execution. And this is something ultimately this project will solve. This will enable very affordable, data-driven, proactive healthcare. I think I don't need to say that. All the speakers have already said that, how AI will revolutionize whole healthcare when you have a platform like this. Of course, it will support multi-omics research, AI diagnostic, and therapeutics. It obviously enable preventive healthcare by way of early detection, by the AI models. Of course, personalized treatment. This center, as I'm saying again, that while we start it for Pittsburgh, we start it for healthcare. If you create a facility like this in Pittsburgh, I'm talking about a wider picture, this can democratize access to this high-performance compute, which is not generally available to people, to multiple verticals also. If you see from a Pittsburgh as a state perspective, healthcare is one biggest domain I should put this platform and this project for. but why not to take Pittsburgh for serving the overall AI ecosystem for the host of the US? And this project can really do that as well. So it will democratize access to high-end compute for other healthcare innovators as well as other industries as well. Of course, it helps collaborations, researchers, providers, because you have a platform, everybody can start using it on a usage model. Nobody need to put in their own capital to create an infrastructure like this. And of course, as I said, it shall position Pitt as a global AI healthcare innovation hub. Well, this is something which I had to speak on. I hope you found it useful. Thank you very much.
Thank you, Sunil. And as you can see, we have a very exciting and ambitious vision for what we can do here from Pittsburgh. And we have great partners like Sunil and Vishnu and Seema, as well as Mani, who are committed to building not only the infrastructure, but also the capabilities for what we can do with the great health sciences schools and expertise that we have here in Pittsburgh. So it's really heartening to see this vision finally coming together. And I thank you all for attending this morning symposium. I think we're breaking up for lunch and coming back in 1:20, 1 hour. Be back here. Thank you. And lunch is served where? Downstairs. Thank you.
We have Dr. Vishnu, who'll be talking to us about AI and the clinical applications of multi-omics data. So.
Good afternoon, everyone. It's post-lunch session, and I'm happy that some of you returned back. Right? This is what happens in all the conferences. Post-lunch session is the toughest because people meet up to each other and then they're talking and forget that sessions are going on and we need to go back. So yeah, it's so when Adil put me in the second half, I knew that this was going to happen. And especially coming after Fiona was even more scarier because I knew that it's going to be amazing talk, and what will I talk about genomics or anything after she talks. Then I thought, I have done like 10 different presentations for this meeting. At last, I didn't even know what I'm going to present. Then I said, let me talk. That's the only thing I do well. I'm not a scientist, I'm not an academician, I'm an entrepreneur who is trying to solve world's biggest problem, I think. Because I'm a trauma surgeon, so sometimes I also forget that I'm a trauma surgeon. I have not operated since last 4 years, to be precise. After having lost touch of all the surgeries and all, what am I trying to do in healthcare is a big question sometimes. My daughter doesn't know that I'm a doctor. She was born after I stopped operating. And my nephew also hardly believes I'm a doctor because he thinks that he never goes to the hospital and never even seen a stethoscope in his hand. So how is he a doctor? It was a very conscious decision I had to take some time back, whether I should be operating or not. Because it's a tough decision. You get trained as a doctor for 20 years. You take care of so many people. And in a place like India where doctors are still now treated as gods, that's the only place where people will come and, even fall on your feet. So it's an amazing place to be there as a doctor because the kind of respect people give to doctors is next only to what they give to gods. So it's an amazing place because healthcare is so tough in a place like India. We are standing in world's richest country which spends $4 trillion. Here I see people saying that accessibility is a problem, affordability is a problem. Yesterday I had a driver who was so happy that Pitt takes care of his annual health. And imagine, like, I never thought that somebody from US would be speaking to me like this before I came to US. So when US feels it's so expensive to do healthcare, how is a country like India, which is having just $2,600 per annum per capita GDP, going to do anything like what we do here? So imagine the kind of accessibility, affordability India would be having. So it's a huge global problem. And when I came to US, I came with the vision that, okay, I will learn a lot of things from US, take it back to India. That was my vision when I came here. I took a lot of courses of healthcare analytics, global healthcare in Harvard and all that. After one class, somebody asked me, like, what did you learn about US healthcare? I said, only thing I can remember it is complex. That's the only word I could remember after studying US healthcare for so many courses and so many professors. It's so complex. There is a patient, there is a doctor. In between, there are 100 different things which happens before, what you pay comes to this doctor. At least in India, we have solved that problem. I can directly go and pay a doctor. That's why India's healthcare is cheap. Because directly you are going and paying the doctor. That's it. Nobody in the middle. There is no pharmacy benefit manager, no insurance, nothing. Just go and pay the doctor, see him, and come back. That's why it's cheap. But in US, so many things are there. It's bloated. So I said, I thought this country will solve my country's problem. Now it looks like I have to solve this problem. Or else this is going to kill the world, because $4 trillion, $10 trillion across the world, still nothing has been solved. So I was looking for something which is amazing. Then I realized why we don't even do this better, because we have built hospitals for sickness, right? Like, I fall sick, I have a fracture, amazing treatment I will get. So because we have designed hospitals for that kind of episodes where you have, I have my friend, Dr. Vivek, who will talk later. He would have stented 10,000 people when they're in the virtually deathbed. So we are amazing for that. But he asked me a question sometime back that, I see 30 years, 40 years old having blocks. What can we do to prevent it rather than just go on stenting more and more people? Can we define healthcare for the people who are not sick? Because healthcare is needed by everyone. But we have designed beautiful systems, big systems to take care of the people when we are very sick, 10% people. What about the healthcare for the 90% people who are not sick, but who will again become that 10% sometime in their life? That's what Professor Lirai would call as transition. When you are going to transition from health, in fact, it starts from wellness. You are super healthy, and then you become okay, but after that you transition into disease. This is the amazing problem which we are trying to solve here with GainMed, with this 1 million initiative. Everything we are trying to do is just addressing this problem. How can we identify people when they are transitioning and then take care of them, or else take health to the people rather than they come to this hospital and then get the treatment? So what technologies can we bring to enable this, not at a small scale, but at a global scale? So I could say that only 2 technologies can enable to do that. One would be multi-omics. Of course, a lot of talk has happened. I would not even touch that. The other one is generative AI. So why generative AI? Why I'm so upbeat about it? Why I virtually put everything, all the apples in my basket on generative AI? Since 300 years, we have really taken a leap and bound when it comes to engineering technology. We have built amazing things. I see 200 bridges in Pittsburgh. It's all engineering. And it has enabled us to, walk on the water. I call a bridge. It's like Jesus walking on the water. Earlier days, we could not have done that. We just walk on the water because we built a bridge. That's an engineering marvel. That's for all physical things. What is the engineering for brain? How can we improve a capability of a person by 100x, 10x, 1000x? Is there an engineering for brain? I feel generative AI is that. So generative AI is that engineering which can expand the capability of a person by any x, at least 10x to 100x. Now imagine there is a best doctor in Pittsburgh. I met one of them 2 days back for my doctor, ENT doctor, Dr. Dohar. He told me a few things which only a person who has done decades of research can tell you in 2 sentences. But I see that information is not there with 99% ENT doctors across the world. Now, should I come to Pittsburgh to get that best doctor? How many can do that? How many this doctor can treat in the world? Hardly few people, right? But now imagine with generative AI, if you can scale this doctor across the world, that's going to be amazing. Even Mozambique, India, a rural place in India can get this doctor's capability. Somebody might be treating him, but the brain behind that would be this doctor, the best doctor in the world. That is GainMed's vision. Why we are building medical twins is that, not for the patients, but for the doctors. Morning, I was talking to another pediatrician here who has amazing insight on how to diagnose autism at 1 to 2 years of age. He says, if you most of the people get diagnosed at 4 years, because when symptoms are already so obvious that everyone can know that this is autism. But their overall quality of life and the quality of the caregiver's life is a lot better if somebody gets diagnosed at 1 to 2 years, which is very difficult to miss the diagnosis. He has an idea how to do that, but 90% pediatricians would not be knowing that. So how can we scale this doctor in Pittsburgh to every doctor on earth? So that's what GainMed's plan is. We will create medical digital twins of doctors. You teach your own model how to do that, and then you scale it across the world. Then it becomes easy for a country like India to get this kind of information, this kind of knowledge, which is there in US at a very cheaper cost, at a nominal cost where we can use it. I would want Indian doctor, a doctor in Africa, to use this knowledge, which is there only in the top elite institutions of the world. And that can easily be done with generative AI, because I have created my own digital twin. I didn't know that Dr. Shekhar would take that twin and create such big vision for that, but I have created my twin. So what did I do? I told the morning one little story that I reversed my mother's diabetes. What did I do there? I'm not a diabetic doctor. In fact, when I was a trauma surgeon, if some diabetic patient comes to me, I will just straight away tell, please go and see the physician, or else go and see the endocrinologist, because they are going to take care of your diabetes. I'm not the guy, right? I just operate on knees and your spine. So that was my knowledge of diabetes. If you're a diabetic, I have to send you to the specialist. That was my knowledge of diabetes. And when I started treating my mother, I found something amazing, that 70% of diabetes problem is because of nutrition itself. What we eat, how we eat, makes a lot of difference. But as a healthcare system, it's very difficult for anyone to follow you and then teach you so many things that you change your nutrition. It's very, very difficult. So we have never scaled it. All the ADA guidelines would say that nutrition is important, but it's in one single paragraph. All the 200 pages of the book is about what tests you are going to do, what medicines you are going to use, and what are the side effects of those medicines, all that stuff. Nutrition, one sentence. So obviously people feel that nutrition is important, but it is just 0.01% important, but I say 70% important. So I built my model, which can tell a simple way like what you can do with your nutrition. It will take information about your nutrition for the last 14 days and then tells like what 5 things you are doing wrong. Correct one step by another. That's the framework we have. Like yesterday, someone was asking from insurance, what is the trick behind such a beautiful success you have in reversing diabetes? I said, we take care of nutrition. They were so surprised. We thought some drug. No, if drugs had sorted, I would not be here in this market. Some drug would be there. It's, okay, Ozempic has sorted it out. It's a different matter. I will not speak on that. This is what we created. So we have created a twin which treats people like me. Now I can scale this up to any doctor. Currently, if you look at what we do in India, I'm not treating any patients. My team treats it. Most of them are not even doctors. They're just nutritionists who are helping patients reverse diabetes at a scale. So now I don't need a doctor with 20 years of experience, tons of research to do this. I can do it with this. I'm only just overseeing these patients. So this is the kind of model which we are trying to build for cardiology here under Dr. Vivek. He will speak to you about it later. We say that you do preventive cardiology, and then we scale this up. This is how we are going to use AI. So I thought of telling like how we built LLM to a elite audience, but I feel that this is a better story to tell why we are building medical digital twins as an application, right? Because generative AI gives this capability. Before 2 years, if somebody had asked me, can you do this? I would have told it's very impossible, because if you have to write rules for machine learning, it's so complex. It never scales up, and that's why you see all that accuracy rate is not high and all that. But generative AI is something, a different capability, which brings a lot of use cases. And with such elite institute having hundreds of doctors who are best in their fields, we feel that we can create hundreds of digital twins, which can be scaled across the world. Now with more collaborations coming up, I see many other doctors from many other institutes. If they become part of this, this is going to be an amazing platform which can scale Parallelly, we are also building that million data sets, which are going to tell so many things about the human biology, which we still don't know. Like currently, all the numbers we talk about, genomes and everything, the actionable insights are a lot less because it's not been done on scale. In India, the only data set available is 1,000 genome data set for 1.4 billion people. We are trying to overfit the data which is there from UK genomics to Indian population and then treat them, guide them, even understand how drugs work on them. So it's not right. So that's why Dr. Adil, when he came with this concept that we have to scale it for 1 million, but with maximum people we can achieve across, the diversity was the key. Because I have seen like some large projects that have happened in genomics. Like example, there is a project which has happened in UAE where they have virtually scaled everyone in that country. But still, if you look at the contribution to the field of multiomics or genomics, it's very minimal because they have only sequenced very 1% of the population diversity. But here we are trying to do it across the world with more diversity so that it represents the true picture. Because many sets don't even have representation of people from Africa, people from India, people from many parts of the world. Mostly this comes from European or Caucasian population. So that really skews away the answers, what is coming out of this. So with this, we see that there is a huge possibility that it represents the true picture of the global population, which can be really applied to all the population in the earth. And such big data set is complex to manage. I have all my genomics reports, all 9 omics reports are there with me, and whole genomic report is like 200 pages, and proteomes are like hundreds of proteins, hundreds of metabolites. Doctors are already struggling with HbA1c and one single parameter. Now, if I take all these extra things and give it to them, how are they going to use in real world? So there is Dr. Philip here who is trying to do that with pharmacogenomics. He has come with amazing concept that when you are prescribing a drug and if that person's pharmacogenomics report is there, can you look at that time and then see whether you need to prescribe that drug or reduce the dose, increase the dose based on that. So that's just one part. Like imagine like I give all these omics to the doctors, how are they going to use it? It's too complex for human brain to even comprehend. So that's where exactly AI comes in. It makes the report so easy that it's a no-brainer. Virtually you get an insight out of it and then you can use it in real world. So that's what we have built in Vizy, and I call it as a cipher model. But we always thought like how to make life of doctor easy, because the biggest challenge in US I have seen is using EMR system. Out of 15 minutes you spend with the patient, 10 minutes really goes to the EMR system, 5 minutes for the patient. And we want to reverse that. We want to make doctors spend 12 minutes with the patient, 2 minutes with the system. And that can be possible only with generative AI, where you can talk into the system, where you don't need to do much. I will tell a small example of the biggest challenge with humans. One is food journaling. How do you know what I eat every day? It's the most complex problem. So many companies have come, so many solutions are there, where you can type into that, where you can take a picture, all that. But still it's so complex. Like I ate something in the afternoon for lunch. Do I really think that somebody will take my picture and upload when I'm in a conference? It's not possible. So food journaling, without that, treating nutrition is an impossible task. So what we have created is a nice app, which we call Vizy app. You don't need to do anything in that. Every day, just spend 2 minutes, just tell what you have eaten, just recollect what you have eaten in the whole day. Just tell that I ate salmon in the afternoon with some salad and one biscuit, cookie, that's it. You just tell it in your own words. And even you don't tell timing also. If you have a CGM, we can make out like, when your glucose spiked, so that I know when you ate your lunch. And that's what is needed for me to do food journaling. Just 2 minutes you spend with the app, and then I have beautiful data of what did you eat and how is your nutrition going on. The insight will next day come that you did an amazing job except for eating that cookie in the beginning rather than you would have eaten later. It would have not spiked your blood glucose. Of course, I didn't eat the cookie, I just making it up. So this is the amazing things which we are going to do with AI. its possibilities are immense. And with such nice data coming up from multiomics, we feel that it's going to be a big game changer, not only in the deep biology science, but also in the very simple things like administrating care, taking care of patients using the EMR systems. So virtually it can really change a lot of things. But the biggest challenge for this was, who is going to build a model which is built for healthcare? If you look at all the generative AI models in the world, they can do everything. ChatGPT does wonderful job of healthcare also, but also works for finance, but also works for everything under the sun. But it's not very accurate. It can hallucinate a lot because it is developed on a lot of data. So what we have done is, we have built a model which is just like ChatGPT or DeepSeek, but we have cleaved out a model out of it using 3 trillion tokens. This is called Cypher. So it's built on 3 trillion tokens. You can call it as 3 trillion words of healthcare. It means everything published under the sun on healthcare literature, which is publicly available data sets. It's gone into that. So it knows virtually all the articles on PubMed which are accessible, every data set. It's been trained on that. On top of that, we are trying to build all the solutions. This is what Vizy has built as a solution for healthcare. Virtually, you can use, somebody asked me, can you use ambient Wi-Fi and get the information? Yes, we can use any biosensor, we can use any IoTs and build solutions on top of Vizy's platform. So this is perhaps the biggest model in healthcare for large language model. And I'm amazed that Pitt has another important thing, which is AI for Healthcare, Center of Excellence run by Dr. Human Rashidi. We are going to work a lot on this, bring this, a lot of papers, publishing, and do amazing things like, so I have seen the work they're doing here, and I'm very upbeat that this model will go miles with the kind of research and capabilities Pitt also brings to the table. So this is what I wanted to show. This is the capabilities we can bring using generative AI, and then creating some amazing things which can transform healthcare. So I have to thank again Dr. Shekhar for giving this amazing place called Pittsburgh to start all this work. He had come to inaugurate our office last year, and I'm happy that again we are doing another inauguration of Keynote and Vizy's lab in Pittsburgh in 2025. So we did that in 2024, and thank you, Dr. Shekhar. I don't know. He's here. Thank you. So I didn't see you. Okay. And then I'm honored to again speak today with Dr. To be in this audience and tell about what we are trying to do. And some doctor gave me a very interesting insight that I have built this, I have used this solution, I'm trying to scale this up. I said, exactly, we are looking for doctors like you who are trying to do things on your own. We are going to give a platform where you can do this on GainMed. This is going to be, any idea you have which you want to use generative AI and build capabilities, GainMed will enable that. We want more and more doctors who are trying to solve their own little problems to use it. This is an amazing technology. So this is what, in simple layman terms, what GainMed platform is going to be. I have kind of laid down the vision. So we'll enable everything, including the infrastructure, because another challenge which I have to mention, why I'm so upbeat that large tech companies should become part of this, like L&T, and because generative AI is super, super expensive if you have to use, because currently the technology is costing, hundreds of dollars. The core things, like if I use a O1 Pro, it would be like $600 for a million tokens. It will just generate few answers, and then you have lost $600. Imagine if someone has to use it at a scale, it's going to be tens and tens of millions of dollars for one single institute, and whole world has to pay. It's super expensive. That's where we have worked again. We have brought down the cost of that kind of AI by 100x already. So we can generate 1 million tokens in peanuts. It's like in 10 cents, my model can generate 1 million tokens. So this will bring down the cost of healthcare, and for that, we need that infrastructure, we need that engineering. That's why we bought this whole thing here, because I feel once it scales, it will be like internet. The cheaper it is, more people can use it, more people can use it at a scale. So that will come only you have this core capabilities. If you are just like taking some model and then trying to fine tune and build it, you can do well in a POC, but it won't scale up because it's so expensive to scale this up. And for a global project like this, we need AI at a scale, which is the cheapest of all and efficient of all. That's another thing which we have worked on. And so this is what I just wanted to submit here, because what suddenly GainMed, why Vizy Multiomics Lab, I just wanted to lay down that this is the vision and this is what we want to do. And we are bringing this to Pitts, and we want to work here and then scale this across the globe. So that's the vision. Thank you for giving me this opportunity to again speak second time in a day. It's amazing to be again talking and telling about what we are trying to do. And thank you very much. And I hope somebody will have some question to me.
Thank you, Dr. Vishnu. That was very inspiring and really grateful for your engagement and participation. My name is Suresh. I'm from UPMC Children's Hospital and faculty at Pitt. But just a quick comment and a question. The comment is, thanks for the shout out regarding autism.
Yeah.
As pediatricians like to say, children are 20% of the population, but 100% of our future. It's really important that such platforms can be used to advance pediatric care. The specific question is, all of this is based on today's technology and today's healthcare problems, and those are not static. We may have newer problems in healthcare, especially mental health issues, et cetera, and the rate at which generative AI technology is also expanding. Any thoughts on how GainMed is planning to keep up with upcoming problems and technology which may not have been even invented so far? Thanks.
Yeah, it's an amazing question. And that's where I think the whole concept of system biology and system medicine comes in. Because system medicine, how I understood, I'm from MIT, which is like, where they invented system dynamics from all where this flows. So about autism only, I want to tell this. There are two schools of thought on autism. One says that, okay, because the diagnosis has improved, so more people are being diagnosed with autism. It's otherwise, autism is always there, so it's not increasing. That's one school of thought. The other school of thought says, no, the incidence of autism is increasing, and some people have gone to even blame vaccines for that. We don't know exactly why autism happens, at least with the current research. There are many hypotheses, many theses, but we don't know. So how do we even know why autism happens, so that I can go to the root cause and prevent it? Because otherwise we can't prevent this thing, and we are only trying to develop tools and technologies to just enable the kids to take care of themselves or their parents to take care of themselves. And if it is increasing, that's a dynamic problem, as you are saying, what is happening in our society to increase the rates of autism. So how are we going to keep doing that, challenging that? I feel the whole metabolic disease has increased because of the fact that we are the only, perhaps, last 2 generations where adequate food is available to humanity. If you look at India in 1940s, people used to die of famine. Like latest famine India had was in 1943, where, I don't know, whole Bengal population was wiped out. Today, we have adequate food in India, but our genes perhaps don't know how to even take so much food, and we are like landing up in diabetes in 40 years, which never used to happen in India. That's a hypothesis. I don't know. Till I test multi-omics, I will not know why Indians get diabetes at 40s and 30s and why they get so bad in like 10 years. This is an evolving problem. So for every solution, there will be another problem which will come out of it, and then you have to, think a second order. That's the system biology approach also. if you do something, it's not going to be a straight line. I use Ozempic all my life, I will maintain weight, is not system biology. It's a straight line thought, and you will see a lot of problems with Ozempic after some time. What can we do to prevent that is what we are trying to do with system biology approach. Because anything you do will intervene somewhere, and it will lead to another problem. So you have to observe the whole to know that what causes this problem and what will be the after effects of it. So that's what we are trying to do here, right, with system biology.
Thanks.
Thank you for asking that question. Actually, it's related to what I'll be asking Dr.
Yeah.
So one of the interesting things that happens in medicine, this is something I've observed from my 23 years of work that I've been working with data on, is that we often call something diabetes as a disease, but it's actually got so many multiple subtypes. So my question is going to be, given that AI and humans are using language to depict something that's biological in nature, so those subtypes, what we can see with systems biology and actual measurements, you don't have to give it a name. You can call it, or you can call it anything you will. But that thing that represents that disease type is constantly evolving or changing, as you correctly said. And with AI and how humans describe these in terms of languages and ontologies, that also keeps evolving. So in some sense, what I'm hearing Suresh, Dr. Srinivasan saying is that how will we in the future represent these kinds of dynamically evolving both language and subtypes and the physical nature of that disease itself as environmental effects change and so on. So I would love to have your insights on that.
Yeah, I will tell something, my take on this. When we say diabetes, it's like 6.5 HbA1c or 7 HbA1c. Until you go and cross that number, you're fine. Like you go to the doctor, they will say, okay, you're fine, you're not diabetic. Once you cross 7, you are medicated. This is how we treat people, right? Because we have to put them in a basket to prescribe drugs. But how I look at diabetes is, if you are HbA1c 5 today, and after 6 months it becomes 5.6, it's an alarming thing, because though you're not a diabetic, your sugar levels are going up. That's where the transition is happening. And after another 6 months, it's 6, still you're not a diabetic, but you are 100% pre-diabetic, you are going to be in that position. So you don't need to wait for people to become diabetic to treat. I would start treating people who are not, keeping their HbA1c. And current system, if you look at it, who is having this information? We are only looking at one number when we are treating a patient. We don't know what has happened 6 months back, what has happened 1 year back, because that's where I see AI playing a beautiful role, that it can predict this. That I have models which predict all this. If your HbA1c is going up, I can predict in like 6 months with the same continued lifestyle, it's going to be 7. So why can't we intervene right now rather than wait for him to become a diabetic and make him sick and then start prescribing tablets? So why even diabetic? Why not you start at a stage of insulin resistance, which is most of the root cause of all metabolic problems, and that can be measured quite easily very long before somebody even gets hypertension, diabetes or anything. That's when we have to prevent that. So these are beautiful things which can be done with AI, because it can, if you just give your lipid profile, it can calculate what is your triglyceride and HDL ratio and tell whether you are insulin resistance or not. It's such a simple thing. And with more multiomics coming in, we will have more biomarkers which can predict much early than what happens. I'm very upbeat on ceramides and all that things, which even work on, whether you have food craving or not. So a lot of things can open up, a lot of biomarkers. Me and Dr. Vivek keep discussing about many beautiful biomarkers which we want to implement in our precision cardiology plan. He will talk to you about them later. So they're going to be amazing than what currently we have, and they're all possible with what we are doing. Yeah.
Thank you so much. I'm going to have to bring this down a little bit. All right, so our next speaker is Dr. Hassan Turkez. He's going to talk about the Anatolian Precision Medicine Initiative. So. You've got to unhook the laptop.
Yeah, okay. Let me use the laptop. Okay. Yes, that's right. Distinguished colleagues, dear guests, it's great to join these activities, such a valuable activities in the terms of Pittsburgh University. I sincerely thanks to the rector, the dean, and the vice dean for their kind support and hospitality. And then I sincerely wish that Global 1 Million Phenom initiative will bring lasting benefits to humanity. As well as, I have no doubt that with the scientific contribution of our esteemed professors, Larry Hood, and then my brother, there, Prof. Dr. Adil Mardinoglu, that this initiative will be successfully completed. We have no doubt on this. Let me introduce a couple of words about myself, then I will make a brief presentation about our initiative. I am working as a full professor of medical biology department in Faculty of Medicine of Atatürk University. I am serving as a vice rector of university, which is the second biggest university in Turkey, and then third oldest university in Turkey. I have some roles in World Health Organization as a toxicology and epidemiology expert. And then I am happy to declare that I have been appointed as a member of Presidential Health Policies Board by His Excellency President Recep Tayyip Erdogan. So now we are performing a huge project in Anatolia. The name of the project is Anatolian Precision Medicine Initiative. And in this initiative, we call short APMI. I will use it. The APMI project is mainly aims to significantly enhance the quality, accessibility, as well as personalization of the healthcare services in Turkey. We are aiming this via integrating multi-omics data with the clinical and then demographic factors which are specific to Turkish population. The APMI project will establish a national reference databases for genetic, transcriptomic, proteomic, metabolic, as well as metagenomic signature or level. Beyond its medical impact, APMI positions the Turkey, our country, as one of the global leaders in biomedical innovation, public health resilience, and then strategic self-reliance in the field of P4 medicine, or comprehensively we say P4 medicine. Now, especially in the last 20 years, the healthcare have, in both private sectors and governmental sectors, have undergone a paradigmatic shift, as Prof. Larry Hood mentioned about in this morning session. But the shift came from the generalized medicine to patient-specific strategies, you know. But this is not only, how can I say, scientific evaluation, but seems to be a societal imperative. This change, this paradigmatic shift. Again, as I mentioned about APMI, APMI is not only a regional contribution, because we are performing in Turkey, where Turkey is the, as known, a part of, two parts of lands. We have Anatolian and then European side. The Anatolia is the largest part of Turkey, and then where east meets west. Therefore, our project integrates both molecular research, digital health systems, as well as cultural heritage. The triple contents of the project makes very valuable for Turkish society. Therefore, in that presentation, I am planning to make a little comments on the scientific rationale of our project, and then I will mention about the content and then operational framework, our initiation. As you know that the first human reference genome has been launched in 2002 to 2003. Then, after launching these first human genome references, it triggered several kinds of national genome initiatives. But the same performance didn't occur in the triggering actionable precision medicine applications concept. Maybe there were between two critical transition points is available between the launchment of single reference genome and then the actionable P4 medicine applications. In my opinion, the most critical transition points is the multi-omics integration into the national genome initiatives became so late. So this is one of the major bases of our initiative. And then, while we are mentioning about our courses, the first original reference human genome sequences, the 70% of this reference is belong to one individual, a blended ancestry. But interestingly, 30% of this reference genome is belongs to 4 different persons. Therefore, the first reference genome from a mix of several anonymous volunteers, you know. So, it quickly became evident that this single and then generated reference genome wouldn't be capture the all genetic diversity of the human population. And then this reference didn't reflect the global genetic variation. So growing scientific and medical research accumulated findings indicated that several national genome initiatives must start. Then I listed there in the right part of the slide, starting from the Iceland and predominantly in European countries, as well as our country started in 2023 with an Australia, the last started genome project. But all these projects aim to reveal population-specific genetic variants or improvement some risk prediction of the disease, as well as improve the precision medicine databases or the bases, and then provide benefits from the equitably from the genomic advances. So I scanned all the literature for 1 week. I wondered that how many countries are dealing with their own genomic projects. So I learned that this is the last report for our scanning reports. 26 of all countries in the world are being completed or continuing or about to complete, but they are dealing with their own genetic or all national genome projects. But 74% of the countries are not related to that kind of activities. National genome projects, yes, large-scale projects, we are sure that they provided essential baseline data at the population level. Again, we could conclude that about these NGIs. There are critical gaps for the shift, for the personalized healthcare strategies or the applications. But there are some limitations on this. I listed them, but I will tell the important ones. For example, population level data cannot fully captured in individual genetic variation. So generally, unique variants in individuals cannot be detected with the large scale of projects. Again, high inter-individual, and then especially in disease and health conditions related to drug response, we are ignored because allele frequency data is available from that kind of large projects, but especially this that's insufficient for the individual level. Therefore, again, the technological feasibility is important. Remember, when the first human genome is sequenced, it cost about $2.7 billion. But it became, as the speakers mentioned about it, it became $100 today, maybe less than $200. So this is the triggering factor, could be from mapping populations to trading individuals. And then the other interesting point is there, as I listed the table, generally NGIs were performed in Europe. The rest of the world thought that the ethnic and ancestral representation is predominantly in European countries, and doesn't reflect the global level. Therefore, these triggering factors made the second shift, an interesting shift, mapping populations into treating individuals. In this background, my team is working on novel drug development since 20 years. We are focusing on novel drugs, novel drug targets, disease stratification. But let me share a memory with you. Son of my team members has been diagnosed with the GBM, and that's due to this disease. So our team became eager to solve that kind of the most aggressive and heterogeneous brain cancers with an effective solutions, with an effective novel drugs. We strictly followed the literature to find out a new insight. For example, in Genomic Insight, we explored EGFR inhibitors but only EGFR mutated cells. We couldn't solve the resistance to the temozolomide, for example. Secondly, we focused on transcriptomic data. We performed many kinds of publications in there, but we learned that this is not related to resistance of the drugs, chemotherapies, others. So we learned that, but it is necessary for making a trial, the clinical trial design, because you are detecting subtype of glioblastoma with using this transcriptomic data. Again, we found PET-wave interactions, and then protein-protein interactions is also important for this tumor aggressivity. And then again, the Warburg effect was understood by the metabolomic analysis. We thought that disturbed amino acid metabolism is also related to this kind of aggressivity of the brain cancers. So we continued to investigate, examine the other multi-omic layers, and lipidomics made a different insight. We learned that especially sphingolipid metabolism is maybe effective to overcome the resistance to the chemotherapy. And then finally, methylguanine methyltransferase testing are suggested to learn early chemotherapeutic response for it. Now, when we make all these omic layers together, we understood that one single omic layer will open a new page for your career or for your project or for your academic journey, but it is not enough to find an exact solution. So the holistic view, which contains multi-omic integration and their interpretation by the system biology approach, would open a valuable way for us. So in that background, we designed our APMI project. It's like a Turkish genome project supported by multi-omics integration. So unlike the other projects, what is the differences between the APMI and the other VGS? Let me list them. For example, we primarily focused on not only genomic level or genome sequencing only. We added transcriptome, proteome, metabolome, lipidome, as well as microbiome analysis for that. Then we produced the similar data, but not only genomic, additional 6 layers of multi-omics data we added in our project. And then we enriched the biological sampling style. Because you know that generally the VGS projects, national VGS projects, focused on only tissue, maybe the cancer tissue, as in TCGA project, or like other national genome initiatives, they focused on blood. But we added blood alongside with the tissue, plasma, urine, salvia, and feces. So we also added, alongside with the omic data, and then enriched tissue types, sampling enrichments. We also added clinical and dietary properties of the individuals to the system. Turkey has a good health record system, which we called in nabız. We are using this in Turkish word. And nabız is recorded all individuals, all Turkish citizenship in the country, so we can follow up to 30 years before. So you can use that data easily with the integration of multi-omic data. And then let me say that the risk prediction, biomarker discovery, as well as applicable personalized medicine potential is high in that potential. This is my university. This is our medical research and application center. This hospital chain is one of the biggest in our Turkey. So it contains 4 different branched hospitals: general hospitals, child's hospital, cardiovascular disease hospital, and an oncology hospital. So in each day, we are accepting 12,000 patients from more than 20 different cities neighbor to Erzurum. And then our university's bed capacity is 1,500. So this center is our main coordinator hospital in APMI in Turkey. After deciding to initiate this initiative, we completed and settled all requirement infrastructures, including hospitals. I will tell in next slide, we coordinated different hospitals under the umbrella of Atatürk University Centers. So we added 4 additional cities and their huge hospitals in our project. Then, immediately we completed our sample collection units since we enriched the sampling not limited to blood. So we need biobanking facilities. We completed biobanking facilities in 5 of the cities and then 5 of the cities' hospitals, both under minus 20, under minus 80, as well as liquid nitrogen, and then freshly frozen techniques and others. So, critically, we need laboratory infrastructure and then computational lessons. Thanks to Prof. Mardinoglu, we are getting services from the CZA Omics, which is the unique facilities in Turkey, located in Istanbul. We choose our clinicians which are eager to work with us. Our APMI clinical teams were included 3 main areas of the medical science, both basic science, internal medical science, as well as surgical science. Totally, more than 150 physicians, only in Erzurum we are working on that. About 25 of them in other cities. And we need supporting team as a non-physicians team. There are about more than 100 members. We are working in different disciplines, as you see from the slide. We took different ethical, just 1 minute, I am complete. We took 2 different ethical approvals because we enlarged continuously our project. Now we are getting ready, I am happy to declare it, we are getting ready to take the 3rd one for the 150,000 people in Turkey after completing. Now, we completed 10,000 sampling. We recruited 10,000 people in Turkey. All samples were completed, and half of them were analyzed now. As you see from the figure, we completed 31 different types of cancers, 82 rare diseases, about 100 complex diseases we completed now. As I mentioned, we took all omic analysis, especially in 6 omics. We are also making B-plan. We have the tissues. We can add other omics, due to our budget and our planning. As you see from the coverage, our countries have distinct geographical areas. We choose the countries representing their geographical, and all patients sampling from the cities coming there as in regional hospitals, and we are collecting and sending the directly to analysis to Istanbul. And then we added some demographic and personal information, including age, weight, genders, place of birth. As you see from the figures, 77 cities of all 81 cities of Turkey, it's about 95% of Turkish population is reflected in that sampling method. We recorded also body weights, additional heights, and then citizenship number, personal ID, unique information were also recorded alongside the all our records. Finally, last two slides. Let me summarize this. Sorry for being late, about 1-2 minutes. We make all diagnosis within golden standard, and both international and national standards of guidelines were used to diagnosis of patients. And then we added both hematological, biochemistry, nuclear medicine, radiology reports, imagings. So we added all kind of clinical data to merge biology and clinical outcome. And we also recorded comorbidities. You know, comorbidities is important to understand the complex disease underlying mechanism, and then finally, to reach a pharmacogenetic profile, we had their medication history. So we added in our previous analysis results. The collection chain is important. We take care of it's being all entities, both digital and physical entities, to be compliant and aligned. We rigorously made this collection chain. And finally, we need a mobile platform that Prof. Mardinoglu mentioned in morning session. Thanks to him, he helped us about developing this mobile platform with a national and international regulated data security properties. And then they integrated multi-omic integration. And then we delivered the results for both clinicians and then patients with friendly user version. So finally, in our country, as I mentioned, our APMI project, we have worked hard on APMI for nearly 5 years, over 300 staff or 300 researchers. And we created a motto for us. We said that science today, better lives tomorrow. I hope we will be success in that area. Thanks for your attention. Thanks for your hospitality.
Thanks, Hassan. In the interest of time, we don't have time for questions. We'll have to move to the next speaker. So thank you. So our next speaker is Dr. Minho Song, as a professor of medicine from the Korea Advanced Institute of Sciences and Technology and co-founder of Thor Therapeutics and Silk Longevity.
Is this the?
Yeah.
Okay. Thank you. Yeah. Okay. Good afternoon. So I'm Minho Song, professor at the KAIST, and I'm the co-founder of the Silk Longevity. So I would like to introduce our Korea People Medicine Initiative in next 20 minutes. So Korea will become the super-aged society by 2025, with more than 20% of people aged 65 or more. So if nothing changes, healthcare spending will jump from 9.9% from GDP in 2023, and it is expected 20% by 2060. To stop this speed trend, we are adopting people medicine, combining AI with multiomics data. So our 25 and 35 goals is to cut the age-adjusted burden of chronic disease by at least 15%, and keep person healthcare cost growth before overall GDP growth. So we'd like to expand the people platforms to 5 or more partner nations. So Korean government already launched the Korea Bio Big Data project in January 24. After the 3 years pilot, that's sequenced 20,000 genomes. Phase 1 will enroll 772,000 people by 28, and phase 2 will reach 1 million participants by the 2032. So actually, we also combined the COGES, Korea Genome Epidemiology Study. So 211,000 people with 20 years of follow-up data, and also the National Biobank, over 1 million stored samples. So our government Bio Big Data Cloud can hold the petabyte of data and is ISO certified. And we use the mobile consent application and the federated learning at 12 major hospitals, so data never leave their site. And with the 1.4 trillion Korean won budget through the 2032. So on this slide, I would like to show our global people medicine network with the partners in Europe, King's College of London and SciLife Lab KTH build the explainable AI models. And Turkey and Korea. In Korea, the KAIST team in Seoul, Sejong, and the Kimhae manage the population cohort, and the Silk Longevity is generated the data platforms. So we can expand the if Vizy joined in this already Vizy joined in this global network, so we can expand our expertise with the Vizy, and also I'm very happy we may connect to the Pitt Med AI global health platforms. So we gathered the data on multiomics and from the pregnancy to the adulthood. So we feed all of the omics layer into our longevity AI engine, and which finds the key disease pathways and the risk patterns. And by combining AI-driven analysis with public education and the active data sharing, we build a collective health ecosystem. This lets the doctors, patients, and the communities work together to spot the problems early and stay healthier and cut the cost of the chronic and the degenerative disease. So we start by collecting 8 types of the multiomics data. Next, we feed this data into our big data platform. Our AI and the systems biology engine finds the important pathways and the disease markers. Then we turn those insights into personal health advice, what to eat and what to take, what tests to take, and how to live healthier. And we also use these findings to create group programs like the longevity, sports, and wellness package for the large communities. After that, we build the digital twin of our study group and run the scientific and the clinical trials to see what really works. And finally, we send each person each follow tips through our mobile AI coach and disclose the loop from the raw data to daily health actions at every stage of life. So we support the health at all very early stages. Before pregnancy, we are going to do the carrier screening and the pregnancy and newborn and the prenatal and the newborn test in child, the growth monitoring, adulthood, the chronic disease risk check and midlife, and the senior years, we are going to do the prevention of aging-related illness and the support to the longevity. At each stage, we run key tests, whole genome sequencing, microbiome analysis, proteome and metabolomic profiling, and feed the result our AI engine and receive the clear personalized advice and the action plans through our secure mobile app and the reporting systems. So we deliver the people medicine in 3 channels, 3 ways. And with the hospital, we partner with the major hospitals to run government-funded cohort and they build the digital twins. And with the government, we set up the citywide cohort in Kimhae, Sejong, and Gwangju City using the federated analytics and the local budget. And also with the B2B, we learned the multi-omics clinical trials and the license our AI platforms to food and the supplement companies. Together, these 3 channels spread people medicine widely and create steady revenue and speed up innovation. So I'd like to share some example from our Big Life project, which is going to be started from 2027 and 2032. We will run 10K Kimhae citizens in 5-year studies, longitudinal studies, and the participants will give the blood samples, track their daily habits, and share the basic health measurement. We'll use the sequencing and the AI to build the one unified database, and this database will help us to spot disease signals and more accurately and improve our risk models and drive down cost as we scale up. So our Kimhae Big Life Digital Twin Code has been featured on the major news channels in Korea, and these stories boost the public confidence and strengthen our reputation as a multi-omics leader and spark the new partnership. And we'll keep using press coverages and the symposium appears to showcase our work and to attract the collaborators inside of the Korea. So this is another example of our works planning. So we work on Sejong City and the clinics to pilot the multi-omics health guide. The participants get the whole genome sequencing and get an oral microbiome profiling and the metabolic testings. Sandbox funding covers the all fees for the sequencing analysis and the personal coaching. This pilot shows how multi-omics can work smoothly under the flexible regulation. So we work with the food and beverage and the other wellness companies to track the molecular changes before and after their product use. Our AI reports turn complex multi-omics data into simple and clear insights. So these insights guide the product design and prove efficacy and the smooth path to the regulatory approval. This story-driven process speeds up R&D and brings the personalized wellness solutions to the market faster. So I'm very honored to congratulate everyone involved in the global 1 million people medicine initiative in Pittsburgh. So in Korea, 5 universities and 12 hospitals are on board, supplying the clinical expertise and the patient cohort we need. Together, we'll merge the multi-omics, clinical, and lifestyle data to deliver people healthcare to people around the world. So we've joined force the 5 top universities and their hospitals, Seoul National University, Yonsei University, Catholic University, Inje University, and the Suncheonhyang University. So together they provide over 15,000 hospital beds and 30,000 outpatient visits each day. And it is estimated 60% of Korea's leading clinical researchers. So our core clinical leaders come from the top 5 hospitals and covering the care from children and the senior across Korea. And this team brings the deep expertise and the broad patient access to nationwide in Korea. So in May, we will open the Silk Longevity in Sejong Lab in Sejong City. The facility houses the latest gene sequences and the multiomics platform and the powerful AI computers. So Sejong City is the Korea's new administration hub, just 1 hour from the Seoul and 2 hours from the South City, Busan. So it offers the modern data center and also has the nice infrastructure, for example, fast internet and strong government support. So we choose the Sejong for the 750 square meter lab, which houses the gene sequencer and mass spectrometers in the future, and also powerful AI computers. So actually, its location makes the collaborating with the Seoul, Daejeon, and the hospitals very easily, while low cost help us lead in multiomics research. So we are now setting up the NGS and the sample prep benches and the analysis rooms. We've built the shared spaces, small auditorium, meeting rooms, and the lounges, and also in the kitchens. The floor plan clearly separates the prep sequencing and data analysis zones. So we expect the final inspections and the equipment checks to wrap up in the next few weeks. So we start with the newborn screening to catch the treatable genetic disorders, and then follow the oral health testing, Echo-Dells testing, and which is based on the microbiome analysis. And also we are trying to add plasma or metabolomic studies and lipidomic studies, and also we are going to do this package tests. So we get each person's multiomics data and feed it to our AI engine, and also we are going to planning to provide this information into the mobile coaching. So in the final slide, I'm very honored to congratulate the PINMAD AI Symposium, launching the global 1 million precision medicine initiative. So I look forward to working together to build this global multiomics database. Thank you very much.
Are there questions for Dr. Shaw? Thank you. Thank you. Okay, our next speaker is Dr. Fritz Roth. He's the John K. Vries Professor of Computational Biology at the School of Medicine here at the University of Pittsburgh. Thanks, Fritz.
Okay, we've heard a lot of big visionary stuff today. I'm going to say something a little smaller, which is I'm going to start by explaining something called missense variant effect maps. But the goal here is to solve the basic problem of, let's say you have all the phenotype data in the world and a genome, and you see changes in that genome. How do you know which changes are damaging so that you can connect genes to phenotypes? And so, one statement of this problem is to say that as we sequence more and more clinical genomes, or more often gene panels, as we find more variants, We are indeed finding more benign and likely benign variants, more pathogenic and likely pathogenic variants, but we're also finding conflicting variants, and those are all down at the bottom because mostly the plot is dominated by variants of uncertain significance, or VUSs. And so this is a giant shoulder shrug in the genetics game, and it makes it hard to know whether a variant you see is a passenger here or is a driver of the phenotypes you're seeing. So in the clinical world, this is the pejorative term of reactive functional testing, and I'm going to take on board this sick care idea because basically you wait till someone has a phenotype, a disease, and you sequence still usually a panel of genes that are smoking guns for that particular phenotype, and then if the clinical lab finds a variant, they can't wait for more data. They can't order more experiments. They have to deliver a report to the clinician. And more than half the time now, especially as we're in the long tail of rarer and rarer variants that we haven't seen before, that's a VUS report that goes back. If you happen to have a little functional data, which usually comes months or years after a clinical variant is first observed, so it isn't there most of the time, but if you have it, you can resolve maybe half of those VUS, get down to a 25% VUS rate. And the trouble with this is there's this back-of-the-envelope calculation that is kind of mind-blowing, that the rate of new mutations appearing in humans is such that every possible variant in our 3 billion base haploid genome, there are 3 changes at each position, and they are all present in someone alive today, on average 40 times over. Of course, if they're dominant lethal, they're not there, but mostly they aren't. And so they're almost all there. So why are we testing them one at a time, months or years later? So I also saw the word proactive earlier. And so the idea is, can we be more proactive about this? And so standing on the shoulders of giants, starting in the late 80s with people who did alanine scanning, Cunningham and Wells, and then advances from Stan Fields and Dan Bolin and others took this to everything scanning, where you make all 19 amino acid changes at every position of a protein, and then test, actually make the clone, make the protein, test its function, and make a map of which amino acid changes are damaging, here in blue, or which amino acid changes are neutral in white, or are occasionally better than wild type in this assay in red, although usually that doesn't mean they're better for us. So that's the proactive mode. Let's test all the variants ahead of time, and then when someone comes into the clinic with a variant that hasn't been reported clinically before, we've already tested it experimentally, and we can do a better job. And why am I focused on missense variants? I should have said earlier that if you look in ClinVar, the database of clinical variation, most of the VUS variants are missense variants. And most of the missense variants are VUS, as I just showed you. So this is where we're focusing our energies. And so my lab, when I heard about this technology, I think we've added a little bit on the technology side, but mostly I just thought this was an amazing thing that we just needed to do. And as we did it more and more, we learned that each protein, each genetic disease is its own special flower and is interesting and is worth all of a lab's time for the rest of a career. And I feel bad moving on to from each gene to the next. Most of my career, I've been a many-gene, genomics person, and now in this latest phase of my lab, I feel like a one gene at a time guy. I'm coming around full circle or something. But we've done 20 maps for 19 to 25 proteins, depending on how you count. And here's a list if anybody wants to. Oops, I got to go back. Oh, the list is gone. Okay. Anyway, there's somewhere there was a list of genes that we're working on. I'm only going to talk about one of them today, and that is the LDL receptor. Okay, there's some sort of delay here. Okay, there we go. The general paradigm for this, I'm not going to go into the weeds here, but it's oligo-directed mutagenesis. You make all the changes in one big batch. You move that library of changes efficiently through a vector system into whatever your assay is. That's the whole art, is rigging a cell, yeast, human, whatever it is, to be an efficient, scalable, multiplexable assay of variant effect. And then you have a little lab evolution experiment. You sequence before and after the selection, or you sort for greenness if it's a reporter assay, and you sequence before and after. And then at the end of the day, you sequence and get the results back. Oh, there's my gene list, okay. So if anybody cares about any of these genes in particular, give me a shout, maybe, or if you have a new one to propose. I love to talk about new projects. Come by, but I'm just going to talk about the LDL receptor. And we've actually heard about that in a few talks, and I think we're maybe going to hear about that from Dr. Goswami next, who's a real cardiologist who's going to correct me when I mess up here. Okay, so I'm going to talk about the LDL receptor. There we go. And so the LDL receptor is the single largest source of genetically caused elevated LDL. And in fact, I've been told it's the single largest genetic source of cardiovascular disease. And it sits in the receptor, and it's got that baby blue arm that wraps around the LDL particle, the low-density lipoprotein particle, which is basically a ball of lipids surrounded by the ApoB protein. And that little light blue arm wraps around it, and the whole package gets endocytosed, dragged to acidic endosome where LDL is released, degraded. LDLR is mostly recycled back to the surface. And so why would we want to test lots of variants? Why do people order genetic tests for LDL receptor? Why don't we just test LDL levels, and if there's a problem, we give drugs to lower LDL? It turns out that if you have one of these so-called FH variants, familial hypercholesterolemia variants, which are mostly in the LDL receptor, if you have one of these variants, you have poorer outcomes, a higher risk of coronary artery disease across all LDL levels. And these people benefit from statins or other LDL-lowering meds, even if they exhibit normal LDL. So this is about being proactive, and potentially, if you can recognize variants, you can tell people at birth that they will benefit from LDL-lowering therapy early. Okay, so no surprise, there's almost half of the variants in ClinVar are VUS. So what did we do? We coupled an LDL Frodo. So the Frodo dye got coupled to LDL. It's pH sensitive. It glows green after LDL has been taken up into the acidic compartment. And now you can use that as an assay. So the cells are green if they took up LDL. I'm not going to go into the weeds on this, but there's a cool method called the BXB1 landing pad where you can put a single variant version of LDL receptor, or whatever your favorite gene is, at a safe harbor site in a cultured human cell. And so we can make a whole collection of HeLa cells where every HeLa cell is expressing a single LDLR variant at a safe harbor site. And then we can do fluorescence activated cell sorting, and we can count the frequency of each variant in the high green bin versus overall. And then we use a method called BARSeq, where we've done long read sequencing to associate the full-length genotype of LDLR to a flanking barcode. And then we can count only barcodes in high green and low green cells. And I looked up some of Fiona's papers and found that we had both been authors on the original BARSeq paper back in the day, which is kind of cool. All right. So this is the drum roll slide. There we go. So there's our variant effect map. We tested all 19 substitutions at every position of LDL receptor. And there's the map. A lot of blood, sweat, and tears got swept under the rug there. There's lots of stories to potentially tell. I'm gonna drill down on one. But the first thing that made us feel good is that the scores were almost uniformly high for benign or likely benign variants here, BLB, and they were a mixed bag for pathogenic and likely pathogenic, but if you had a low score, you were really likely to be a pathogenic variant. We looked at how did this relate to risk of having high LDL, and we also wanted to know how it related to polygenic risk score, so what you might pull together from lots of common variants across many genes. And you can see that if you have a low-risk polygenic risk score and there's no damaging rare variant in our map, you are indeed protected from high LDL. But in fact, if you have a protective PRS score and a damaging variant in our map, you are at elevated risk of high LDL. And if you have a high-risk PRS score and you have a damaging variant in our map, you're at really high risk of having high LDL. And I won't go into but there's earlier age of diagnosis of high LDL. The only thing I'm going to say about this slide is that even amongst, and I should have said this is UK Biobank data that we're mining here, and it would be great to mine the kind of data that we're talking about here today, but mining UK Biobank data, even the participants in UK Biobank who already are known to carry a known pathogenic variant, yes, their LDL risk is high LDL risk is higher, but it's quantitatively higher if they have a damaging variant in our map. So our map is quantitatively adding to the assessment of risk of, in this case, of having FH, familial hypercholesterolemia, if they have a more damaging score in our map. All right, so I'm going to come back to this little arm that wraps around ApoB and grabs the LDL particle. And it's made up of 7 conserved repeats called LA modules, or LA repeats. And those are important for binding LDL, as I mentioned. And the ones that look good, those sort of blue vertical striped ones, conform exactly to what we would have expected. And so you can see many of these vertical stripes you can't see. But anyway, I should have said yellow is the wild type residue. And many of those are cysteines, which is, I think, 3rd or 4th from the bottom. And so there are these conserved cysteines, which are involved in disulfide bridges. It all makes sense that these are perfectly conserved. There's a calcium cage, and the residues around that are perfectly conserved. Those all make sense. But what the hell is going on with LA1, 2, and 6? The literature said, well, LA1's a little more controversial, but the literature said that LA2 through LA6 are basically the same in terms of their importance to LDLR function. And yet we found 2 and 6 to be night and day, less important than the other ones. So what's going on there? And if you look at a structure that just came out, it actually kind of makes sense. LA1 is completely unstructured, flapping in the breeze, not connected to ApoB. LA2 is set back, LA6 is set back. So in one level, it makes sense. We missed those, but they're also not even touching ApoB in the structure. But actually, it doesn't really solve the question because there are bona fide pathogenic variants in LA2 and LA6 that we missed. So what's going on? I mean, so in one level it makes sense, but still, why did we miss it? So I'm not going to save you another 20-minute talk that I now insert here, 20-minute talk where I take you down wild goose chases, wrong hypotheses, and then finally we do another map in the presence of high levels of VLDL, which is a larger version of LDL that's a competitive inhibitor of LDL for binding by LDL receptor. And it turns out that if you have more competitive inhibitor around, LA2 and LA6 variants start to become more important. And so the idea here, the implicated role here, is that LA2 and 6 aren't so much important for taking up LDL directly. They're important for excluding a competitive inhibitor. And so this is all a long tail, really, to say, you know what, variants aren't just constant in their effect. They are dependent on environment. They are dependent on genetic background. I've got other stories that I haven't told here. And AI is getting better at doing what we're doing. And sometimes it's so good we don't start projects anymore. But what it doesn't do is tell you which variants are going to be changing in their effect depending on environment or genetic context. Okay. I'm going to skip over much of this, but just to say you can calibrate this data and translate it into the language of the ACMG clinical variant interpretation approach, which the clinical labs need to classify variants. And so we are providing variants from our map for over 10,000 amino acid substitutions. That's not to say they're going to resolve those VUS. We don't know that. We're working with Ambry Genetics to figure that out. But we do know that we're providing new evidence, functional evidence for over 10,000 amino acid changes proactively. So just to sum up, we did LDLR, and I should have said maps because I didn't tell you about our surface abundance map. I did mention the plus and minus VLDL. I think I've said most of this, but I think the key take home here is that variants are hard to interpret, that functional data helps, and that environment and genetic context can change variant effect, and that AI isn't yet doing that. So lots of people to thank, and so I'm going to jump to the last part of the talk, which is the Atlas of Variant Effects Alliance. Very quickly, I know Mark's getting worried, but I can do this in 3 minutes. Okay. So what is this? So I would encourage people to go check out this genome biology paper from 2023. We're working on another white paper that does this from a more clinical perspective. But the idea is there's a whole growing, I'm not the only person, we're not the only lab. Hi, lab, thanks for coming. Talk to them later. We're not the only lab doing this kind of work. There's a growing international community with this shared vision of let's functionally, experimentally test every variant in the human genome, at least in known functionally important regions. And actually the collaboration is larger than that. We're interested in pathogens, model organisms, evolution. It's a bigger field than just human disease genes. But there are implications of knowing which variants are bad in clinical diagnostics, of course, in protein science, drug development, precision medicine that we've been talking about. How does disease work? LA2 and LA6, is familial hypercholesterolemia different for people who have variants in those two LA repeats? I don't know, but now we have something functionally specific about them. We can subclassify variants and begin to look at disease subtypes. So we're going from an era where we have maybe 100 of these maps published, or maps published for about 100 of these proteins in humans. And now the Sanger Center, new director Matt Hurls was a co-founder of this Atlas of Variant Effects Alliance, and they've turned the might of Sanger Institute mostly over to this kind of work. So expect lots more of this data soon from us and others. And so this is really the last slide, and I hope you'll check this out. There's a great seminar series, and if you want to get updates from this community on how this large-scale effort to test variants is going, please reach out. But I think, actually, I just want to say here, I skipped over this. One of the major challenges, and I did want to put some AI in here at the end, AI is getting better at this. This data will provide new training data to make AI still better at this. And I think we need to come up with a new paradigm to look at environment or genetic background dependence of variants, where we test some, but not all variants across different environments and genetic backgrounds, and then use AI to fill in the gaps. And I think that's a real potential for a marriage of AI and experiment here. So thanks everybody for your attention, and I'll stop. Are they doing the same for regulatory regions? We are not, but other groups are. There are many groups working on that, and you may know Jay Shanduri and Nadav Ahitov. They have an effort. They've published a bunch of this where they went systematically across promoters doing these variant effect maps. It's harder to calibrate clinically because there's just so little data out there on what's pathogenic and what's benign and what expression level change you need to see in a transcription regulatory element to be disease relevant. But it's happening. People are doing it.
Yeah. This is a philosophical question. Maybe at the end of it, I will convince you to maybe add 2 more genes to your
okay.
Make it 21 to 27 genes. So you talked about LDLs, right? LDL has been controlled by excellent statins, and there's also a PCSK9 inhibitor that controls it very well, and I'm told it might be the standard of care when the prices for that thing come down. So I'm curious, why didn't you focus on LPA? Because there are 2 genes, there is no drugs for it. 25 to 30% of the US population have higher levels of this particular bad molecule. And then, though it is measured, it occurs in small numbers and is measured in nanomoles, but yet it's got 6 times greater propensity to cause atherosclerosis than LDL.
Let's go.
So why there are no drugs for it?
Let's talk after, and we can sit down and see what the challenges would be.
I'm not a cardiologist. I'm not a physician.
No, that's it. You signed up. It's too late. Thank you. That's a good suggestion.
So you're suggesting that we can do something like AlphaFold does, that you have some map where we read information and then we train a model to predict other.
Well, there are already many variant effect predictors, and in fact, one of them called alpha missense, in theory, made some use of AlphaFold, although in practice, it's not clear it helped them do the great thing that they did. But that's one of the best predictors now is alpha missense from the same group that did AlphaFold. And they are getting good. It's for sure. I mean, they are better than the experimental data sometimes. But the trick is knowing when.
So the variants are suggested, they are not causal.
Sorry, I can't.
Variants, for example, I have an Alzheimer's patient I'm looking at, and at some location, chromosome 19, there are variants there. But this variant is just suggestive that the patient might have, but it's not causal. It does not always lead to a functional.
Yeah, this is a much bigger question, the question of penetrance, of is this going to cause disease in this person, not is it generally pathogenic? And then there's the whole other challenge of if you have asymptomatic people and you see a variant, it's much harder to say it's pathogenic because your prior is much lower. It's a longer conversation, and Mark's going to take me off. Thank you.
Thanks, Fritz. Actually, he's done well on time, so we're probably going to have a bit of a longer break. But our next speaker is Dr. Vivek Goswami. He is the head of the board of directors of the American Heart Association in Austin, chairman of the nuclear medicine department at Austin Heart as well.
All right, good afternoon. Okay, so first of all, I hope I'm not offending anybody by wearing my University of Texas burnt orange while visiting this beautiful campus. I really want to thank the University of Pittsburgh for hosting this symposium. I think the integration of multiomics in clinical medicine is going to be the largest transformational change we've seen during our careers in terms of advances in medicine. I finally feel it's going to bring medicine up to where it should be in terms of technological advances. So appreciate the foresight there, and I want to acknowledge the speakers that have spoken today. Very well accomplished and eloquent, and just want to commend them for all their contributions to multiomics, AI, and in medicine. Unlike the other speakers, I have no data. I'm a clinical cardiologist. I'm a preventative cardiologist in Austin, Texas. So this is going to be very different. In fact, I don't have any slides, so I don't know if that's good news or bad news, but I have no data to present. So this will be very conversational, and what I hope to achieve is really describe through my observations as a physician who sees a lot of patients. I'm in a large private practice group. Some of the things I think we do well in medicine, and a lot of things I think we could do better. These are my own views from my own perspective. It's not to slight anyone who's practicing medicine or to question anyone's motivations. And in actuality, I think academic medicine is wonderful. Unfortunately, just a small percentage of our patients end up at teaching hospitals for their care. So with that said, I'd like to start by talking about what I think we do well in medicine. I think if you get sick in the United States, we have a relatively well-oiled, efficient process. So if you break a hip and you go to the emergency room, the pathway to get you admitted, having the appropriate team come see you, and eventually get you your hip surgery 48 hours later, I think that's pretty efficient. I think we do that well. Arguably, we do it better than any place in the world. If you come in with a heart attack and you need bypass surgery, the sequence of events for you to come in through that ER and get your bypass surgery, I think we are very, very efficient, and we do that really, really well. What I don't think we do well is keeping people healthy. And that's evidenced by the fact that despite spending more money than any developed country, we're not even in the top 10 when it comes to longevity. We're not living healthier, we're not living longer, and we're not necessarily living happier. I think health span, which is the amount of healthy years we're living, so not just longevity, but how many of those years we're living healthy, I think is a understated metric. And it's something that we should consider. If we take heart disease, for example, I'm going to use a lot of heart disease analogies for obvious reasons, but a lot of what I'm describing will apply to other diseases as well. But if you take heart disease, for example, in the last 12 years, for many demographics, we've gone the wrong direction. So despite all of our advances in medicine, despite drug-coated stents, despite medical therapy, imaging, our surgical techniques have improved, decrease in door-to-balloon time when you have a heart attack, how long it takes to put that balloon up. These are all advances that we've made, and many more. For a lot of demographics, we're going the wrong direction. More people are dying of heart disease than ever before. And part of the reason is because for a majority of folks, the first symptom of heart disease is a bad one. So most people, the day before they have a fatal heart attack, they're sitting at home and they feel fine. So if you don't make it to the hospital in time to use all of our advances, we might as well live in 200 years ago, right? So majority of the people, before they have a fatal or a large heart attack, they feel fine. So that reactive mindset, the more we focus on reacting to heart disease, will never put a significant dent in the mortality related to heart disease. That trend is true for other diseases as well. Obviously, if you look at diabetes, we're going the wrong direction, right? In 20 years, we're going to double the amount of people that have diabetes in the United States. If we look at obesity, metabolic syndrome, chronic kidney disease, stroke, dementia, if we look at a lot of mental health diseases, we're going in the wrong direction. So I guess the question is why? Well, there's probably a lot of reasons, and one of those is I think our financial carrot is misaligned. I mean, we get paid a lot of money when people are sick in the United States. So when you're doing something, a procedure, imaging, etc., that's when the payments start. There's not a lot of money in our system when people stay healthy. And it's not to say that healthcare providers are malicious in their focus. I think some of the best people I know go into healthcare, great humans. I just think our system from a very, very early stage, from our training, programs us to think that way. All those years of training to be a cardiologist, all the med school, the residency, the fellowships, you name it. There was never one class on exercise or diet. I mean, it was first day, here's how you hold a catheter, here's how you blow up the balloon. So we're almost programmed from the time we're in training to this is what we're supposed to do, and we do a very poor job of staying ahead of disease. It's just not in our training. In addition, I think as a society, we're not really accountable for our health. We're working more, sleeping less, eating poorly, making a lot of excuses for why we're not doing the things we do. And again, just like the physicians, I don't think 90% of our population woke up and said, I want to be metabolically unhealthy. I think this is something that has basically surrounded us as part of a lifestyle. And it's become kind of interesting to the point where we've normalized unhealthy. So if you get out and you start running 3 to 4 days a week and you're eating healthy, your neighbors will probably ask you what you're training for, why you're working so hard. So I think it's interesting that if you look the way you look, you're considered too skinny. If you're exercising a lot, you're a health freak. I think that should be the norm, and everything else should be abnormal. So I think all this has kind of contributed to the unhealthiness that we're experiencing. So I guess, is preventative medicine the answer? I actually think it is. I think it's a huge step in the right direction. But it's not as easy as it sounds. So when we're creating a preventative model, we are trying to basically put clinical pictures, pieces of a puzzle together about the person sitting in front of us to form a clinical picture. And then depending on the individual's risk, we'll treat them aggressively or maybe very conservatively, depending on what we come up with. The problem is the pieces of the puzzle are not necessarily precise, and they're not necessarily specific, and they're not necessarily complete. So for example, we were just talking about LDLs. So the interaction between LDL cholesterol and heart disease is the most heavily studied interaction in medicine. There's more prospective studies with LDL and heart disease. Despite that, we still don't have it all figured out. There's people that have high LDLs that don't have heart disease. There's people that have normal LDLs that have heart disease. So, and in fact, it's actually a very poor differentiator for who will develop a heart attack or not. If we look at people that have heart disease versus people who don't, 80% of them have the exact same LDL. So while it's true if your LDL is excessively high, you're at high risk, excessively low, you're at low risk. A majority of people, you can't tell the difference based on the annual cholesterol your doctor's getting you to figure out who's gonna have a heart attack and who's not. So we need something more specific. We do see a paradigm shift. There are physicians such as myself that are becoming preventative cardiologists. I never thought I would be a preventative cardiologist when I was a fellow. It took me about 10 years into practice and reacting to disease with procedures, et cetera, to make me realize that we were spending a disproportionate amount reacting and not enough amount preventing, and that's where the gap was discovered. Patients themselves are tending to move to a more of a preventative pattern as well. So patients are more educated than ever before. They're listening to podcasts, they're watching YouTube videos, and they're reading books, and they're coming to the doc asking for specific tests. I know someone just mentioned Lp(a) or ApoB, so patients are taking it upon themselves saying, hey, I have this job, I have kids, how do I stick around and not let those happen? And I think it's our job to make sure that we see that through appropriately. So here's where I see multi-omics coming in. So when we're forming this picture of the person in front of us, a lot of those metrics are somewhat generic. We're very binary in our thinking when it comes to lab tests, and we create what's high and what's low and what's normal. The reality is 3 people could have the exact same lab test, and that can manifest in 3 different ways depending on what's happening in the person sitting in front of us. So a missing piece of the puzzle is what's happening that's unique to that person that may predispose them to a certain illness. What's happening in that person that may make them more susceptible to experience a side effect with a treatment or respond better to a treatment. We don't really do that. If we have 3 people that have the same cholesterol or the same blood sugar or the same blood pressure, we generally treat them the same way when those people could be very, very different. So very often we'll have patients ask, how do I know this drug is working in me? It's a very reasonable question. And you know what the answer is? The answer is, we really don't. We just know that when we did randomized controlled trials with 30,000 people that look like you in terms of their inclusion criteria, it benefited them so it should benefit you. You can see how that doesn't necessarily sit well with people. And when we look at those randomized controlled trials and we really analyze the benefit, number needed to treat is a very important statistical metric. How many people do you need to treat to prevent one event? For statin trials, that number is 50. 60 is a typical number needed to treat, meaning for every one person that benefits, over 50 people do not. So what if there's a way that we could be more precise with who could benefit from treatment and who could go without treatment? So I'm sure you guys have heard this analogy, but you know, if there's a tree and there's a branch of that tree that's diseased and you call in an arborist, the first thing the arborist will do is take a soil sample to figure out what's deficient in terms of going into that tree to cause that disease. And maybe they'll supplement that soil accordingly. That's how medicine should work. But medicine doesn't necessarily work that way. Medicine will treat the individual branch, and if another branch gets disease, we'll treat that branch, and if another branch gets disease, we'll treat that branch. And you end up on a bunch of medications. And it gets siloed even in our training. They ask us what kind of doctor you want to be. Do you want to be a cardiologist, a nephrologist, endocrinologist? And then we see that organ system just as that organ system. But obviously we can't treat the heart without treating the kidneys, without treating the endocrine system. All these things are interrelated. So even though our training and the way we practice medicine is very siloed, that's not how the body operates. So I see the incorporation of multiomics in medicine helping to have more of a holistic approach to our patients and personalizing our risk stratification models to figure out what that individual, not someone that's like him, but what that individual is likely to develop and respond to. So I see a scenario where someone has high cholesterol, but based on their pattern, we may not treat that cholesterol because that pattern is not indicative of development of disease. I see people who may have family histories. Their father had a heart attack at 57. Now they're 57. They're real nervous. They're anxious. Is this going to happen to me? I see multiomics being used to help us with that pattern recognition to say, hey, this isn't necessarily something that's genetic. That was learned behaviors, maybe because your father smoked or had poor lifestyle. So I think this could be very beneficial in terms of serving a sense of calm as well. And then I see, you know, changes in spikes of various proteins that can predate actual disease. So, you know, we're following these patients annually, and all of a sudden, 6 months before someone develops pancreatic cancer, we could see the signaling, thereby telling us that we should be screening that patient a little more closely, or weeks before a heart attack, looking at individual mechanisms and saying, hey, this heart attack was driven by inflammation as opposed to LDL. Maybe we should repurpose anti-inflammatory medicines to help treatment. So I think this is the personalization that's missing from medicine today. And as a result, we are starting to incorporate this with the help of the lab that's being created here into our clinical practice. And we have lists and lists of patients that, when it's available, are ready to get the test and help implement it clinically. So I'm excited to be here. That's my vantage point from just a very practical sense, seeing a lot of patients, knowing how patients think, physicians think, and healthcare systems think. But I think this could be very transformational, and I'm excited to be on the front end of it. I'm happy to take any questions. Sir.
Why is the maximum death in cardiology space? Is this due to that there's a lot more unknown in the cardiology than other diseases, or that, you know, the factors which drive the disease are multifactorial and we are never able to predict?
Yeah, so, right. So the question being, why is heart disease the leading cause of death, essentially, right? I mean, to some degree, the formation of this plaque in your arteries is a normal process of aging. If we live long enough, we're going to form some plaque in our arteries. It's just a bell-shaped distribution, some people forming it early, some people late, and some people as expected. The unfortunate thing is the day before someone has a heart attack, that plaque is non-obstructive. You know, our approach to prevention of heart disease is very archaic. The Framingham Heart Study, which is over a half century old, that told us, hey, if you have diabetes, if you smoke, if you have a family history, if you have high blood pressure, et cetera, you're at risk. The amount of physicians that still use primarily that to assess their risk is incredible. And actually, even cholesterol, when you look at your HDL, your LDL, and your triglycerides, that lipid panel is the same lipid panel we used when I was a med student 30 years ago, right? And the same lipid panel they used 20 years before that. So the reality is we've been very, very slow to understand that there are other drivers of disease besides cholesterol and besides sugar, including Lp(a) and ApoB and inflammatory markers, and things we don't understand, which is another important metric of multiomics. Someone had mentioned PCSK9 inhibitors earlier, and I think the way that drug class was developed is fascinating. I mean, these labs were in Dallas, UT Southwestern, in Paris and Montreal, were following patients that had FH, exceedingly high levels of LDL, and had a lot of heart disease as a result. And they noticed all these patients had high circulating levels of this protein. And then when there's a mutation in the gene that rendered that protein inactive, these patients had really low LDLs and almost no heart disease. Well, that protein is PCSK9, and as a result, it became a therapeutic target. That's a great example of looking at various proteins, receptor, transcription factors, et cetera, to see what trends correlate to disease, where we can use them as future therapeutic markers as well. So I think we've been very, very slow to understand that there's other things that are driving disease. Lp(a) is exponentially more atherogenic than LDL. Exponentially. It affects 20% of the population. And in the last year, the ordering of that test has gone 900-fold, but why weren't we ordering it 10 years ago, 5 years ago? There's doctors still today that don't order it, right? So very, very, very slow to be proactive. And the other thing I'll tell you is that we screen for breast cancer, we screen for colon cancer, mammograms, colonoscopies. Routinely, a lot of people don't get screened for heart disease. We judge the book by a cover. Do you have risk factors or not? We treat the risk factors. The risk factors are not a one-to-one correlate. So absolutely everyone should get screened for the actual disease. Doesn't matter if you're speeding or driving the speed limit. If you're getting in car accidents, you need to be protected. We should be screening for the accident, not the risk. And we don't do that. Was there another question? All right, well, thank you for your attention. Really appreciate being here.
That's great. I'm going to fulfill an obligation and a promise I made to the Chancellor. Please pull your phones out and take a picture of this slide as a reminder of the event that's going to be occurring in October around AI and health. This then de-obligates me from doing it at the end of the day. So now we have a break. We have a break until 4:30, so please be prompt and come back at 4:30. Rashidi. He's the Associate Dean of AI in the School of Medicine and a professor in Lombardi. Shinozuka, Endowed Chair of Experimental Pathology Research, Executive Vice Chair of Computational Path and Informatics Division at UPMC, and Executive Director of CPACE. Thank you, Huma.
Thank you so much. Thank you, Mark, for that. And this is what advances it, right? Perfect. So, well, thank you for joining me, and I hope you enjoyed this quick talk. My talk is a little different than all the great talks that you saw earlier. It's more in tune with where our vision is with respect to our educational and operational missions within our schools, and I'm starting within the School of Medicine, and also showcasing what one of our centers did in terms of, trying to expedite some of those things, although we work with many other centers and departments, So some key things to note, I'm following a DVP rules. This is kind of in tune when you'll see it at the end of the talk because I'm kind of copying Jason Rosenstock's recommendation to do these things. So I'm disclosing that ChatGPT and Napkin AI were used and outlined in certain figures within this talk. I verified all the material included in this talk, and I was protective that some of the data that I'm going to show you is not real patient data. So there's no sensitive information used in anywhere. And you'll see why that's relevant. Just to make sure we're all on the same page, I want to kind of quickly take us all, so I apologize if I'm making it too basic, just in terms of what is AI, a brief history of AI, and also Gen AI, traditional AI, and especially within our educational landscape, and examples of some of these tools that we're enabling us to move forward, specifically within our AI center. So exactly artificial intelligence, I'm just paraphrasing Arthur Samuel and others that ML models are constructed by a set of data points and trained through mathematical and statistical approaches that ultimately enable prediction of new, previously unseen data without being explicitly programmed to do so. Basically what this says is you build a model, the model can now make predictions on future unseen stuff. Let's say you now can identify cancer versus no cancer, or if it's a ChatGPT model, it can now give you an answer on some question that you have, but without it being any more like directly programmed in there. Some examples of what we call ANI, are Siri, Alexa. As you know, we're all using these in our day-to-day activities. So I don't have to sell you on that, especially the spam filtering is the great classic classification task that we've been using for a long, long time. But there's lots more that we use too within the AI in our day-to-day operations in life. But within the healthcare space, it's a different ball game. So if you use the AI or use ChatGPT and outside of the healthcare space, and let's say you make a mistake and it gave you the wrong recommendation for your travel plans or maybe Netflix recommendation, no major harm. In the healthcare space, a great deal of harm can come in there. So I think familiarity and knowing the limitations within this space is very important. None of these tools came out of thin air. I think it's very important to understand, that we're standing on the shoulder of giants and that they are based on decades of research and innovation within fields of statistics, machine learning, and so a quick little history of it that we have in this, if you feel free to read it in one of our recent review articles, where it started off with the Turing test, and then from there, went on with the idea of a perceptron, which basically was the unit of our neural networks that we basically dealing with nowadays, or at least related to it. Rule-based and advanced, approaches to follow, and then more sophisticated algorithms, and now we're basically in the era of deep learning platforms where those are complementing, and some people are arguing that have taken over the world in terms of what we do within AI. Not surprisingly, having been in this space for 30 plus years, I was very happy to hear that some of our folks actually were recognized finally for their work. So as you've seen recently, Jeff Hinton and his colleague John Hopfield received the Nobel Prize in Physics, although if you see the interviews with him, he's not sure why he got it in physics. That's a different story. And then Demis Hassabis for the AlphaFold and with his team in terms of what it's done. So finally, AI is being recognized. Important to note, AI is not one thing. So it's an umbrella term, and in my head, and I think most of us agree, we could separate them into 3 big categories, although I have other categories that I'm going to show you as well. So the one big category is what we call artificial narrow intelligence. This is our typical machine learning tools that we're dealing with, and includes the ChatGPTs of the world. And then what's coming in the future, and I'm convinced that it's going to come, is, and that's where I think Craig will probably give us a thing, because he actually showed me a glimpse of GPT-4 6 months before there was a GPT-4 when we were at Cleveland Clinic. So maybe he'll show us some cool stuff, pretty soon, but I don't want to guess. So AGI, although AGI right now, as I know it, although you could argue that we're in the glimpse of it, well, we're touching the AGI surface, but most of the stuff we're dealing with is in the ANI world. And then artificial superintelligence is really, you can think of that of the Terminator world. That's, hopefully we will never get there, or if we do, we find a way to control it. So I'm going to basically just highlight the ANI world. Within the ANI, it's very important, as somebody who basically be in the field and somebody who may want to, deploy some of these as you're basically reviewing articles or tools. There are 2 major types of them. There is generative AI, and these are things like ChatGPT, where they're generating, and it's kind of in the synthetic data world, if you will. And then there's the non-generative AI, and the non-generative AI is stuff that we've been dealing with for decades. I was a fellow, actually, for Mike Besage's informatics travel awardee, and we were doing, classifications and regression stuff, for a long, long time. So that's not a new thing in our world, So it's very important for us to separate these, because we're very familiar with the non-generative AI landscape, and they're part of our day-to-day operations, quite frank, and our research. And yet generative AI has now come in and disrupting things, and quite frank, it's going to start mixing with these 2 elements as we now have the multi-agentic frameworks to follow. So there's various things that the AI applications can do in education and healthcare. So tutoring, clinical support, research assistance. So I'll give you some examples of these as we go through this. So mainly, I'm going to concentrate in the first batch around AI education, specifically what we're doing within the school. And so in terms of personalized learning, real-time feedbacks, administrative efficiency, and knowledge access, but first and foremost of what we're doing with curriculum design for our students. So, because the goal here is, if we're incorporating AI in our landscape, to incorporate it the most efficient and responsible way, education has to be central to everything we actually follow suit. And quite frank, your efficiency of deployment goes up 10-folds, or I would even argue even 100-folds, if you have more educated AI champions and AI users at the end. And so what are our plans within Pitt to actually make us a premier institution, especially around healthcare AI education? And the way we see that is finding ways that actually truly democratizes AI, and not just a word, like in terms of what people usually say, let's democratize AI. No, we want to actually mean it, where it's truly accessible to everyone. And so what are our plans to becoming a premier institution and truly democratizing AI? And so the way we're actually planning on force multiplying our AI champions and bridging that major gap amongst the AI know-hows and the AI folks who are basically AI limited knowledge is through this 3-phase curriculum that we've now have in place. So the first phase we've already put in place, where it's 3 lessons for all of our students that basically teaches them the basics. We have now 8 new review articles that basically are now adding to our other 6 that we had at Cleveland Clinic, that now have 14 that basically are now giving access to our students to learn the basics. And we've developed podcasts and whatnot, knowing how busy med students are, so that they can actually do it more efficiently as well. The claim to fame of the curriculum is really the second spot, whereas basically what we're doing is something similar that we did at Cleveland Clinic, which was we deployed a no-code AI course there that's actually Cleveland Clinic wide. And so here we're doing something similar, but a more enhanced version of it, where we're going to have a lot more interactivity with 30+ AI apps, educational apps, so that the actual engagement from the user becomes fully interactive, along with a lot of AutoML tools and synthetic data that we're providing. So we're providing securities for the patients as well. And then finally, in the third phase of the thing is a smaller subset of the students or even clinicians may want to actually take an extra step. They may want to learn more about AI. They might want to be developers. So that's where we will have partnerships, and we currently have some, and we'll build more with known leaders within this space, like Department of Biomedical Informatics, computational biology and Fritz's group, and CMU, and other, SHRS, and others that have established, degree programs in that. So within this way, you're basically kind of like following this stepwise model as people gain access to AI knowledge, and then as they want to learn more, they can learn more and add on to their CV. So the key points about that elective course that I want to concentrate on, because I think that's what makes it unique, is it's a no-coding elective course, no software engineering needed whatsoever, and requires no machine learning experience. It's literally made for everyone. And so, and to show you what I mean by this, I think it's best to show you what the secret sauce is for this. The secret sauce for this is its interactive nature through its AutoMLs. So having multiple automated machine learning frameworks in there enables it to actually make it accessible to all. And then also we have numerous interactive AI education apps, which I'll show you an example of it too. So what I mean by this? In full disclosure, some of these are my own tools that I had developed that are now startups, and so Milo being one of them. So exactly how can you use Milo's AutoML to get anybody to be able to do machine learning? And so it's as simple as this. If you're building a tabular data set, let's say machine learning classification model for diabetes, this one happens to be diabetes S for synthetic. So you just upload the data sets for the diabetes. If you can attach a file into an email, you can follow and do stuff within Milo. It's that simple. It's drag and drop. So I can just now upload the synthetic data sets that we've had. It basically puts in the data set 1, the training initial validation, and then you put in the secondary data set, which is the generalization test set, and then you pick the target of interest that you're trying to build the machine learning classification model for. It's a simple classification test, so the students and users kind of get familiar. And that happens to be diabetes, and I go to the next step. So it follows best practices within machine learning in terms of what is it that we want to what is it that we brought in. So the second step happens to be a quick little univariate analysis in terms of the data that was ingested. So now if that looks good, you go to the third step where it's the brain of it, where you're now building out, and by the way, it's an open source framework, so you can pull the full Milo Pro in there for your things to use. And then, but for the sake of time, the pipelines that I'm going to build with these algorithms, with let's say logistic regression and KNN and whatnot, this then creates this wheel of pipeline that's now independently running multiple AI model generation pipelines. And so again, you can see that you don't need to be in machine learning experts and code it, because this did it for you, and you can now finalize and see what the model of interest was. And it shows you that the best model was the one that had the highest balanced accuracy with kind of sensitivity specificity average with the highest sensitivity, because most of our people want screening tools, but you could change it within the table and say, I want the one with the best F1, and so on and so forth. You can see that it's fully transparent too, so it had 11,000 models that were just built with the different neural algorithms. You can also deploy it and start testing it right away. So if you have a clinical trial and you quickly want to test it out, or like right now I just want to, let's say, automatically uploads the Python files within your LIS and uploads the information from the patient and then gives you a quick little response. Or I can manually do it and show that that person should have had diabetes, and it says you the confidence is pretty high, and it's a positive case. So this is a quick little glimpse, and of course there's lots of bells and whistles, and it will take me hours to show you all the other stuff. So this is one of the 7 apps within Milo that the users will get engaged in. So it's truly interactive. The other thing is around vision models. So and these are, by the way, the stuff that I'm showing you right now have nothing to do with Gen AI so far, right? So the vision model is a simple vision model that will do the same thing that I showed you in Milo, where we're going to upload basically a bunch of faces of myself in the cafeteria, and I apologize for the face, and And then you basically now upload it and you train the model. And you train the model, and within a minute or two, you now have a fully capable model that can tell a difference between somebody who has a mask or somebody who doesn't have a mask. But this could be any kind of an image. So it could be, flow cytometry. It could be, H&E, so on and so forth. I can open up my camera and start testing it right away. So that pickled model is ready to go. Again, no code whatsoever. So you can imagine how engaged somebody will be in that first 30 minutes of the course, just starting it off. But the main, and you can see it made a prediction of me with no mask, and then pretty good confidence, and then also with me with a mask, and then I can actually make it actually give an example of how well it actually performed. And then it did a good job. It said I had with a mask, but the confidence was not as good because the number of cases that I had was not pretty high. Bless you. So the best way I personally feel to actually teach more and more people is a no-code approach and an interactive approach. I think that's where I'm convinced from the past 10 years that's basically the way forward, to capture more people. And I have to say, the people who actually convinced me on this, to be fair, happened to be Barry, my old boss at Cleveland Clinic, and Brian Rubin, where they basically said, no-code approach is the way to go to get a lot more people engaged. So, and I have to say they were right, because we did an internal survey and we found out that more than 99% of our people were somehow fearful of the terminal. So not surprisingly, if you want to teach them code, they don't want to touch code. So you only have 1% of the people who would want to do the traditional machine learning coding approach, while 99% are still very interested in AI, but they don't want to touch the terminal or actually play with Python and open up their IDE. But even though there are many pros within this approach, like with broader adoption, responsible use, accessibility, practical applications, it's also important to note that there's lots of cons involved too with this approach. One is it can oversimplify something, and somebody who should not be building a model, they may be building something that's not relevant or actually go down the wrong path. Potential misuse, lack of depth, over-reliance on AI. So by no means I am suggesting that the no-code approach will make everyone an AI expert. Our main goal here is just have more AI champions so that they can bridge the gap between our clinical teams and our AI development teams, or let's say vendor AI platforms. The idea there is similar to what we've been doing in tumor boards, where we have more and more subspecialists who are there who are actually making a co-decision for the final patient's outcome. If we now have AI tools that are making part of those decisions a part of our triage, why don't we have any data science enhanced clinician or practitioner at that tumor board to also talk about the limitations of the AI tools, that's where hopefully things like this will address. So we're force multiplying it because of that and other reasons. So what else makes the curriculum unique? And it's things like our automatic, interactive applications. So one of them is, like, a lot of people ask us, how does ChatGPT work under the hood? And so instead of basically going and showing them a bunch of PowerPoint files, wouldn't it be better if you just showed it to them through an application, let them play with it? So you can show them that really a ChatGPT model is very similar to the traditional ML stuff that we have, except combined together. We're putting together unsupervised learning with supervised learning that enhances its capabilities and alignment, and reinforcement learning with human feedback in this case, and nowadays we even do with AI. And then so once you've basically aligned and you see how well the actual thing does, you can now also get the user to interactively see how the thing actually works. So you can open up the interactive part of it and ask it a question. And so I can ask it, what molecule carries oxygen? And hopefully this will come back and say hemoglobin, except as it's a ChatGPT type model. So this will initially tokenize your words, and for the sake of simplicity, I didn't split them into true tokens, it's just keep them as words. But then it assigns them numbers, what we call vector arrays, and then those numbers are then fed into the neural network, typically a transformer nowadays, that then goes through its layers and finally is able to make the final call in terms of what the final call for that particular statement was. And in this case here, it will come out and says the answer is hemoglobin, but as you can see that it's a long, basically, response. So you can imagine the tiniest word change within this could also change the entire context of this thing. So although it has some similarities to your classification that I showed you earlier, it's also very different in terms of its output. So that was a little glimpse of our AI interactive educational app. And so now I'm going to show you some quick little things in terms of where our computational pathology and AI Center of Excellence have come in to actually be able to drive many of these initiatives and efforts forward. Our vision and mission is pretty simple. We wanted to become a premier AI center within pathology and medicine. I think we're on the right track for that because lots of folks are trying to emulate this, and we're happy to share our knowledge within that space. And the goal is pretty simple. We want to advance the field of pathology and lab medicine. Now we're doing stuff outside of that as well. And so by doing that, we can help basically make established robust ethical regulatory practices and other standards that others can actually kind of follow. Some examples of CPACE-generated AI tools. There were lots here. Rob told me I can't show some of them, so I kept some of them out. But I will show you one of them. So we do have a lot of exciting stuff coming out of CPACE, and several of them are going through patent filings as we're speaking. So the big thing that most people ask us, are there any specific generative AI tools, like ChatGPT-like stuff, that will help with my various educational or operational missions? I think Greg will appreciate this. And the answer is yes. So exactly, our solution happened to be a pretty novel, but quite frank, not patentable to be objective, solution, which is, we call it Pitt GPT Plus. And what Pitt GPT Plus is an on-prem, that's a key thing, is that it's not talking to any cloud-based setup, so it's fully on-prem. And it's using multiple agents to cross-talk to each other. So that way, it basically enhances its final output better. So that's that if you're just comparing it with other models that are more capable. Because the agents are involved, it actually improves its output. And it follows a RAG-based framework. And I'm not going to go into details in terms of what part of frameworks, but I'm happy to answer those questions. So that stands for Retrieval Augmented Generation, which is basically what it basically means that if you want to talk to your own personal data and nothing else, this enables you to do it. So let me actually show it to you in action of how this works. Since somebody in another talk accused me that this was making an API call to either OpenAI or Azure or something else, I did this on purpose now. So I'm blocking it right there, showing you that there is no Wi-Fi call, just because this became a 10-minute discussion between myself and this person during a call. So no Wi-Fi connection whatsoever. So this is all running 100% on my own laptop. Okay, so how does this work? So I'm going to upload two very simple files. Nowadays we're uploading hundreds of them. And having it so that it becomes an in-context discussion with those things that are only relevant to me. So that then it only speaks to the files that I'm uploading. So I uploaded a file, I cover the leukemia lymphoma service. So I'm uploading some hematology and hempath relevant papers. So I'm uploading Fellini, and I uploaded Dan Arbor's paper on ICC and WHO myeloid neoplasms and lymphoid neoplasms. Now I'm going to ask it a question that it's not in those documents. So I'm a big soccer fan. So who are the best soccer players in the world? So if I ask it that, since it's not in those documents, it should not know it. But if you ask the ChatGPT-like thing, it will hallucinate for you. So this will basically say, well, I don't know, it's not in my knowledge base, and so can't give you an answer. Now, if I bring in the list of the best soccer players, so I say, here's the best soccer players, and now give me the answer, now it gives me the perfect answer. And it says the best answer, the best ones are Mbappe, Messi, so on and so forth, and let's not forget Houman Rashidi. But kidding aside, so obviously the type of data brought in makes a big difference in terms of what you get as an output. But I could also ask it specific questions around our particular discipline. So mine happened to be a lymphoma leukemia question. Because I'm asking the question of a CLL-SLL of an out-of-a-box Llama 3 model, it's not doing a great job. But if I now have fine-tuned the model to the Houman 22G and ask it that same question, it now is able to give me a much more confined and better answer that now I can feel confident about. So the type of model that we're also using matters a lot. And so why is our Pitt GPT Plus platform special? One is by doing this approach with a multi-agent framework, so you can basically drastically reduce hallucinations, so it's more accurate. In other words, its cost effectiveness is going up quite a bit because you're not making an API call. So, and I personally, as an administrator, I'm in the hybrid camp personally. I think for certain things, we should have great vendor partners that we do stuff with, and for certain things, we should have on-prem things that we develop in-house. And so it depends on how many users you have, quite frankly, how accurate the stuff are. So cost effectiveness is a major thing that we should consider. Scalability within the institution, enhanced control, and data governance. And then last one, which is a huge thing, especially nowadays when people are getting attacked in their institutions, data security. So having something that's on-prem, that's just local on your environment, and not cross-talking to something else, even if it's a very trustable vendor, I think a lot of our cybersecurity people feel more at ease when it's on-prem. So in addition to all of these interactive tools in AutoM, most importantly, our educational material that I've been showing you, they've come with scientifically validated and reliable references. So I'm not going to dig into a lot more details of this. This came out of 6 papers that was part of the Cleveland Clinic's course and 8 of them that was part of the Pitt's new course. So together, this will basically give the foundation of, if you will, a textbook, although they're all review articles, that would enable, your end user to be engaged, but in a reliable fashion moving forward. And if you're wondering what the new series is about, it's basically a very simple, and we've removed 99% of the math and heavy jargon out of it. It's more around, do you understand the basic language so that if you are talking to an AI developer, you can actually understand each other better. So it's around introduction of AI, generative AI, deeper dive, non-generative AI, and how that's different from generative AI, and then statistics of generative AI, and then of course regulatory aspects, ethical, and then machine learning operations and multi-agent framework aspects that we're dealing with nowadays. This was not, even though it was led by Pitt, I do have to credit that it was with 50 of our AI experts from across the country. So I do want to credit them, and they were from various institutions, and I apologize if I did not list some of them in there. And this is the list of it, so I will leave the talk here if people want to have them, but if you just Google it, you'll find it. So what else should we be aware of when it comes to AI? The various challenges, and this is what I'm going to finish with, and limitations in AI. Even though, there's lots of great things that AI can do for us, it could also have misinformation risk, so we have to be aware of that. Data privacy is a major concern. Critical thinking is a major one, especially within the education world, especially if you're talking about younger, learners. And then finally, equity concerns is one that you should be also aware of, so that it basically can be truly given to as many people as possible. Processes are very important to put together to minimize those issues, and we do put them in place, but just as equally important is to make sure that the human element is also present. And I do want to credit my friend Jason Rosenstock on this, where he put in a very nice mnemonic for us to use, which we are using, and it's in tune with Pitt's, how we approach, generative AI, which is the Disclose, Verify, Protect, or the DVP framework that he says. And what that stands for is honesty is best policy, so make sure you always disclose so that people know that the stuff that was created, that's why if you saw in the very beginning, I did say the stuff. And then verify the information, because we know a lot of these things can hallucinate. None of these things are ever perfect, so be aware of that. And then lastly, just as importantly, is protect the information and make sure that you're not putting copyrighted information or sensitive patient information or other types of sensitive information that's not allowed within an institution's privacy rules. In conclusion, AI is revolutionizing medical education and clinical practice, no doubt. No-code AI education is key to a broader adoption. We feel strongly about that. And the faculty and students must understand AI capabilities, but just as importantly, their limitations. Teaching the teachers is just as important as us teaching our students. I think that should be part of our strategy, this plan as well, while we're implementing, responsible AI usage policies in our institutions and continuing to explore AI's potential for educational and clinical environment excellence. So all of this is only made possible with all of my friends' partnerships. So many of you are in the hallway here, and many who are not, and collaborations and continued support. So I do thank you for coming to this talk, and I do want to thank our incredibly talented CPACE team, some of which are here, like Dr. Wong, Dr. Pantanowitz, who's my partner in crime in CPACE, and of course, Dr. Shaker, who this was his vision in terms of what he wanted, in terms of what we would do as Pitt as a whole, and all the numerous partners and collaborators they have. And I thank you for your attention and happy to take any questions. Thank you.
We have time for one question, but Dr. Billiard gets it.
Okay. Great.
That was wonderful.
Thanks.
So I'm sure you've given this question a little bit of thought.
Yeah.
Is there a rating scale in terms of gaining competencies from beginner to expert in the field of AI?
Yeah, I mean, personally, that's like kind of an unknown camp for me right now, because I think, you know, what we consider an expert, if I'm looking at it in our AI center, an AI expertise will be as subjective and relevant to if I'm talking about somebody who's a developer, or maybe our data scientist, that's what they've trained on. If I'm talking about in the no-code world, you know, are they more capable about, you know, assessment of statistics and, you know, how basically a study design should be evaluated properly. And so I think it's a very good question. I don't have a perfect answer in that, but we are developing standards in that stepwise fashion, which we're hoping heuristically it will kind of build itself out over time. And we're hoping that with certifications that hopefully lead to higher level certifications, let's say degrees, that basically will be, you know, kind of objectively panned out, and that we can, but I don't have one right now. But that's a good point. Well, thank you so much. Appreciate it.
Thanks, Human. Okay, it's my distinct pleasure to introduce Craig Mundy to you. Craig is the president of Mundy and Associates, and he'll be talking about artificial general intelligence and its impact on biology and medicine. Forward, backward, next slide.
Okay, thank you. Good afternoon. When I started to develop this talk, I wanted to talk about the long-term impact of artificial general intelligence on biology and medicine. And as I sat down to develop the talk and thought about how would I try to share with you some of the things that I think are going to happen, I actually ended up doing something similar to what Lee did this morning, which was go back a little bit in my own history with this question and try to work forward. In order to do that, and in the spirit of the last comments, I sat down with ChatGPT 03 and 4.0 and basically worked with it and talked with it about some of the things that I wanted to say. And so this presentation is a blend of my own thinking and things that I learned in conversation or interaction with that. One other thing that you'll see as you go through this slide is as I prepped and looked at the materials that were sent, I saw that sometimes people talked about P4 medicine and sometimes people talked about P5 medicine. I've known Lee since I think he started P4, but I hadn't really heard about P5. So I kept looking and saying, well, you know, is there a real definition of what was the fifth P? And I couldn't find one. So I started thinking, okay, well, I'll use my own fifth P then. And you'll see as I go through the talk, I added psychocognitive as a P. And that's because one of the things I have felt for a while is that we focus a lot on sort of the body itself, and we know the brain's in there, but we don't know that much about it really. And yet we have come to understand how important it is in our overall health and well-being. And so I decided in this talk, I'm going to speculate about that a little bit as well. The first talk I gave about artificial intelligence and its relationship to biology medicine was at Northwestern in 2017. And what I'm going to show you in this talk is each of the talks I gave that I'm going to cite, and there were 6 of them over the years, lasted as long as the one I'm going to give today. And the question is, how could I get a synopsis of that? So I just took the 6 PowerPoint decks and I sent it over to the AGI and I said, summarize each of the key things that happened in those talks, and when you're done, show me a timeline of what you think the key insights were that evolved. And I'm going to blast through this really quickly because I don't want to give the talks over and over again, but you know, as you go through this, you'll see that even back in 2017, I was forecasting that there would be a radical change in the architecture of computing, in particular for artificial intelligence, that the machines would come to have, you know, through these tremendous speed ups, that the machines would start to outperform humans. And despite Human's comments about, there's sort of 3 distinct phases, I don't really quite agree with that. I think there's a blend of all of these phases. In some areas, the machines are already super intelligent relative to humans, and in others, they're not quite there yet. But, and at that time, 2017, I actually took the P4 idea and I speculated about how artificial intelligence would affect it. In 2018, I went to the National Institute of Health and gave a workshop, and I think it was the first time where I posed the question, is human biology too complicated for humans? And I concluded that it was, but that it wasn't going to be too complicated for the machines. And that was 7 years ago. And I think we're starting to see that that was probably a good bet. I actually created a concept of a game of life where I thought, okay, if the machine knew what it was trying to optimize for, like we do in games. And as we saw, this was about the time when AlphaGo and AlphaGo Zero were done by DeepMind. I said, well, could it actually start to think about how to optimize for these things? And I advocated for data-driven discovery, notably suggesting that you should discard legacy assumptions. One of the things, I did a lot of work with SomaLogic and others in that same time period, and I started to realize that the humans didn't really have it right when it came to the understanding all of human biology. And in fact, more often, if you started with what the humans thought the answer was, it took a lot longer for the machine to get to the right answer. And so it became really imperative that you try to stage the question properly, but don't bias what you think the answer is too much. And I think now, 7 or 8 years later, we see that kind of thing happening too. In 2019, I gave a talk at the Cleveland Clinic at their annual healthcare advanced kind of thing. And this was at the time where OpenAI, where I was also involved and still am, had started for the first time to show their work in agents. And I showed a little video of agents trying to solve problems by themselves. And now today we hear people talking every day about agentic AI. At the time, I envisioned that the AI could, in fact, think about predicting the future state of your proteome. That if your proteome was sort of the best of the batch of things that define the, sort of the instantaneous state of your whole body, then if I knew what it was now and at some points in the future, could I, in fact, predict what I think the proteome would be? And interestingly, now a few years on, I harbor the idea in my brain that the way that the AIs produce images and now movies, which is a diffusion technique, is essentially starting with, kind of a blank slate and then filling it in until it looks like it matches the picture that you asked for. And it isn't clear to me that we couldn't perhaps borrow those, think of the proteome as a picture. And in fact, the idea for this came because with the help from ISB people, when my wife had cancer, we did the first ever high-scale proteomic assay of every step of her cancer. She was also one of the Pioneer 100. And so we could go back several years before we actually discovered that she had cancer to see what her proteome did. And what was interesting, the only way at the time, with the tools that we had at ISB, that we could make sense of the proteomic data was to use genetic tools to visualize it in self-organizing maps. There was no other way for humans to kind of gain any insight. And through that, we could see interesting things. But I said, okay, well, that picture represents my wife's state at every moment in time. Could I ask it to predict what that picture would be, and in essence predict, by some perturbation, what might happen in the future? In 2021, I went to Oxford and gave a talk about these things and said, hey, AGI is coming and it's going to change almost everything in human health, and used the example of AlphaFold at that point as an example of how this would happen. And of course, that went on now to get the Nobel Prize. We projected that computing would go 10x, a year, and in fact, that has been sustained as was predicted. And I formed a challenge to the people in the audience that says, look, basically, you now have to think of this machine as a polymathic prodigy. And the question for us is not just how do we use it, but how we raise it right. And I think that's increasingly an important comment. A year later, I went back to Oxford. This time I was talking to the physics people because they wanted to understand that. But one of the things that we could confirm at that point, looking at the trajectory we're on, was that at that moment in time, the computational capability we had looked like it could train a human equivalent brain capability in about 15 years of computing. But we recognized it was going 10x each time. And so you speculate, all right, well, if it's 15 years then, which you could argue is about how long it takes a human baby to grow up and kind of have, you know, a pretty complete cognitive capability. You say, all right, in 2022, if I trained it as long as I train a human, you know, they would be kind of equivalent. But we could see that while humans don't learn a lot faster than that, the machines have in fact advanced to the point where we are now training human equivalency capability in a matter of weeks or certainly months. And so it became more and more clear to me that the future was one where we needed in high-dimensional spaces to let the machine guide the humans and not the humans guide the machine. And that is a very difficult thing for humans to get their head wrapped around. But it's one of the biggest problems I think we're all going to face. Last year, I gave the keynote speech at the American Association of Neurological Surgeons. And I contended that AGI's impact on healthcare and longevity would be profound. I outlined the idea that we need a universal supervisory AI that would actually allow us to trust these machines one to the next and even on an international basis. And in fact, that's been my own personal focus from a policy and technology point of view for about the last 4 or 5 years. And I presented the concept, which was also shown in the book that Eric Schmidt and Henry and Kissinger and I wrote that came out last November, that in fact, these machines, increasingly, you'll have to just think of them as a new intelligent species. It just isn't biological the way we are. But it otherwise will exhibit all of the speciation issues or capabilities that humans do. And that creates a huge long-term issue for all of us. Anyway, the AI summarized those 6 talks and said, here were the milestones. You know, we predicted the exponential advances in 2017. We questioned whether humans, you know, were going to be superseded by this with respect to biology in 2018. We called for model-based regulatory science and N-of-1 therapies. And my belief then was that one of the biggest impediments to progress in the field is the fact that we have no regulatory science that is prepared to deal with this. And unless we bring AI to bear on the equivalency, I'll say, of what we have historically looked to statistical methods for validation, then it'll become the long pole in the tent. In 21, you know, we looked at the proteomic scale prediction capabilities, and I still think that that's interesting. And much of what Lee talked about today and you know, is moving in the direction where we really want to see that level of predictability. And so I think, and ultimately that in last year I predicted and are working hard on the idea that we'll have an AI that can supervise other AIs. So that led to 6 core predictions, most of which I've just summarized. And that leads me to say, well, okay, what is the state of the art now in P5 medicine? And so with the AI and search engines, I just did a little searching to find, okay, what does define this relative to each of the Ps today? Because in 2017 and these earlier speeches, I guarantee you that the people in the audience, most of them thought I was crazy. All right, they couldn't imagine that these things would happen, let alone happen by today. And yet, almost every one of these things, you can now see actual evidence, you know, proven by clinical trials and studies. And all of that has largely burst on the scene in the last couple years since ChatGPT lit everybody up. And so I'll just give you a few examples of that. I mean, we've heard a lot about, in the predictive area, AlphaFold 3 predicts protein, RNA ligands, et cetera, multimodal risk models, et cetera, from the UK Biobank data. You know, digital twins and early warning are actually emerging. I mean, there's a couple of them, the twin Twin AI thing that came out in 24, a new one from the NHS records coming this year. In the personalized area, interestingly, the first AI-generated completely in silico drug was done and went, from idea to clinic in under 30 months. This is the kind of acceleration we want, but the question is, how do we ultimately get past having to go to the clinic to decide that, that this thing is actually safe and efficacious? Amgen and NVIDIA got together to use NVIDIA's general physical modeling system, put some bio capabilities on top of it, and essentially are cutting the time between when you propose a drug and when it might actually be opted to take it forward by 70%. This is the beginning of a really important acceleration process that I'll talk about a little more in a minute. In the participatory area, in the UK now, the NHS has a named physio chatbot that basically gets 1 million people now every day get counseling on their back pain care through their phones. And Apple Vision Pro, their augmented reality headset, is now starting to be used in cognitive behavioral therapy. And we can now see, a lot of people in the neuro area have recognized, well, if I stick some wires in your brain and tweak them just the right way, I can get behavior change. I can even get the brain to reorganize itself, based on those signals. But it turns out you can do the same thing under software control using our own, exogenous senses, your sight, your sound. I think Lee talked about some of that, in the work that's been done in San Francisco. And then I added good old psychocognitive because I wanted to show that indeed people are already trying to address the brain and that the AI may give us unique capabilities to do that. And we can now see, in trials, reductions in anxiety and depression, where the coaching is either guided by or being done directly by artificial intelligence interactions. And the ability for the AI to detect in a conversation with a person a suicidal tendency is 10 times faster than a trained clinician can do. And so, its ability to see and find patterns that elude humans is the greatest gift of these things. The polymathic nature of the machine that we're building at scale, it will do things that humans will never be able to do. They can't do them one at a time, even if they're individual polymaths, and they will not be able to do them n at a time because the cost of communication between humans is just too high, and it goes up with the square of the number of people collaborating. And so, that's just a fundamental issue, and I think the greatest opportunity that these machines bring is this intrinsic ability to be polymathic at scale. I wanted to focus a little bit more specifically on the multi-omics issue because that's a very big topic for this meeting. But there have been breakthroughs just in the last 2 years in this multi-omics area. The frontier models are now being used to fuse all this genomic and proteomic, metabolomic, and phenomic kind of data. And to some extent, I'm going to show that in each of the P's of the P5 medicine, you already see live multi-omic exemplars in the clinic or in large trials. So this stuff is happening. And the question is, how do we accelerate it? So in the predictive area, AI has been used to link plasma proteins and genomes to disease subtypes, showing again that it can see patterns among this data that might elude a human analysis. In fact, when Lee and I first met and started talking 20-odd years ago, he was using classical bioinformatics techniques to analyze the big data cloud that was getting produced. And Nathan was there too doing this. But it was clear, even with all those things, mostly the best thing that popped out were new visualizations, the wagon wheels with the big swirling lines among them, to allow a human to look for patterns in these things that otherwise we didn't have, even with the biggest machines, good ways of finding. And now the AIs essentially look at this, if you will, through a completely different lens, and the rate of progress will be much, much faster. again, in the UK biobank, they've been used to now predict 150 different conditions years before they are clinically detectable. And so I think somebody asked Lee about Alzheimer's. He and I have been very involved in that of late. And it is, I think, completely obvious that we will be able to detect or predict, whether an individual will get Alzheimer's and over what period of time, and even how to prevent it, simply because we'll be able to understand these things very early in your life and to monitor it at a very low cost. And these, I think, will represent qualitative changes in each of these disease categories. In the prevention area, lots of blood tests and things to detect colorectal cancer. The medical digital twin work is showing really interesting results. And you can combine that with the wearables, already being combined with that. Lee mentioned that as a part of what was done back in the Aerovale and Pioneer 100 days, but it's clearly even more available these days. Personalized, we can now match 81% of tumors to targeted drugs computationally, using AI, where before we'd do organoid growths and other things, and trying to do the drugs, and these are obviously much more expensive techniques. So as we move along, participatory, people have now developed a combination and gone through randomized clinical trials of combining your microbiome, continuous glucose monitoring, and blood lipids to improve, to plan improvements in cardiometabolic markers. 500,000 users have received real-time omics information on diet through an app. And so these things are really moving quite quickly. In the psychocognitive area, there's immune transcriptomic multi-omic clustering that can uncover depression subtypes to give precision therapy. Integrative omics are poised to deliver molecular diagnostics for MDD. And so, I wasn't involved in any of these, but 7 or 8 years ago, there were none of these, all right? And what I'm trying to emphasize is it's important to understand, what's happening and how fast it's happening. And, I feel like I was pretty good in those 6 or 7 years worth of talks, predicting some of what was going to happen. And I'm going to use the last half of this talk to guess a little bit more about some of the things that might happen as the capabilities of these AIs get better. One of the things that I talked about, maybe it was the first time, was at the Cleveland Clinic talk in 2019, was this idea that we need sort of regulatory science 2.0, some model-based way of validating what we're doing. Lee didn't get to talk about it in depth, but when he was talking about the future drugs that he's talking about, one of the things that I'm intrigued about with that kind of work is the ability to design drugs that don't have side effects. Side effects come historically from having small molecules that bind to a particular spot, but you don't know that that spot is only in the target you want. And so the only way we find out where the other ones were was we go try it on a bunch of people and see what manifests. And that's slow and expensive, and I think is the thing that we ultimately have to get rid of. Obviously, if you want to get the N of 1 medicine, you can't test it on a bunch of other people because it was designed just for you. And so if you can't validate or design to eliminate side effects for safety purposes, then you can't practice N of 1 medicine. And so I think there's been inadequate emphasis in the community on how we're going to help the FDA get to the point where they're willing to accept that. When I was one of Obama's science advisors, Peggy Hamburg, who was the director of the FDA at the time, actually came to that advisory body, and actually she said to us, we need help. We see all these things coming down the path. We don't know what to do about them. And we need a new model of regulatory science. That was her words. It was the first time I'd heard it, but it has become more and more important. I get drawn regularly into trying to help young companies in the medical and drug field. And the thing I get incredibly frustrated by is people have these incredible capabilities and new ideas, and then you look at them and they keep trying to engineer how to use it in a way that will allow them to survive the duration and cost of a drug trial, which is 8 years and $1 billion or something. And that is the single biggest impediment. Only the big pharma companies, they've sort of, perhaps unintentionally, ended up in a position where they're the only people who can essentially fund things that have to wait that long without making any money. Venture capital doesn't have that kind of patience, by and large, and it's why all the little efforts historically get absorbed into the bigger efforts. But I think with the capabilities that are coming from artificial intelligence, small groups, as we've seen in so many other industries, might actually outperform the big groups because the AI more than gives them enough capability. The question is, they better be shooting at a different model of safety and efficacy determination, or otherwise, well, it'll all be stymied. So this is one of the things I've been pleading for 7 years, or 8 now, or maybe 6 years at least. And I can't emphasize enough its importance that people in these communities start to think that we have to come up with ways of getting this done. But there was sort of interesting news. December 2024, the FDA provided final guidance on predetermined change control plans for AI-enabled devices. This itself was a big step forward in my mind. It wasn't more than a few years ago that the FDA had decided that a cell phone was going to be considered a medical device, and that the FDA had to review every cell phone version by version, software change by software change, to tell you could run the app on the phone that was going to, listen to your heart rate. And of course that didn't work. I mean, that was a completely implausible idea, and they got past that. But there's still a long distance to go, but this is an indication that they recognize that they're going to have to do things differently, and that the AI will in fact not be a stable thing. Part of its significant capabilities in the future is that the distinction, I think, between training and inferencing will also become blurred. That more and more the AI, just as a human does, will gain capability merely by doing what it does every day. And that this idea that I train for a few months and then I put it on a different machine and I do inferencing against that trained model, I think is going to be changed. That also has a lot of implications for people that are thinking they're going to build and operate their own infrastructures and other things, because they may be chasing sort of the last generation of how we build these things and how we operate them, and thinking about what the capabilities have to be at each point in that process may change. This industry is incredibly dynamic. I was working with a guy on a problem in essentially signal processing, and we were using the AI not to do the signal processing, but to develop the research strategy to create a novel way of processing those signals. And in the course of like 8 or 9 days that we were doing that, and we were playing with 2 different models, each model changed significantly in that week and went from pretty good to really good that week. And it's hard to imagine the rate at which the progress is going to happen, but it is going to continue. And if anything, it's going to accelerate. And so I think we are at a point where there's now an open path, and I think maybe an open door at the FDA for people to bring ideas for how you can do that. So whether it's showing that you can use AI-based capability to design drugs that use, for example, multi-epitope binding or other things to become totally target-specific, and therefore say, look, we can show you computationally that these things are not going to have the kind of side effects that we would have had in the past. But bringing these things together in some integrated way, I think, is the challenge for the community. So what does all this mean for P5 healthcare with my fifth P? I think, predictive stuff you'll have to think is going to become capable of doing everything from population to atomic scale forecasting pretty much on demand. In prevention, continuous digital twins, which will anticipate disease and recommend lifestyle and drug tweaks that, every person won't just have their own phone or something. We may have, in fact, digital twins of people that just continually tracks you on a day-to-day basis. Personalized, I think AI-designed drugs and biologics for each molecular signature, should ultimately be a goal. Participatory, mixed reality, and chat agents, will coach patients in everyday life. So their ability to participate is not confined to when they go to the doctor or get online, but in fact, it's just part of living. And this psychocognitive component, I think will provide real-time mental health assessments that can be built into every interface. I remember more than 20 years ago at Microsoft, I was doing work in health, and so were my friends at Intel. And one of the things that we recognized was how in just, good old personal computers, we could put people down in front of a PC and have them play solitaire. And by doing incredibly fine-grained instrumentation of how long it took them to see a change in the card pattern, move the mouse, and do the click and drag it and let go of it, you could predict the state of mental decline of people way before there was any recognition of those changes by even the people they lived with, let alone at a clinical level. And so there's lots of data that shows that these kind of subtle changes in human behavior are good indicators. And so now we're going to start to see, I think in the next few years, that there'll be more and more ways of detecting these subtle changes in human physiology and mental capacity. So now I want to talk a bit about how AGI accelerates biomedicine. And so here I sort of collaborate a little bit with my AI and made some predictions for what I think will happen in the next 5 years. And I'll show in a minute why I think it's important to think of the next 5 years as really different than the 5 years after that. Assume that over this time period, we're going to see something on the order of a 10 to 20x performance improvement in the ability to develop a hypothesis, do in silico validation of it, and prototype it. So all the aspects will get faster. We'll start to see robots to work in labs that are specifically designed to be driven by an AI. And the AI will then be able to not only perform these functions, they'll do them tirelessly, 7/24. They won't make mistakes. And they can be trained very readily to do another one. It's not like a robot to build a car in a car factory over the last couple decades where it was a painstaking problem to get a bunch of super smart programmers to sit down and figure out how to write the code that would make the AI do exactly what you wanted when you wanted to. And so you don't have to do that anymore. These things can be taught easily. You can almost just talk to them and tell them what you want to do or show them yourself what you want to do. Their ability to see and talk and understand, is just an incredible advance in how we deal with the machines. And I think, we'll have to see, of necessity, the regulators will start to pivot to model-based evidence and real-world telemetry. And the more we can show that there's, computationally, that there's safety, the more you can say, well, the only thing to determine is efficacy. And if there's not a safety issue, then I can just put it out there. And if I can instrument everybody all the time and watch for all these subtle changes, I'll know a lot more than I ever would. And I can start to withdraw it, from the people that it's not going to service because we discover something else. But it's a very big inversion of the way that we think about producing these things now. I think we'll see live digital twins. Today, they're essentially a static copy, of something, and then we try to put something in and ask a question. So just like today, we train the models and then we do inferencing separately. And today we build the digital twins and then we operate them statically. I think that that'll change, and these digital twins will become a sort of a continuously functioning capability. And you can basically feed them not only clinical data, i.e. all the multi-omic stuff, but the lab streams and wearables that will continue to get more sophisticated. I think you can see the AGI monitor everything that's happening to you, both clinically and behaviorally, and recommend counter moves when it thinks you're doing something that's trending in a wrong direction. And that we'll see these certified sort of twin kernels pass a pre-market review, and after that there won't be a human trial required to validate whether you can use them for a new task or not. I think you'll see always-on molecular sentinels, the things that have been developed for environmental DNA monitoring, can be used, also some of this cell-free RNA work. The exolome, is actually your breath. It's been noted for a long time that dogs, can sniff out cancer in people. And, we can't do that. But it turns out the instrumentation at a molecular level is coming to be able to do that. So every day, you breathe into something, and maybe we learn things from that we wouldn't have otherwise known. You might find pathologic evolution in a matter of a day or two because you're monitoring everything continuously and you can detect the changes. I think nutraceuticals and behavioral adjustments to things can happen. Nathan may talk some more about this. I mean, he's been really big into the nutraceutical stuff lately, but some of the work that Lee and I've been involved in actually show that nutraceutical chemicals, can be used to compensate for things that are perhaps root cause issues of brain problems. It's just no one knows you're short on it, and it isn't something that you get enough, or you have a genetic predisposition, that says, your diet's fine. But it turns out you end up with the problem anyway. And then, we get to the point where we may do some clinical trials, but these sentinel networks, will allow you to basically declare that you have an intention to treat something, if it happens, as opposed to just wait until it has to be clinically diagnosed and then intervene. Personalized 6-month drug horizons, I think there's no reason other than the regulatory problems that we can't move rapidly toward these individual drugs. We can compute them and synthesize them maybe in as little as 3 days. And the question is, if you can do this, at that rate, the classic concepts of clinical trials, it just doesn't make any sense. Keep going here. Participatory, mixed reality coaches, as people wear, glasses and other things that have all of these capabilities in them. You just get more and more daily guidance. It's like getting your feed, when you wake up in the morning. If any of you have these, for example, the beds, that basically have sensors in them. You wake up in the morning, it sends you a text. Mine does now, and says, here's how your night went, and what you can do to make it better. Human feedback interfaces. One of the things that I think will happen is we'll see, just as we saw with computing itself, back in the day, you used to just type text messages, to your computer, command lines, and then we came up with graphical interfaces, and then pretty much everybody standardized on the Windows, model, whether it was Macs or PCs, and then you got the phones, and the phones converged on a particular one. So whether you're in the Apple camp or the Google camp, people still basically understand how these things work. And I think we're going to start to see, as it relates to allowing people to interact with these health maintenance capabilities, that there'll be some standardization that'll emerge mostly driven by products. The products that people like the most, they will be standardized. And I think we're going to start to see closed loop mind care, that we'll be able to track what what people's mental state is, whether it's depression or some other kind of disease. And this last bullet here, software as a therapy, there are already, I know personally, instances that have been cleared that says yes, you can keep updating this software in a way to have it adapt to individuals, and every person's adaptation, which is beneficial to them, is not a new product. It's not a new drug. It's not a new therapeutic approach. Also, this idea the AGI can design stimulation waveforms. As people put more and more energy into brain-computer interfaces, and you can combine those with exogenous stimulus, then an AGI can actually figure out, okay, for people with varying degrees of mental illnesses, what would we do? How do we stimulate it? How do we force the brain, to use its plasticity and do reorganization? And I think that's going to have to lead to this regulatory fast lane where there'll be more and more model-based validation. That'll probably result in guidelines coming out for what does it mean to have a good experimental practice, and how do we move these things so that we can, for example, take the live digital twins and use that to essentially, say these digital twins are considered accurate enough that when they're aggregated together, they provide the signal that you want the AGI to use to say you validated that approach. So the last 5 years, briefly, by 2030, there'll be a 10 to 20 times step function change in the rate of discovery and engineering as we cross this threshold to essentially superintelligence. And so I talked with my AI and said, okay, now we're getting, it's hard to imagine these things, but one way to think about what does it mean to have a 10 or 20x step function change in discovery and engineering capability. Dario Amodei, who was the founder of Anthropic, last October wrote a paper where he made such a prediction, which I tend to agree with. He also said for people, how should you think about what that means? He said, all right, here's one way to think about it. Think of all the capability humanity has as you know and understand it today, and dream big about all the things you hope we can do in the next 100 years, or by the end of the century. He said, if this happens on this time scale, basically by 2025, pretty much everything that humanity would have achieved by the end of the century will be done by 2035. That's a 20x kicker. And so things that really sound like science fiction are only a decade away. And so you have to really start thinking about the effects of that kind of acceleration. And when you make plans like the ones that are being done here at Pitt, it really becomes imperative that you think hard about that. So maybe here's some ideas that we came up with for what might happen with that kind of acceleration in that time period from 2030 to 2035. The AGI design, repairs for organs and regeneration. We'll bioprint tissues in general. We'll have closed-loop CRISPR editors. Regulatory fast-track, will become commonplace, and maybe we'll have compassionate implants. Population-scale preventative genomics, universal newborn sequencing, will probably become commonplace. Dynamic consent, where people can essentially say they'll either sell their data or put their data forward because they'll be given a benefit, like as your teenage kid. If you let the thing track their speed in driving, you get a reduction in your insurance premium, and we may do that for everybody. If the more data you'll share, the lower your costs will be. We'll probably have some genomic or more broadly AGI guardrails, that say, where are the limits on any given day to what the AI can do directly in terms of AI-mediated counseling. Universal digital care, sub $50, chips at the edge will probably be available. So instead of just having your sensors at the edge, you may have things that can do interpretation at the edge in real time. We'll stream all this stuff together. They create an early warning system. Maybe the World Health Organization endorses some open AGI model to use as a pandemic sentinel. Continuous licensing via AI sandboxes. We'll have virtual clinical environments that run perpetual adaptive trials with synthetic avatars. Regulators accept dynamic safety envelopes monitored by a supervisory AI. So themes that came, from my conversation with the AI about this 30 to 35 time period are just listed on the slide for your amusement. AGI optimized epigenome rejuvenation, home-scale omics labs and personal biobanks, adaptive vaccine platforms that make a new vaccine in under 24 hours, quantum and AI, real-time drug safety simulators. Turns out I'm also very involved in quantum, and what most people don't understand is that there's an incredible symbiosis between artificial intelligence and quantum computing. Both of them are only accelerator architectures and still have to be hooked up to a big machine. But they used to be thought to be kind of addressing independent problems. But now we know that if we're trying to synthesize new materials or new chemical processes, that the quantum computer basically can generate the data necessary to teach the AI how to forecast in that super high dimensional space. Neurosymbiotic wearables for continuous mind care and global AGI governance will come from a mesh of AIs that essentially can watch over the whole world and all of the AIs that have been deployed. So I think our vision for 2035 should be that health span extension becomes the default goal of healthcare, not just fixing people when they're broken. That AGI mentors co-evolve with each individual's biology and psyche across their lifetime. And that it's essentially infinite loop learning. The AIs at scale can learn everything all the time. Humans cannot do that. And therefore, we look down the road at a promise of every intervention, every action, everything we do basically just produces more learning. So this AGI-driven scenario is already happening. It'll only accelerate from here. I urge you to think deeply about the implications. Thanks for your attention.
All right, thanks, Craig. So for our final talk, Dr. Nathan Price will be speaking. He's professor and co-director of the Center for Human Health Span and the Buck Institute for Research on Aging. Nathan?
So it's a pleasure to be with everyone today who's stayed here till the very end, and also a pleasure to be able to go following Craig, who's gave us this really terrific talk on the future of AI, I guess past, present, and future a little bit, as well as hearing about the remarkable efforts that are happening here at Pitt in terms of trying to get AI much more into medical education and practice. So I think this is really an exciting time. As was mentioned, I'm a professor at the Buck Institute. Actually wear two hats at the moment. I'm also chief science officer for a company called Thorne, which works with about 5 million patients and about 50,000 medical providers. And so we doing nutraceuticals, testing, and AI systems for trying to interpret data. So I'll jump into this. So obviously we are at this really incredible moment in history right now, and I don't need to belabor this given that we've just heard such a beautiful talk about this. But we are at very much this inflection point, the birth of AI, and we're seeing the emergence of capabilities, and very much like what Craig was saying, this is what I really challenge my lab and all of us to do all the time, which is try to think about what is it that was maybe hard for us to do that's now super easy, and what's going to be easy next year that's hard for us now, because I think that the challenge before us is really to accelerate the pace on which we're making progress. And I think a lot of this will come from the AIs, but it's really a function of trying to set up what are the kinds of problems that we can turn into something that the AI can actually get its mind wrapped around and actually start to advance in a major way. And there's a lot of difficulties and challenges, obviously, before us. The regulatory space is a key one. I'll comment on that a little bit later. But the challenge is really to harness all that right kind of data that we need with the right kind of strategies, and we'll get into that more. But AI is promising an era of an unprecedented abundance, and I think we're all in agreement, or at least we would probably are if we're here in this room, that the abundance of health and the ability for us to take these AI and give us as much healthy life years as possible, probably the greatest application of AI to humanity that we could make. And just sharing a piece that Lee and I had written for the Wall Street Journal a little while ago about AI and the transformation of medicine. So I come at this also thinking about aging, as I'm a professor at the Buck Institute, as was mentioned. And so one of the ways that we think about aging just comes from this kind of a curve, which is the increase year over year in the likelihood that we will die from many different things, right? And these are all tied together, and there's fundamental biology of aging that plays a role in making that more and more likely. I was at a really good talk recently, and they were making the point that as you go through their life, there's one thing that you really don't want to have happen. It's incredibly dangerous, and you need to avoid it at all costs. And that is that you should never turn 50, because after that, it goes badly. And this is worrisome to me. I turned 50 this year, and so I'm just about to start the ramp up here. So we're of course trying to solve that at the moment. But anyway, so this is a really fundamental problem that we can address by leveraging AI into these systems. And of course, what we're really trying to do when we think about this is to extend health span. So as was mentioned, along with Lee, I co-direct the Center for Human Health Span at the Buck, and you really want to extend life as much as possible. And I was going to give a talk on scientific wellness. I was just driving up there through rural New York, going to, in upstate New York, going to RPI, and I was catching up on some reading. I try to read the latest. I'm slightly behind. This is a book that was written 2,000 years ago by one of the Stoics, by Seneca the Younger, and it's called The Moral Epistles. And as I was going through, the next chapter up just happened to be longevity. I was like, okay, what do they think about longevity 2,000 years ago? And what he said, which really struck me because it's such a perfect encapsulation of what we're trying to achieve, is for it makes a great deal of difference whether a man is lengthening his life or his death. And I thought that was just such a great encapsulation of what we're trying to do, which is to extend health span rather than extend that terrible period at the end of life. And it's interesting because when you worked on longevity, I was surprised by this, the number of times that people will say to you, oh, I don't want that. I don't want that. I just hope, beyond all things, that you will not succeed in your life effort to achieve this. That's a little exaggeration. But they'll say this, right? And I think it's because they have this image in their head, right, when you talk to them, of that extension of life as being a terrible period. And this is dealt with very beautifully in a fabulous book by Atul Gawande that I imagine many of you have read, called Being Mortal. One of the great book titles of all time, I would say. It could have been called On the End of Life in Large-Scale Healthcare Systems in Developed Countries, or something like this, and instead it's called Being Mortal, which is, of course, so much more poignant. And so it is really about that, though. How do we leverage these tools and move away and move towards areas where we can extend life rather than death? And so the way I come into this, much like Lee, who spoke, and Lee and I have worked together now for 20 years, so it's been a while, on and off in different ways and in different capacities. But basically, one of the things that we have done together over the last 12 years or so is this area that Lee talked about this morning, called scientific wellness. And Lee and I wrote a book on this, which was a bestseller for Harvard Press in '23. But basically, what this is an argument that rather than wait until we get into late-stage disease and trying to do something when it's maybe too far gone, the problems are maybe not tractable, or at least not as tractable, how do we, in fact, push that in a way that we can intervene much earlier and also not only intervene earlier, but be armed with much, much more data. And of course, now that we're in the era of AI, the ability that we have now to harness such data is radically improved from where it was, at the beginning of this. Lee and I had co-founded this company, Aravel, that he had mentioned a little bit this morning. And we kind of commiserate about this sometimes, because if we could have kept that going, now maybe we'd have 500,000 of these data clouds over 10 to 12 years. At the birth of AI would have been insanely useful at this period of time. Unfortunately, it went out of business, so we got part the way there, but not as fully. So excited for Adil, these new groups that are getting started, as well as we try to amplify million-person projects across the world. Adil, I've also known for many years, I think since we came out of the same era, so I think 20 years probably as well, 15 to 20, something like that, a long time. So when we started this, 12 years ago, with the Pioneer 100 project, Craig was one of the pioneers there with us as well. And then on to Aravel, we were able to get data on about 5,000 people over about 4 years. And this was data of various different types. So this was whole genome sequencing, measuring about 1,200 different analytes out of the blood 3 times during the year. Gut microbiome, again, 3 times during the year. Wearable devices, not as extensive as what we have now, but we're able to monitor this. This gave a database of actionable possibilities. And the idea was that if we could provide valuable health information, you can have people come with you, that you would be able to create these personal, dense, dynamic data clouds, get a lot of information, and then be able to monitor for health in those ways. And so I wanted to share a little bit of the science here, just to show some of the things that we were able to pull out with these earlier tools, just to make it concrete. And then I'm going to come back and talk to what we can do now with the AI tools that are at our disposal now. So I'll just give a few things that we learned from this, from the microbiome and metabolism. So one thing we found was that starting around age 50, your microbiome starts looking less and less like anyone else's microbiome. So they diverge away. We created this score called uniqueness. And in essence, it is just what I said there. And what was really fascinating was that when we looked at this, we were able to show that this uniqueness score, how tailored, I guess, your microbiome had become to you over time, was predictive of things like all-cause survival. So we looked in a group of, in this case, men in their 80s, and asked the question, who would live for 4 more years? And it turned out that the microbiome uniqueness score was predictive of that. A diversity of the microbiome actually was not predictive, but uniqueness again predicted things like fast walking speed in old age, excellent health, composite healthy, things of this nature. And that was validated in multiple cohorts across, at the end, nearly 10,000 people. Another thing that was interesting, though, is that even though your microbiome was diverging and looking very different from other people's, or at least more and more dissimilar, the metabolic transformations that would occur were conserved. And so there's a number of things that you can do to monitor for what's happening in your microbiome that appear in metabolites, for example, in your gut. So one of the things we saw was that there's a number of molecules that give added health, or at least are health beneficial, like indoles or p-crossyl sulfate, as well as negative ones like TMAO. TMAO is actually an interesting case as well, and I'll relate, coming back to nutraceuticals or nutrients or compounds, one compound that seems to be valuable for brain health potentially is a compound called phosphatidylcholine. And we also proved this out in these digital twin models that we funded and worked on with a collaborator for 3 years at Thorne. But basically, one of the things that you can do with that is you can have people that eat diets rich in phosphatidylcholine, for example, get Alzheimer's about 3 years later than those that don't. But the point I want to make here is that if you're eating phosphatidylcholine, for example, if you have certain kinds of bacteria in your gut, it will eat phosphatidylcholine, turns it into trimethylamine, which your liver turns into TMAO, which is a risk factor for cardiovascular disease. So as we think about personalization and the deployment of digital twins and all these monitoring systems, that's the kind of thing that you'd like to know that, hey, am I trying to do something that might be helpful to my liver or my brain, and I'm instead poisoning my heart? And I give that as an example because it requires a lot of detail to know whether that's true or not. And there are, of course, thousands, millions of such interactions happening on this. The metabolome does carry a ton of information that's relevant to your microbiome. And we actually saw that the diversity of your microbiome, you can capture about half of the variance in terms of predicting that from metabolites in your blood. And you can do that from as few as 11 metabolites. There are about 40 that carry a lot of signal. And when we looked at that across even a lot of different diseases, different categories, what we saw was that this predictive power held true across things like abdominal pain, antibiotics, constipation, diarrhea, type 2 diabetes, et cetera. And it only failed in one case. And this case was in extreme obesity. So what the World Health Organization would classify as type 2 or 3 obesity. It's above a BMI of about 35. And what we saw there was that signals that would carry about half of the predictive variance for the diversity of your microbiome actually went to 0. And so when you get into that kind of, we think maybe driven by high inflammation, but that sort of state, this relationship between the metabolites circulating in your blood and the microbiome gets totally destroyed. And we saw there no ability to predict that. And I should mention, this is, if you want to see the details on things I'm flying through here, this is published in Nature Biotechnology in 2019. The previous story on aging microbiome is in Nature Metabolism in 2021. We also looked at effects on weight loss. So we had people going through a wellness program and just saw what was predictive at baseline. So the day they walk in, right, we make some measurements. Was anything predictive at that point of who would go on to lose weight versus not? And it turned out that when we tried to do, once we controlled for BMI, because BMI itself is a predictor, metabolites, to my surprise, we saw no signal in the metabolome outside of BMI that was predictive. We saw no signal in the proteome that was predictive. We asked people what they ate. That was not predictive. It was trending like maybe if we asked more, but this is pretty well powered. We had 3,500 people that went through this. The weight loss is a subfraction of that. But the only thing that really predicted at baseline whether or not people would go on to lose weight was the microbiome. And it turned out there were two features of the microbiome that mattered. One was how fast the microbiome was growing. One explanation might be every calorie that you eat is a calorie either for you or your microbiome. So if your microbiome's churning along, maybe that's a little bit better for you. But the biggest effect was the gene content of the microbiome. And what that was is by analyzing the genes, you could say whether or not a person's microbiome was more likely to take, let's say you ingest a complex carbohydrate, like you eat a sweet potato. For some people, their microbiome will metabolize that more into simple sugars, and for other people, it will metabolize it more into short-chain fatty acids. So if it makes more simple sugars, those people, by and large, were not able to lose weight during the program, or they didn't lose weight during the program. And those people that would go to short-chain fatty acids had a much easier time. And the correlation for that was 0.3. So it was very significant, not totally predictive, but a significant effect. And so one of the interesting things of that, which we did not know at the time, it is not mentioned in the M Systems paper here, is that butyrate, which is one of those short-chain fatty acids, actually stimulates the release of GLP-1, which of course everybody knows now. So, but interesting, that was the only thing that was predictive. Speaking of BMI, this was also another paper done with Lee and with Noah Rappaport, who's a senior scientist who worked with both of us. Basically, this is a biological BMI. So this was a measure taking a very simple, BMI score that we're all familiar with, and instead asking about metabolic health that you could pull out of the data clouds. And what we showed is that you could generate this, biological BMI, and that was more predictive of health outcomes. And so you can dive in and understand that in some detail. And then bringing this back to the, and that's in Nature Medicine in 2023. And then bringing this back into the microbiome, we actually showed that the microbiome itself was more related to biological BMI than it was to BMI itself. And so again, it was emphasizing that deep connection that exists between all these metabolites in your blood and the microbiome. And the microbiome actually co-synthesizes about 18% of the metabolites that circulate in your blood. Final story that I'll share on the microbiome is that it's also predictive about the effects of drugs. So for example, we just clustered the microbiome into 4 sets. This was done with Sean Gibbons at the Institute for Systems Biology, with Tom Womansky, one fabulous postdoc, now working for me at Thorne. But he was lead author on this for Cell Press for Med. And basically, what it showed was that for one of these types of microbiomes, we saw double the lowering of LDL cholesterol by a statin than we did in the other groups, which is way bigger than any known genetic effect. And the other thing that's important to know about statins is that you get a 9% increase in the rate of diabetes among statin users. And it turned out that when we looked at this in the microbiome again, what we saw was that for 2 of the microbiome groups, we saw significant increases in diabetes markers, and in the other 2, we saw none. And so again, it's suggestive that the microbiome likely plays an important role in the efficacy of statins and likely other drugs. There was another paper in Cell a few years ago, not ours, that showed that first estimate was about 13% of drugs are metabolized away by the microbiome. These are things that are largely ignored by pharma right now, but needs to be a lot more. I think healthcare systems, as you start understanding more the role of microbiomes, are going to have to put pressure on this, because you could be on a drug and you're literally not even on the drug you think you're on because the microbiome is metabolizing it. And I'll just say the strength of this result we saw in statins, I just want to make the point that this is not cherry-picked from 100 drugs that we've run. We've only run 1. We only had enough statistical power to run the 1. So it's the first one we looked at. So it had a lot of effects. Okay. Okay, then I'll just say a few words about biological age. I'll be quick here. Lee mentioned this a little bit as well. So biological age is basically just the idea of, if we saw your data cloud, how old would we think you are, based on an analysis, right? So the age your body says, as opposed to the age the calendar says that you are. It's worth noting that there are many ways to do biological age, and they're not highly correlated with each other. If you do a straight-up correlation, they're about 0.8 correlated, which sounds really good, but that's because they're correlated with age. If you correlate residuals, they're about 0.25 correlated. I didn't put that slide in there, but we've looked at this on the proteome, the metabolome, clinical labs, so forth. They're about 0.25 correlated. But I'm just going to show you one set that we did here. We do this using something called the Clamara-Duval algorithm, which is an algorithm I really favor for biological age calculation, because it has one really important aspect to it, which is that you do a transformation, basically a principal component transformation, so that you get orthogonal vectors, kind of the usual thing. But when you can do the transformation into the Clamara-Duval algorithm, and what it enforces is that it makes the slope of the line that you fit to be very close to 1. The reason that matters is that it gives you the expectation that year over year, your population will tick up by 1 year per year. A lot of scores don't do this, and they tend to predict everyone young, old on average, and they predict everyone old, young on average. I will tell you from the commercial standpoint, that actually is great, because people if you want to be commercially successful, just tell everyone they're younger and everyone is super happy. But this enforces that it's a 50-50. It actually enforces that in the population, it has a slope of 1. Okay, and I mentioned that already. Oh, I did show the data. I thought I left this out. Anyway, that's the data that we enforce as being near 1. So, but of course, when you're doing a prediction, you're going to get a prediction high, a prediction low. That will always happen. So the question is that meaningful in some way, right? Do we care? And so we ran 2 tests on this. So the one here on the left is we had a whole bunch of different disease conditions. This is on 3,500 people. And we just asked the question of, did people have higher or lower biological age when they associated with disease. And you can see the statistical significance is everything that's starred there. And the point I want to make is that in every single case where there was a statistically significant signal, it was in the direction of higher biological age associated with disease. And we saw no cases that went the opposite direction. So you'll notice there are no stars on the left hand of any of those calculations. So that to me was a very strong signal that, okay, the biological age was measuring something that was related to health because it was very strongly associated with disease. Now, more interestingly, at least from my standpoint, was what happened to individuals going through the wellness program. And so that blue line there on the far left, that indicates expectation, right? Because I took you through the math, or I showed you the plot, that the algorithm enforces that as an expectation. And so what we saw was that overall in the Aravel program, that on average, there was a slight decrease in biological age estimate of 0.16 years per year, which meant that the population on average was getting about a year lower than their chronological age year over year. Women did particularly well. They were improving by 1.5 years per year, and men were doing not as well. They were improving by 0.8 years per year, meaning that they were getting a 1 year increase in biological age on average per 5 years. Now, I'll point out the obvious. This is not the announcement of immortality or agelessness or anything like this. The further down you get below your biological age, the harder that is to change. And so it does, there's kind of limitations to how much you can do this, but you can have that effect. I will say, much to my surprise, I was actually a pretty big outlier on this. The 4 years I was in Aravel, which is a big outlier amongst the men, my biological age went down 10 years in 4. So it went down 2.5 years per year in the program, which was actually nice to see. Okay, at least according to that metric. All right, so now I want to talk a little bit about genetics and looking early. So one of the things that's really interesting that we can look at is the role that genetics plays in the interpretation of clinical lab data. So I'll come back to a familiar example, LDL and statins. For what it's worth, there are better markers for cardiovascular health than LDL, but this is the example we use for this point. So one of the things I want to showcase here, though, is just that you can predict a fair amount about a person's LDL level from the genome alone. Okay, and so that's what's shown here, is just that if we bucket people we just bucketed them into 5 groups based on their genetic risk for LDL, that's the x-axis, and the y-axis is the actual value in their blood. And you capture in this about 12% of the variance here on the R squared. So that's enough to move you from below clinical range to above clinical range. So it's meaningful. I will also say that this is from a few years ago. We can actually now predict about double this amount from the genome. So the genome predictors of this have gotten a lot better, and that's true across the board. But when we did this study, this is where it was at. So the question is, if someone comes into the doctor's office and they have high LDL cholesterol, does it matter if they're there because their genome predicted them high or not? Because in medicine today, nobody uses the genome. I know we're trying to change that here, which is exciting, but to a first approximation, no one uses this. And so we just looked to see what happened when people went through a lifestyle program. And so what we saw here then was as people were going through a health program, did we see their LDL cholesterol lower? And what we saw was very striking. So on the left there, what you'll see is that people whose genome predicted that they would have low LDL cholesterol saw very significant drops in LDL cholesterol in a lifestyle program. Where people on the right that were predicted high by their genome, we saw no statistical difference in their LDL cholesterol levels compared to by going through the program. So the point I want to make here is that this introduces an entirely new category of variable into medicine that is unused today. And you can apply, and I just showed it for LDL, but it's true for HDL cholesterol, it's true for hemoglobin A1C. You can do this kind of across the board. And that is that the gap between the genetic prediction of the marker and the actual measure of the marker tells you how easy it is for that to move via lifestyle. It could be lifestyle, or I think it's likely true for nutraceuticals. But do you have to hammer this thing with a drug or not? And that's one of the questions. Now, one of the other questions that arises is whether or not a high LDL cholesterol in someone whose genome predicts them high is in fact equally worrisome. So I'm not really sure on that for LDL because it has its mechanistic roles. But one of the ones that seems a little clearer is hemoglobin A1C, for example. So some people, based on genetics, have longer residence time of red blood cells, right? Red blood cells circulate in your body over a course of about 120 days. Some people it's more like 130, some people it's more like 110. There's variability there in the genetics. So if you have genetics where your residence time, the amount of time you have for all those little glycosylations to happen on hemoglobin A1C, if that's longer, a higher level of hemoglobin A1C is less likely to transfer to diabetes. Whereas if you're on the low side and you have the same level, more likely. Anyway, all kinds of things you can pull out of data. You can also look for early warning signs for cancers. This is I'll give a few simple examples. These are just people that were going through the program. So small n here. But one of these markers that's interesting is CEACAM5. It's a known metastasis marker, but we were curious to see that it popped up in the wellness population that we were monitoring. And so here, we had a woman, age 57. So she comes in, she has this very elevated CEACAM5. 6 months later, we measure it again, still elevated. 6 months later, it's there. 6 months later, still there. She then ends up getting diagnosed with pancreatic cancer, and unfortunately she dies shortly thereafter. I will mention, if you're wondering, we were not allowed to monitor for these within the commercial program, so these are bank, this was bank samples that we looked at, afterwards. We could talk about that from a regulatory perspective too. Anyway, here's a man, age 69, somewhat elevated here, starts to pop up again, starts to pop up, metastatic lung cancer. Caucasian female, 57. This one was, I think, the most interesting. Starts out super low, then it transitions here, and it jumps up. Now this in and of itself is probably, if you're monitoring longitudinally, is already a marker, most likely, because we've looked at the likelihood of a jump of that much is actually very unlikely. So even though it's in the normal range, right, that delta is unlikely, and you'll see what happens to her the next time point. If you see way over there on the right, the highest we'd ever measured, and as you no doubt expect, she got this metastatic cancer. And if we zoom out here, what you'll see is that that's only 3 cases. They were the only 3 people that got transitioned. But the point I'll make is that one on the bottom, though, there's a lot of cases that we did not see it. And so it's an interesting, it's not provable. There's much more you have to do for a screening marker, obviously. But we know CEACAM5 is metastatic. We see it elevated in all, in the 3 people, and it looks like it may be much more useful as an early marker than maybe we had thought. So I think that's definitely worth checking out. Okay, so now I just want to chat a little bit more about the future. So I just wanted to take you through some concrete examples, papers, just to give you a sense of what you can pull out of even a small amount of data with more conventional computational biology approaches. And so I just want to talk a little bit about kind of where we're going and what I think the future of AI holds for us. So in my lab, there's kind of 3 big areas that we're working on. Scientific wellness and deep phenotyping, which I just talked about. And this is really getting at the substrates for AI. You've got to have a lot of this deep multi-omic data, and it's more than that, of course, as well. You've got to have wearables, you've got to get data passively, because I think to get to some of the kind of things like what Craig was talking about and others today, the amount of data that we're going to need is massive. The amount of compute that's going to be required for every single person is huge. And so there's big need for us to generate the kind of things that we're talking about here today, the 1 million person project that Adil's working on, that Lee's working on. talking with a number of groups, around the world as well. We're really trying to get as much of this as possible. Digital twins and the N of 1 analyzer is a huge thing that we're working on. And since Craig had brought this up, with his wife and cancer, honestly, this was a lot of the inspiration for this, because when we were working through that some years ago, what became obvious, as Craig alluded to, is that we do not have really good tools for analysis of the one person. And so Lee mentioned that we've gotten, Lee is the PI of the whole thing, of this ARPA-H grant for up to $52 million award we got from them. My part of that is to generate what we call the N of 1 analyzer. And so because of the scale of data we need, and because of the individualized nature of people, it's very unlikely that we can brute force statistics, using statistical models to where we get to go to. In fact, I think I'm pretty sure that's provably impossible. So we could get to that. So what do we have to do is something very different. And so what we have to do is to apply knowledge, deep reasoning, and these AI systems so that we can actually analyze what's happening in the single individual. Craig and I have talked about this for many years, but I feel like, and we were working on this actually a little bit before, but it was very hand-wavy, I would say. But now that we have like LLMs, generative AI, like the path that you can see to do this is, there's a lot to work out, but it's pretty clear. Like you can leverage all of the known biomedical research. So we're working on something that I'm calling, at least for now, context-aware data analysis. And so the idea here is that, for what we've done in the past, it's typically just been like a big data matrix. But once you're in the matrix and you're just analyzing, it's just numbers, right? And the algorithm doesn't know anything more about it. That no longer needs to be true, because the algorithm can understand that when it's analyzing this piece of data, it's actually a protein that's made primarily in the liver, that's associated with these 5 diseases, that has these side effects, that's on the surface of the membrane, et cetera, et cetera, et cetera. So there's a lot of, so in my mind, and I'm talking a lot about this in my group, is I think we're gonna view so much of all the things that we've done over the last, the last decades in analysis as being incredibly shallow. And we're actually going from being shallow to being deep. And we don't yet totally know how to do that, but there are some elements to do it. In fact, I wanted to put in some slides on this today, although I decided not to, because we're kind of in the middle of this. So we've done some analysis, and what we have as an output for one of these things is a 120-page paper written by ChatGPT in various iterations, right? And we've built some RAGs and knowledge graphs and stuff around it and so forth. But it's a 120-page analysis written about everything that we know about all the things that we saw, that came out of that. That's a lot to digest, right? So there's many things that we have to dive in to understand this. But the N of 1 is absolutely essential for us to get at the challenge of truly personalized medicine. From the regulatory standpoint, and we looked at this a lot with some digital twins we had built in other contexts with a partner, but basically, and I've presented this at part of the advisory count to the NIH director. I've presented this at MIT, at Johns Hopkins. I've presented this on the board on life sciences, the national academies, where I'm a member of the board, and not of the academies. But it's like, and when we've gotten into this, basically what you can do is you can actually run a standard clinical trial for N of 1 analyses, and you can do it in a very robust way. And the way that has to happen is you have to build the algorithm. So you can test a single algorithm, and it can be complex, and it can be, it can encompass basically everything that you think you know about a disease or a health state. And so then you take every individual measurement, then you analyze that fact pattern, and you can give a unique therapy combination to every single person. And so what you're testing is not the combination. You could never actually run that on the individual, other than potentially if you get the AI systems to be good enough that you believe them mechanistically downstream. Craig alluded to that in his talk. but you can do it right now by looking at populations that are analyzed by these kind of algorithms. And I'm a big believer in this, also from the standpoint that in prevention trials, I think we have thrown away lots of valuable things for prevention. Particularly, I would argue in some of the lifestyle and nutraceuticals and things like this, because we force it to try to be on one compound at a time. And there's just no way that one of these things is like powerful enough on its own to like change the clinical outcome. But what we saw in the digital twin simulations is that when you start doing combinations or you start like really taking a personalized approach, that the predicted efficacy goes from like 1% to like 80%. Your expected response rate jumps very significantly, and I think that's going to be hugely important. And then the last thing I'll mention here are these virtual systems biologists. And this is the notion that you're really trying to build these agents for the analysis of data in such a way that you can analyze and understand. You're basically trying to represent how would a human or a team of expert humans think through this data if they had the incredible computation and memory and speed of a computer, and trying to really represent that. And that's really the essence of what we're trying to do. So building out these systems research agents, which is a major focus of what we're doing. And then we've kind of started breaking this down into and I'll use Daniel Kahneman's language here, thinking fast and slow, which is that there's these, what we're calling the fast algorithms, which are these, computational quick hits of analyzing data, a lot along the lines of what I showed in all those different examples. And then the slow is really mining all of the known literature, thinking about hypothesizing, like what's going on with that, and having this like flip back and forth, very much like we do in our own minds, but being able to scale that up and amplify it in ways that we just couldn't get ourselves wrapped around. So with that, I'll wrap up. AI in health represents a transformative opportunity. I think we can all see that. I showed that the microbiome has profound effects on aging, health, disease, drug responses. I think it's an incredibly promising area. Biological aging is one metric for wellness. I think there's many of these. We're really looking at this a lot as what we're calling resiliency pools. I didn't get a chance to get into this too much today, but we're very interested not only in what gives you higher genetic risk, but when does it give it to you and why? What mechanism of aging plugs that in? And there's some really interesting examples of that, which we can get into later if you'd like. We can tailor approaches that reduce the chances of manifesting diseases for which people are at risk for. And we show that this gap analysis can give you clues as to what's the easiest thing for people to change in their molecular makeup as well. So AI insights are obviously going to power the future of 21st century medicine, and it's incredibly exciting to be here with all of you as Pitt takes its steps into becoming a leader in this space, which I think is terrific. So thank you very much, and happy to take questions.
If there are none, on behalf of, wait. I didn't. Come over to the microphone because it's being logged, so thanks.
It's my experience that any particular node, like genetic information or protein, they have a limit of accuracy. At a certain point, it saturates.
Yes.
So your idea of combining multiple, so there is a more possibility that this information aggregation would lead us to better results.
Yeah, I think it's a really great question, and this accuracy question is one we think about a lot. We actually just wrote a paper that gets into this quite a bit that'll come out here shortly. So a couple things. One is that when any particular data modality, when you do these statistical models, yeah, you kind of hit this accuracy, and we tried for years, the whole field, right, to develop tons of methods, and basically we didn't get a breakthrough there. So there's a couple things. One is that if you take data, the simplest one is if you take data from different pockets, if your errors can be as uncorrelated as possible, even like just simple majority vote, if the errors are uncorrelated, you can go to like arbitrarily high accuracy. So those errors, the fact that they're so correlated within data sets is a big one. And then the second thing for me really is this, the N of 1 analyzer type of idea where we're doing reasoning. Because the other thing that really comes up is our field, I think, has been a little too myopic on this, because there's really good reasons why you don't want to measure lots of data because of multiple hypothesis correction. And we've done a ton of, I actually helped write a chapter for the National Academy of Medicine on that exact topic, so I understand that very well. But if you're thinking about reasoning, all that extra information is not only other things that add to your multiple hypothesis correction, it's actually also error correcting. Like a good example of this I like is, if you're just doing matrix analysis, too many variables, not enough observations kills you. But if I gave you like a question that was based on knowledge, let's say I was transmitting a YouTube video to you, or a snippet of a TV show or something, and I wanted you to tell me, is it a tragedy or a comedy? If I sent you billions of pixels, it would only make it easier for you to do that, right? There's no actual overflow. There's no point at which you'd be like, well, that's just, it's too many, it's too much, it's too much data for me to care about. The data set is too long. That would not happen because you actually have a filter for interpretation. So that's what I think also happens when we get smarter about the AIs, is that we can break through that accuracy by actually understanding all these things in deep context rather than only running like matrix algebra on them.
Thank you, Amy.
Yeah, thanks.
Actually, I think, first of all, I got to apologize to Nathan. I call him Craig. I'm looking at Craig. I got it wrong. I think what we're going to do is we're actually going to go ahead and thank all of you for coming. If you have questions for Nathan, please join us in the reception afterwards. So on behalf of the University of Pittsburgh Health Sciences, thanks to all the speakers for sharing their great knowledge. Thanks for all of you participating in such a great way. Downstairs is for refreshments and food and an anthem.
Thank you. Thank you, Mark, and thanks everybody for the great talk. And just a simple closing remark. We are continuing this effort to improve and integrate AI more and more into health sciences. And the International AI Conference is going to be hosted in Pittsburgh in October. So please, if you're interested, mark your calendars for that. And that's just going to be not only just health care, but overall global AI event here. So thank you all for sticking to this till the end. And it's wonderful to receive you all and host you here in Pittsburgh. Thanks.